morning, everyone. This is Schmidt Ocean Institute, Research Vessel Falkor, and ROV Sebastian. As you can see, we are just coming down to the bottom now. We're at 2,199 meters. Our science team will be just with you shortly. We're going to set up, do a few admin things, and get going on this dive. We are diving just to the east of where we dove yesterday in dive 427, and today's dive is 428. We are still within the US EEZ, and we'll show you some maps here in a little while. So thank you for coming and watching. We should be in the water for at least the next 8 to 10 hours or so, I would imagine. And we will answer your questions. So let us know where you're calling in from. Ask your questions on YouTube and Facebook. And thanks for coming along for the dive.
Good morning, everyone, from Falkor. This is Science Watch Lead Steve Oskovich. I'm the 12 to 4 watch lead here, and it's just about 3:30 in the morning, and we are starting our dive at this site directly adjacent to where we were yesterday. We are on a different seamount, but at a similar depth, where we're going to be working our way up slope, similar to how we were yesterday, up the steep portion of the slope. Hopefully rocky substrate here once we got out of the soft bottom, and then start to see some deep water coral and sponge habitats. We just finished fully mapping these two seamounts, both yesterday's site and this site, a little bit earlier in the day, and set up on station. We'll be going up to a depth of 1,200 meters today from about 2,200 meters. All these rocks look really tempting, but I do want something that's a little bit, these are probably all the loose ones are here. Everything else in the wall is going to be cemented.
You want something more challenging?
Yeah, I want something more challenging. Yeah. I want to know that the rock that I took from this depth actually came from this depth. Yeah. Because these could have come from anywhere. Upslope. We're talking about somewhere in the 30 to 50 degree slope. So this is going to be pretty steep stuff today in certain places. Did you put lasers on me by chance? Thank you. Some mushroom corals. Nothing too unusual yet. Lots of xenophyophores at this site, though. These potato-sized nodules of sediment. Yeah, maybe this stuff looks a little bit more cohesive. Maybe something attached. If we can break off anything attached to maybe some of these little outcrops. Yeah, I mean, if it's not, if there's nothing in the immediate area, we can scan around. So we're gonna try and pick up a rock first. Can you just tap that to see if it is loose? No, no, I don't want that one. I want,
Break something off the middle.
yeah, break something.
Looks like we
Yeah,
got-
yeah, yeah. Can you get something?
Not that one either?
It's a big patch in the centre.
Yeah, dude.
There's no black, joint or anything. Taking it off.
I mean, it would be an ideal piece is something like. Oh, yeah. Actually, that might be really good. Can you see, tap that again?
Yeah, a bit loose.
Yeah, the one underneath. All right. Let me image it before you move it.
It's pretty soft.
Oh, okay. Then forget it. Yeah. No, we don't want that.
Alright. This is gonna be probably pretty soft as well. Looks like we can just test it.
It's good to know. Yeah. The ones on top that you touched first, the kind of like black angular ones,
Yeah.
those are the right size. I just.
That's a little firmer right there.
I don't want the angular shaped ones. I want more ones that look more loby, kind of like what you're touching now, where it's bumpy.
Yeah, this one's a little bit firmer, so I'm gonna be able to
Yeah, let's see.
do something like this.
That's a whole bunch of
You want that? I just
Yeah, let's throw that in there. Can I image it first?
yeah, get out your way.
Worst case scenario, we'll just get another one. But you know, at least we have something in the basket. So the first number, we'll wait for the sediment to clear just for a minute. And then the first number is going to be 337. Yeah. You can do the in situ photo after some of the smoke clears a little bit.
Okay, 337, rock walks.
Rock box, yeah.
Any additional tips?
Yeah, about let's see, about 15 centimeter rock broken from
2 meter rock?
yeah, broken off of a larger slab.
A larger swamp?
Slab.
That's it.
Oh, is it moving? That rock might be moving. You might want to grab it.
Is that?
The rock is moving. It's sliding down the hill. All right, can you hit the? Yeah. All right. Let's sample it now.
Good to submit.
Wait till it's in the basket, and then we can submit.
Ah. I'll get more of it.
It's broken.
Yeah, I think it's very brittle.
Yeah.
You want that little piece? Or you just wanna get the
Can
bigger
you
bit?
just grab the bigger piece?
Okay.
Yeah.
Bye.
Yeah, that's tough stuff. That's we'll still throw something in there, but we'll still be on the lookout.
Yeah.
It's tough to tell. You know, this is not really rock. This is might be different types of sediments, or you know, substantially altered, such that it might not be useful. We'll grab a piece anyway. Did that one come off of the same? I think it was the one on top. Yeah. Just since we imaged that one, we'll take this one instead. If we can, if it breaks apart again, it's okay. We'll take whatever comes. So we are taking a rock collection. First thing, we're looking for some very particular types of rocks. Mostly, we're looking for rocks that appear crusty, that have like lots of this iron manganese crust, as well as we're trying to get some rocks that might have some of the underlying basalt that could be used for dating this age of the seamount. But this, the rock that we just picked up is not ideal, since it seems to be a little bit out of place, but also it's pretty substantially altered. So we're going to keep our eye out as we continue on for another rock collection that's more in place. All right, good collection. These types of outcrops are more look like they're more in place. Let's keep going up a little bit more and get into it a little bit more. I think there's some better stuff to choose from here. Let me see, where's a good spot? We're looking, keep your eyes peeled. We're looking for something like maybe 15 centimeters at least in size. Solid looking, but also, has that crust on it. Maybe something like these, I like this one right here.
Brown one?
Yeah, lower left. Yeah. I don't know if this is one or two rocks, but even this one could be useful too. Either of these three or two.
Maybe that's four again. That's why I said set up for.
Just give it a tap. Oh, cool. Yeah, I like that one. Can I image it?
Yep.
That's a great rock. Can you, just out of curiosity, can you tap this one up here too? I don't know if this is one or two.
Yeah.
Yeah.
I
What
might
about,
be able to mess with it for a little while. Or
is there, is there one behind it? I can't tell if they're connected.
a little bit.
Kind of lumpy stuff immediately behind it. Yeah, that one's. No, I like the first one you touched.
Alright.
Let me do some sample imaging. All right. So we're gonna do another rock collection. Hold up, and with the in situ sample.
You
All
want
right.
me to take the photo or you take it?
Yep, you could take the photo. Hit the
Just
button
let
now.
me know.
Yep. Hit the button now. So yep, and then I'm gonna do one more. All right, hit it.
You did?
Hit the button one more time. All right, and now you can hit the sample storage button, and we can start the collection.
Is it the rock box as well?
Yeah, this will go in the same. This is going to be 338. This is about a 20 centimeter square rock.
$27 square block.
Yep.
that
You,
was
you,
it.
you could, yeah, you could throw it in the bucket. I still want the rock though.
Yeah, I think there is.
You can just write 2 pieces. Well, we'll say, yeah, also includes about 10 centimeters crust shard.
Marsh.
Crust shard. This is Regadrella.
What is it?
Regadrella. It's a euplanctillid. It has all these like lobe things.
I've got a little bit of trim down as well, even just to keep
I'm not sure. I thought that's what we collected yesterday, but I guess this is different.
I think it's different from the one we brought up yesterday.
Yeah. Definitely has more lobes.
Go back.
That's
I think
okay.
that
We
was sufficient.
we gotta look at it. We can keep going.
These little mini canyons running down slope here.
Yeah, like carve outs in the sediment.
Well, good morning, everyone. I'm Brian Kennedy. I'm a marine ecologist with Boston University.
And I'm Alexis Weinig. I'm a research scientist with the United States Geological Survey.
And we're your 4 to 8 watch on the ship. So we'll be with you for the next 4 hours as we work our way up this unexplored, only recently mapped seamount, about 175 nautical miles west of Howland Island in the U.S. EEZ in the remote Pacific. So we're currently at 2155 meters depth, roughly. Moving up this feature, our goal is to get up to about 1250 today. We're targeting a very similar depth range on a somewhat similar feature as we were yesterday. And this is the same depth range our deeper dive on Howland was also. So these comparative depths will give us a little bit
coral coming up.
of an opportunity to do some comparisons of these sites and whether there's any longitudinal differences within the U.S. waters here between Howland and the couple degrees of longitude further west we are right here.
I'm just gonna zoom in a little bit. We probably don't need to land for this.
Thanks, Dad.
It wasn't me. It's Logan. But yes.
Oh,
it's
thank.
funny.
Good morning, Schmidt Ocean. It's good to be with you.
It's usually me at the other end of the room, but it's not. So good morning, Logan.
Is this something that Steve said we should sample more bamboos? Yeah.
I mean, he suggested that if we saw a lot of the sand corals that we crouch through some of the bamboos yet.
Okay, so we are gonna set down. I think we're gonna sample a piece of this coral.
Mm. 15 centimeters branch point.
Okay. Not the whole thing, no. We are on dive number 9, 10 for this cruise. Count the slacks.
2, 3, 4, 5, 6, 7, 8, 9. This is dive 10.
Dive 10. Wow.
You can tell what happens from working around the clock. Days don't matter. Dives don't matter at this point. Just keep track of how many dives you're doing.
It's pizza night?
I understand. Yeah, I believe it's party on the outdoor lounge
Oh
with pizza.
yes.
Once we got a long transit tonight.
I will be taking a nap after this, and then staying up for the party.
I will be finishing my
Oh, yes.
vlog.
Oh.
I'm gonna like come in below where it branches off on the right side.
Yes, that'd be great.
We can get the shorter branch on the.
Or you want this one?
Well, I was thinking. Right
Can I
there.
pan up?
Is there? Yeah. Is there?
I move this so I don't knock my coffee over.
Like about a 15 centimeter piece with a branch would be perfect.
So
I would say this piece on the right.
Okay.
I would say right below here. 15 centimeters looks much smaller on the surface than it does underwater.
Totally a true statement. Everything seems to look smaller in the lab.
Did you get a
than it does
photo
on
of
the- I
it?
did.
Okay.
sure. Let's look out. Yeah, no, bring your spring camera back. Say again. Spring camera back a little bit.
Sorry, I had no sleep last night.
Me neither.
I know I did.
Oh, you
I'm
did?
actually awake.
Oh, good for you.
You care how it goes in?
No, whichever way is best for you.
Be pretty long actually. Like
What is this? 339.
Yes.
how bendy is this thing?
It's not that bendy. It'll break.
You want me to keep going in or am I gonna put it in the wild box pit and search it?
Maybe if we can put it in the other way, like so that see where this branching is. He just doesn't want that to be broken.
Gotcha.
But if you have to
there's
clip-
not a lot of current, so it should
Yeah.
flow fairly well.
If you have to clip the top, that's okay. And like
That's where it's gonna get risky. Stand by, stand by.
hello from Streamworks Underwater Robots Camp in Tennessee. That's fun. We're working with Sebastian, who is a remotely operated vehicle. So an underwater robot, and Sebastian's about the size of a small car. I always say like a Mini Cooper. And we send the ROV down with cameras and all of the sampling equipment, and then we're able to sit in a control room on the ship while the pilots operate the ROV, and we watch video and decide where we want to go explore.
I don't understand why that happened. Ben, you went to grab your arm. Kind of push it to the left. We're good, we're in. We're in.
There we go. Nice.
Perfect.
Yeah, you can
Alright.
you can probably just push it down with the cap. It'll.
Totally how I expected it to happen.
Alright, so that's sample 339 in quiver 16.
We're done with that, Ben. Good job. Right, we're gonna squeeze it there. It's still sticking out the back. You hear that sticking out?
That's okay. Yeah, just go ahead and as long as you squish the top in nice and tight, the rest of it'll be fine. Nicely done. We can carry on, yeah, whenever you guys are ready.
What? You have no other machine.
So on this expedition, we are focused on characterizing the habitats on the seafloor, so mostly deep sea coral and sponge habitats, and looking at the animals that associate with them. So what types of animals are living on the corals or on the sponges, and also what types of animals are predating or eating the corals and sponges. We're documenting all of this with the 4K video on Sebastian and with taking samples as we go along, so representative samples of the region as well as anything that is new to our team on board. And then once we complete the expedition at sea, we'll head back to various labs in the United States and process that data, whether it's rewatching the video to do video analysis, so actually very strategically categorizing how many of each type of coral and sponge we see, or doing DNA barcoding to figure out exactly which species of coral or sponge or associate, and then also running a couple different other types of analysis, so isotope analysis to try and figure out where in the food chain these animals live, what type of nutrients they're taking in. Looks like we've got something over here we can take a quick look at. Maybe a crinoid. Right here. Oh, it looks like an umbellapathes, maybe. I'm just going to come in and let you fly. So a black coral and a little cup coral down there too.
Alternopathes.
Alternopathes, so not umbellapathes. Okay, that's good, thank you. Can go ahead and do like we were doing yesterday and kind of chase the rocks with the sonar.
Maybe be like as you wish to me the other day, and then as you wish to her. It's like you already said you understood the person's pride, right?
I don't think you did. I think you lied. You have to say it.
Wow.
Here comes something.
What?
It's a camera.
Oh yeah. There we go. Another nice different type of bamboo, I think. Yes, please. You can take a front facing view of it. Looks like an internodal branching bamboo, maybe?
There's
I don't know, the
something
big ones.
going on in that top right, but I'm not sure
Okay.
what's going on over there. Or it might just be one branch behind another.
That's better, thank you. There's something in a branch right around here. Right, yeah, right there that we want to see. That weird clump of polyps. This looks like
But we need a couple bollards.
an area where there's been just this really strange branch growth.
Design to delivery. $6 million.
Any interest?
No, he said he
Okay.
sampled it.
I think we're
in
good on that
2017.
one. Thank you.
I mean, yeah, but like from like research, development, and design, build, delivery, $6 million. 5 or 6.
Do you want us to say that? Deb? Okay.
I never go with numbers anywhere. Schedules and numbers are the two things I don't talk about.
Get Ben to come pilot it. Crisis.
What? I don't get it.
Getting Ben to fly it.
Oh.
Priceless.
To come pilot it. That's what he said.
Yeah.
I was like, I don't know. Maybe Chris or Ben could give us some specs on Sebastian. Or
Chris.
do you have a mic over there, Deb? So we're gonna have one of our ROV pilots talk a little bit about Sebastian and some of the specs or the capabilities of the vehicle while we continue to fly along.
Is there a mic in there?
Yeah.
Where?
It's a headset, dude, just like
Oh,
the
there it is.
one
It
you
is.
saw the
I see it.
Oh, looks like we have coral out here. Brian, your coral radar is
What?
on point today.
I'm actually awake for the
Good
most
morning
part
or
of the
evening
day.
to whomever's out there. My name is Chris. I'm one of the pilots for Sebastian here on board Falkor. I'm just going to give you a little bit of an overview of the sub. It is rated for 4,500 meters. Some of the parts on it can go deeper, but as the max we will go is 4,500 meters down. It is a hydraulic electric vehicle, meaning that we send voltage down through the umbilical and operates
This looks like Zoe
an electric
that's
motor and
going
then a
over-
hydraulic pump.
or
And the
Zoe
hydraulic
that's on
pump
a
functions everything
dead
from our
stop.
thrusters to our manipulators
I
to
think
various
there's
valves
one
for
right
different
up here, Ben,
purposes.
that we'll take a
And
look
then we
at.
have
That's okay. This
the
is
electrical
good.
aspect, which is transformed down to operate again, like I said, the electric motor, but also
This right here.
our lights, our cameras, and all the other functions. Assuming that we did our pre-dives correctly, Sebastian can stay down pretty much as long as we are capable of staying in the
still
pilot
in
seat, or
difference.
until the scientists need us to come back up. And from research development and build and delivery, I think Sebastian probably cost around $5 or $6 million, but don't quote me on that. That's just what I believe I remember. We have two 7-function arms, one port and one starboard, as you've probably seen us use.
That's pretty
We have our
strange
starboard
to me.
set up
It's
with
like a
also a manip cam that we use if
stolen
we're in
from
tight spaces and
overgrowing,
need an extra look.
because this is all
And it also
on
has
the
our
base.
coral
Do you see
cutters
the polyps
with
down?
the soft jaw red tips, if you've noticed those. Helps it makes it a little easier to collect things that are a bit more brittle.
Uh-oh.
And we are capable of hooking up
You wanna poke it?
a vast amount of different
Okay.
scientific equipment depending on what the
Can
job
we
requires.
set up to, yeah, we're gonna first just
look-
touch it with the arm and then we
and
might
then-
end up sampling it.
We'll have to do some work in a second.
It looks like soloniferous, right?
Yeah, I think it's anthophylla though.
Let's see.
In which case it's.
If you have any other questions,
Close
just let me
family
know.
of Victor Gorgia.
Oh,
Sister family. They're closely related.
Are you into the question? Yeah.
we're first gonna poke the coral.
There's a little cup on the other
That
side
might be
too.
if
This
the
might be
polyps are
enough,
closed,
yeah.
that might be enough to get a taste.
It's on a precarious little spot.
You mean perfect?
Big sea star. Right?
Yeah.
So we're on a slightly different aspect of the seamount, which might be why we're seeing different species. At this depth, it's actually kind of surprising that it's so dramatically different.
We really need to get Randy to put a screenshot in the what you call it, the dive plans.
I think that just means whenever retrieving the waypoints, you just take a screenshot on that computer. Is that right? Okay.
Yeah.
Okay. Well, it's a little. Can you send the camera out? Yeah, perfect.
I don't want to touch the trunk.
Well, we might be able. This might be good enough for
Thruster
us to
wash
see.
might have been enough.
There's a crab in there.
Oh, there is a crab in there.
Yeah, I don't think this is a still enough for it. I would like a piece of it. Because
Okay.
I think it's Anthophila or something similar. And the crab, maybe for sale.
Yeah, yeah.
Spell that?
Okay, so we are gonna want to sample this. There's a. Did you see the little crab right in there?
Right in the middle?
Yeah.
Okay.
So if we can sample below that, we can just try and see if the crab stays on.
Okay.
And put it into a quiver.
Spell Anthophila.
ANTH.
Might have to take most of it, which is okay.
I'll see.
If we need to take lower on the colony, if you need to take more of the colony than less, that's fine. So just take lower on the branches.
Yeah, if you can off spin, that way I can actually see. Well,
Yeah, I think I shot myself in the foot yesterday. I had a coffee like right before dinner, and then I couldn't fall asleep after. Which is tough when your wake up is 3:30 in the morning.
And are you 5 plus on ship?
On the new one or on this one?
This one?
Yeah, with the seat map.
Yeah.
What are you talking about, your
Winch.
winch?
The one that
Yes,
references
are you?
it. 5. Is that 5 plus or 5?
I think it's a 5, but I cannot know, I do not know. It's the one that was here before, right? You guys didn't get it, it was the one they used for the helicopter.
That's right, yeah.
Yeah, it's already 5.
we have porch out. It's a lot of Say it again.
We don't actually use it for that.
I don't know how it's titled, I don't think so.
We don't use it for
No, it's vessels and compensation.
games.
think it is tied into that, but I'm not sure. You don't turn it on. That too.
No.
Julian Day.
I got the off deck one right, but the in water was mad. 12 to 36, so.
So there's a question about how many pilots does it take to operate Sebastian. There's 2 ROV pilots that work in each. Wow. Okay, there's 2 pilots in 2 different chairs. A pilot and a co-pilot, but there are
7 of us on board.
7 pilots on board since we're working 24-hour ops. So there's 3 of them that are on midnight to noon, and then the other 3 start noon to midnight, and then there's 1 member that is from 6 to 6 so that he can provide some continuity between the 2 different shifts and be an extra set of hands for both of those shifts.
And then there's additionally 3 more people in the control room. There's one of our marine technicians for the ship who's operating the streaming and some of the data logging, and then there's 2 of us scientists sitting here kind of pointing and asking them to do all the things.
There's 23 full-time crew members on board.
And there's 23 full-time crew members on board, allowing Falkor to operate 24 hours a day, including multiple, are they called officers?
Yeah,
Bridge officers, and
Officers.
many members of the galley and the stewardess team that keep everybody fed and happy. Very important part of the team. Plus members of the deck crew, and
I'll have to come up underneath it if you want to try to keep that crowd.
you can go ahead and snip it down here, Chris.
Yeah, so we'll
Yeah, that's fine.
These chefs, what you
Engineers.
Engineers, which are very important for keeping the ship operating.
Ah,
It's okay, he's a swimmer.
I can try to get him with the,
Suction afterwards? Yeah, we can try.
this is gonna be big too.
If you need to put it,
You can jam it in there.
I just,
it's bendy. What did we call this?
Bendy.
amplifier. 340.
340. I'm not gonna spell this right on my sheet.
Steve, you should just set up shop in
I know.
I tried earlier. He told me the
I don't
TV was
like
there. You
better.
got yourself in a live.
I don't like loomers. I don't want to be a loomer.
You would just be part of the team.
Then we just don't hear your Slack go do do do.
Just put it on silent.
Then I
No,
miss
but then
it.
we don't know what's
Then I
happening.
miss it.
Yeah.
Yeah.
We need
that
more.
needs to be
We need a
over.
few more screens in here, I think.
Do you want us just to wirecast your laptop up there so you can work?
Should I use WhatsApp instead?
I wrote anthophyllum. That's not right. Anthoptylum.
No.
Can you say it?
Anthophylla.
Gila. Like that.
T.
The TH.
T-H-E-L-E.
Wait,
Antho.
T-H-E where? I can't.
Antho.
Like anthro.
T-H-E-L-E.
T-H-E-O.
No. Oh.
Jeez, I can't spell. It's like,
Antho.
stop, just give me the letters. Stop trying to make me sound it out. I failed this in first grade and I still fail it.
A-N-T-H-O. T-H-E-L-A. Yeah.
Okay.
I mean, there's a slight chance it could be like some really weird white, pale, like Victorgorgia, but they're so closely related.
Oh, okay.
In fact, let me see. It was recently revised that several Anthothela were moved over into Victorgorgia, and some Victorgorgia were moved back to Anthothela.
Are there Victorgorgia that aren't purple?
It's possible.
Oh. That destroys my like single
Single identifying
Identity
characteristic
feature,
for
yeah.
that entire family. Yeah.
Pretty much.
Yeah. No, they can be pale, but they have the same skeletal type, which
Yeah, they
is
have.
an indicator.
Yeah, and they have those really fleshy
Yeah.
polyps.
Keep going. I just want to see past this. Keep going. That's good right there. That's good to see past that.
And if you notice, like, you know how we noticed it was a stolonephrine, or
Yeah,
it looked
or
like
it
a
had- yeah.
stolonephrine? They all kind of run a little bit along the rock around
Okay.
the colony.
What part
That's
two
what
do
looks
you
like.
want to do? That's stoloniferous?
Well, I
No,
think
no,
no,
I
I think
think
that's
that's
the
what-
same thing.
That's what that is.
Oh, it's the same thing.
Yeah.
Okay.
That's like it's starting out.
If you look at Victorgorgia, sometimes it like the polyps run down, covering the base of the rock.
Yes, you're right. Nicely done,
Alright, now put this on and I'll grab the,
slurp.
get this and get us ready to grab. What's up?
Oh, those are backwards on here for us.
Yeah.
Those numbers.
If I could catch it, put it in
Last
there.
time it was.
Yeah.
I'd be very proud of myself, but I don't see that happening. Why not?
Same with that. One. 7, 8, 6.
I got faith in his swimming ability.
Is he still on the rock? Do we know?
It's a good variety. Swim it, see.
Oh, I see. Yeah,
He's still there.
he
Yeah,
is still there.
I'm not even looking. Can't see that far. I can't,
So we don't have a set number of samples as a goal or anything. The vehicle can accommodate has 24 quivers, a rock box, and 3 bio boxes, 6 Niskins for water samples, and 7 jars in the suction sampler.
If you pan up, it's
And so we're kind of just collecting the species we need and trying to minimize the samples as possible for identification and some experiments we're doing on board the vessel. When we're inside the monument, we have a few more limitations to the samples we can collect. But here in the US EEZ, we don't have a specific number we're limited to.
I guess that's it. Zoom in. Same way. Go. Go. Go for it.
Skin move?
Past them. Look up. Look up.
Do you want me to zoom out on this?
I don't know where he is, so.
It's okay.
What, you got sticky thing?
Yeah. To the left of the line.
I think he may have a. Oh,
He's
he's
still
dumped
right
on
there.
the rock, but I don't know if that's too far down.
Where is he?
Right
Oh, I
at the
see.
base
Okay,
of the
okay.
science
Yeah.
camp.
We can go back. No, no, no. I mean, right now. We can better be.
His escape mechanism is going to be to fly backwards.
Yeah.
Not that far back. Can I go for it then? It's on.
Yep,
I think we can. I think he's gonna just.
he's going to elude us.
Yeah.
Back in reposition, standby. Reposition.
Now it's a challenge.
Yeah. Yeah, it is about getting kind of like either right on top or behind it, the crab, though. Oops, sorry.
I get that far, dude.
Get up a little bit.
Yeah, I think I go that far. It's way to the right. Way out to the right. Down. See that stalk that was to the right? They're underneath that.
There's a cup coral right there, too, I think.
Down. Still down? Yeah, it's still down.
So I made a mosaic last night of the Colotrophora
That's right.
field, and it worked pretty well.
Oh, you made a mosaic of that? How, like the whole thing?
No, just the north-south pass we did.
Go now.
But I think I could do a lot more if I curated the video better.
He swam away, dude.
Yeah, I lost him. He was. I agree. He was right there, but he's gone.
It might have been just the ship. However, since you have your section sampler out,
Uh-huh.
this cup coral right here, see on the,
You want it?
yeah, we'll section that. Don't want to,
Picture, get pictures of it.
yeah, let me zoom in. This does look different than the Javania that we've been seeing and sampling, so. You good? Okay. Okay, we are good. Crab lives to see another day.
See something like that, it's quite better to capture that. It's cool.
Mm-hmm.
That's what we did with the shrimp.
To the right, could be about 4 feet long.
Am I too close?
Might be a little bit.
Mm.
It's all right.
Yeah. Too close.
Right. Secure.
Alright.
That should be
Just watch it. Yeah.
All right.
Alright, go for it.
Thumbs up.
Nice.
I don't know if it went in.
Oh yeah, just make sure it.
It might have gone to the side, but we'll see in a second. Alright. Come on.
There it
Got
is.
it.
Oh, you got in? Okay.
Yep, it's in. Jar 5.
Anything else before I put this away? You wanna?
No, I think we're good. That was 341.
I looked over your shoulder. I didn't,
Yeah,
but,
that's okay.
Yeah. 341 in jar 5.
Cup of coral.
Alright. So let's start that.
The other coral, yeah, at least on our sheet, the 12 and 13 are switched from what they normally are, just so you know.
Okay.
The quiver, so it was in quiver 12.
Alright, so 13 on my side.
Yeah, yeah. We'll keep carrying up slope. We started the dive at about
2200.
2,200 meters, and we're at 2124 right now. There's a big sea star. Some whipped bamboo corals. Some interesting feature. It's like a little ridge. Camera up some. Do you have your cool new overlay on YouTube, Deb? Oh, I mean, you don't have to. I think that's a sponge. It's a Walteria.
Do you wanna do the where are we overlay?
Yeah, Brian, you wanna, Brian?
What?
You wanna, she's gonna put the overlay of where are we in the world?
I'll switch the screens.
There's a big
That's
hymenaster,
a big one.
slime star.
So we're operating in the U.S. exclusive economic zone around Howland and Baker Islands out here in the central Pacific. From where we are right now, we're probably about close to 1,800 nautical miles from Honolulu, and we're way on the west side of the U.S. waters, right almost at the international dateline. And so if you zoom in, all the multicolored stuff you see is mapping we did over the last 2 days. So this feature there was one little transit line of data from another ship, but basically completely unmapped until we got here. And so we did all that mapping in the last couple hours, and yesterday's dive was on that more northern feature, and then today we're on the southeastern feature where Deb is pointing at with her cursor.
And now we have our dive or our bathy data, so the mapping data, and you can see when Deb zooms in that where we are on the feature that we're diving and how far up the feature our track is planned. So that black line, that little yellow box at towards the bottom of that black line is us right now. That's represents Sebastian. And then we are moving up this slope. Will probably take us roughly 12 hours of bottom time to complete this dive track, but plus or take a couple hours depending on what we see. And like Brian said yesterday, we dove on the Seamount. You can see that red line that is connecting to the other Seamount, so that was where we dove yesterday in a very similar depth range, just on a slightly different orientation of the other Seamount.
Looking at these tracks give you a pretty good idea, though, of how little of each of these features we see. So both of these features will have one dive on them, but when you think about that track line, at most we're seeing 20 meters, 30 meters in either. But really, it's probably more like 10 meters when we transect up these things. So it's a really pretty small amount of geography that we actually document with the ROV on each of these trips. So while we think about a site being that we've explored at least once, it's really still just scratching the surface of what actually might live on that seamount. We could be missing huge coral gardens or new species, and they could live there in great fields, and our one track line up one ridge of one side certainly isn't giving us a full picture of everything that lives on that feature.
Is that exaggerated, Deb?
Just one.
Just one? Okay.
Yeah, that's really more. I went up to like 2 times.
Oh, gotcha.
When I was
Yes.
zoomed out, because this feature over here looks really the larger of the two.
Yeah.
Looks really cool with all of these like.
It
various
does, yeah.
edges. Go on home and see what like. Sort of weird. This I don't know. Just these weird sand baby mountains.
It is. It's a very long feature. How long is that thing all the way from one side to the other?
About 30 kilometers.
Oh wow.
So, measure here. Is about 16 nautical miles long. And 7 nautical miles wide.
Okay.
It's more square.
Yeah. Sorry. Umbalula.
Nice.
I was watching Deb's screen.
Yeah, so I realized we both were.
I'm gonna just quickly zoom, see if we can. That's a crinoid.
It's yet another
Is it?
stalked crinoid. Yeah, I think it is.
Oh, okay. That might actually be umbellapathes. What I said about the other coral before. Yeah, umbellapathes. No, it's okay. We can keep going. It's very like rocky, not so much the large boulder outcrops or the lots of looks like tumbling down broken off pieces versus cemented. I mean, I say that, who knows, these might be cemented. There's a couple umbelulas. You're good. We can keep going. Yeah, people in the chat really like the overlays and seeing the kind of, I think, where we are in the world, and then the graphics from creating the maps before we actually are able to dive. And that's one of the benefits of like modern day research vessels, including the Falkor, is that they're equipped to not only have a really well operating ROV that can do these types of dives, but also the technology to map the seafloor and then process that data on board before you're actually able to dive. It's very botryoidal rocks.
Good use of the word.
Lots of crust and crust over probably some type of igneous rock that was laid down a long time ago by volcanic activity.
So you hear us talk a lot about crust, manganese crust, polymetallic crust out
Take a
here.
look at this right
and,
here, Ben. I'm just gonna zoom.
The water chemistry in this part of the Pacific is pretty interesting
Okay.
in that
Okay.
it's super mineral laden to the point that it actually precipitates out of the water to form this mineral encrusting layer on top of the original rock of the seamount. So this botryoidal, this kind of small ball rounded shape here you're seeing is indicative of old crusts, meaning these rocks have been exposed for millennia and have grown this layer of hydrogenous precipitate on top of the rocks. And there's a lot of minerals in these crusts that could be commercially viable for extraction. And so when you hear about deep sea mining, most of that's talking about polymetallic nodules, but there's
Which are
also
not-
some
we have not seen
which
here.
we've not seen here.
Yeah.
But there is also some concern that of these areas being targeted for crust mining as well.
This is an urchin. Since he sat down, I figured I'd take some good imagery of it. I don't really. Are those its 2 feet coming up from underneath? you
That would be
think?
my interpretation, yeah. So you can see the
Interpretation.
pentaradial symmetry of the arms still
Yes.
there, where you see the arms come up where the 2 feet are, where what used to be kind of a common ancestor with a sea star that ended up changing into these solid
That's good.
test.
You can keep going. I haven't seen that color urchin yet.
Yeah, that one doesn't look familiar to me either.
We did already sample some rock, but I'm assuming these are all really cemented, so it might be difficult.
That particular spot, yeah.
But I know this is what the geologists like.
Yeah, I think the rock that Steve got was pretty good.
Okay.
A chunk of crust fell off, so it's nice and
Oh,
crusty.
good.
I think we should get a
Yeah,
few
we were
hundred
looking
meters
at
shallower first.
one of the rocks from a previous dive where some had chipped off, or had a coral base on it, so he chipped that off, and like the crust was like almost 5 to 10 centimeters thick.
Yeah, and I forget the exact rate of deposit, but it's something like 1 millimeter every
It's like a million
years, I think.
Yeah.
I mean, it's an insanely slow process. And when you see these crusts that are super thick, it's an amazing thing when you think about how many years, millions of years, these things have been exposed to the seawater. The clicking sound is our Slack alerts on my laptop. So Steve is sitting in the library, feeding us information, notes, and identifications as well. And so every time you hear that clicking, it's my Slack notifications telling us that someone in the library right now, it's Steve, is
This is Anthomastus.
giving us some additional information.
No, we're good. Sorry. I'll tell you. I'll try and point out with the stick if we need to go look closer at something. No, I'm a nice scientist. Yeah, we have so we have 8 participants on the science team on board, and so we're working 24-hour watches, which means, or we're working 24 hours a day. So we're all working different shifts, but we have many places on the ship that if you're sitting, you know, in the mess, or if you are sitting in the library, or in the dry lab, or in the wet lab, the video from the dive is still playing. So we have a Slack channel. If you're familiar with Slack as like a workspace communication device, we have a Slack workspace just for this cruise. So every dive, we create a new Slack channel so that people can communicate with each other without having to always run into the control room, which is actually pretty helpful. I think it's worked really well on this cruise.
Yeah.
In 2017, we used Slack also, but it wasn't quite as... People didn't have quite as much experience with Slack back then, so I don't think we used it super effectively. I'll take a zoom on this little coral right here.
Yeah. And that fish is a type of species of halosaur. Try and figure out what this
I'm
coral
just going
is.
to come in a little bit, and then you can fly in.
So wet lab, dry lab, what it sounds like.
Pretty much. The dry lab is where our computers are, and we don't like water, and our wet lab is where we put the samples that come up and splash around and have fun in there in the water.
Splash around and have fun.
Just not a lot of splash.
I mean, it's true.
Sounds like a kiddie pool.
I mean, the giant ice bath we put the samples in kind
Yes,
of is.
you are right. There is some splashing. I'll make sure to splash you tomorrow, Brian. That's a
Oh,
pair
pair
gorge,
gorge.
yeah. You can tell
Enough
because
wind
when
for
we,
you?
yeah, when it gets hit by the thruster wash, the coral the skeleton bends. That's good. We can keep carrying on.
So
Had a little Ophiroid on it too. We didn't talk about that, but associates are important too.
So we actually don't have a geologist on board, but we are working with some colleagues at the U.S. Geological Survey who will be looking at the rocks that we bring back. And
Small little.
I don't believe that they age the rocks using the crust, because the crust is continuously being laid down and the thickness changes. You don't know when the rock actually became exposed to the seawater. There's other types of radioisotope dating that you can use to age the rocks. So when you think about aging a rock, that's usually like a uranium or some form of isotope dating that gets back that kind of millions of years. Same thing as carbon dating, same principle, just using a longer half-life element.
I'll look back at yesterday's notes before we started. That's interesting. Can we take a look at that purple blob?
Zoom the purple.
Blob zoom.
Yeah, you heard me correctly. We're looking at the rock structure here. Is the crust is millions of years old, and the rock is tens of millions of years old. I believe this part of the
This is definitely I know Steve's typing. This is a purple orb that we have seen before. Oh, and take off.
it's a gastropod.
Oh, cool. You can see its foot underneath there now
Yeah.
that it's... Yeah, we can just keep going.
Never seen one quite like that before.
Does it have a velutinid? Does it have a shell? Do you know?
I mean, I think that purple thing is the snout because it's got a pretty obvious separation to the foot.
But that purple thing is like gelatinous. That's not a shell.
Is it?
What is it, Max? What's in here?
It's some type of gastropod.
Oh.
But I'm saying it just doesn't have a hard. It doesn't look like it has a hard shell. It has like.
It is kind of transparent. That's different.
Yeah, we can keep going.
My lab safety training expires.
Oh. It's fun to do at sea. Looks like we have some corals right up here and a sponge that we'll take a look at. Couple different kinds of coral. I'm gonna want to zoom on this one down here, Ben. Thanks. Hi, sir.
One of those unbranched whips down below was Candidella gigantea.
Oh, is that a big coral over there? It is.
Yeah.
We'll go look at that after. It's in like
This
the
one?
smoke screen. Sorry, what'd you say, Steve?
Those unbranched ones, which actually look branched, which is kind of silly.
These right here?
Yeah, that's Candidella.
Oh, you do? Okay, so we want to sample that?
Yeah, Tim wanted
Okay.
a piece.
So if we can set up to where we can zoom and then sample, that'd be great.
Yes, I'm throwing him under the bus just to get some more.
Nothing new there. I love this, like, how he's looming in the background. And that very tall sponge,
That's
too.
a Walteria. Look at that.
Is it a Walteria?
Yeah, look at all those little projections.
Oh, yeah. Oh, I'm sorry. Which way do you need me to go? This one, move it a little bit to the left. Okay. Okay. Okay, thanks. Thank you, Russ. Got two corals here. One, Steve is saying, is Candidella, which is a unbranched primnoid octocoral. And then there's also a little chrysogorgid right next to it. Let's get it tight in on the polyps.
Really weird polyp arrangement.
I think it's having a hard time because looks like you've got polyp pairs
counted those three per whirl, but I'm surprised to see.
Some of them are.
Yeah.
Every once in a
I'm
while.
surprised to see that 2 dominates here, which is really unusual. Maybe just the conditions. Or something. Or it could just be a younger colony.
So do you want just one piece of this,
I
Steve?
think one whole one. Yeah,
One
it's-
whole piece. Okay. Let me just take a look at this coral up here before we do that. There's a barnacle living at the base there too, or just next to the colony. I don't see any squat lobsters. Normally these have an associate crab living inside the colony. This one looks to be lonely. Okay.
here as well on the way up.
On the way out. So there's those big colonies over on the other side of the
Over
rock.
more?
Yeah, so
Over
we'll
there.
go look at those and then we can do a Niskin. Can we just take one of these branches, one of these long thin branches,
Okay.
Russ?
Just a second. Do you want it a little bit quiver?
Yeah.
So it might be a bit long for the quiver. Do you want a whole piece or do you want it to
They're
really
bendy.
quiver and cut in half and
Yeah, it's fine. It doesn't need to be whole. You can cut it, or you can just bend it and squish it down. They're really flexible.
So one quiver length.
Or just, yeah.
One
I'll
quiver length.
figure it out.
Yeah.
I think it'll fit just fine in a quiver.
Okay.
Here, Dad.
Yeah, give me your mugs, because we're running out of mugs down there.
Oh, yeah.
Where is this? This is.
I don't know what that is.
This is.
If you haven't taken a look at the new post on the Schmidt YouTube video yesterday, they released a really nice, heartwarming Father's Day video with featuring a couple of the dads on board, including Steve and one of the ROV pilots, J-Rod. Puts a little bit of a human perspective onto the science and the ROV operations, which is always nice because it is, you know, we come out here as scientists and as ROV pilots or ship crew, but everybody's got somebody at home, so it's an interesting perspective to watch that video.
That'd be okay.
Yeah, that's great, Russ.
So a question in the chat about how do the corals decide which rocks to live on? That's an excellent question. Frankly, we don't have an answer for you. That is something that we are still very much puzzled by. And I can give you answers about, you know, current flow and sediment load and potentially microbial biofilms and stuff that all may play a role in the selection of where something lives, but frankly, we just don't know at this point. We know all those things kind of contribute, but to what degree and which are more important, we really don't know at this point.
Yeah, and same with like larval dispersal. We have very little information about how most of these corals, how their larvae
You just back camera.
disperse and where they settle. So we don't even know where the larvae are coming from. In all honesty, for most species of deep sea corals. Oh, this is backwards too.
Well, I just need to get
Or why is
back
this?
into this.
Did I mess that up? Oh, I messed that up. Sorry.
342.
Yes, 342.
And quiver 15.
Yes.
And it's a primnoid.
Yes. Candidella. C-A-N-D-I, I think. Candid, D-E-L-L-A. D-E. No. Great. So then it looked like when we were flying to the right around the rock, there were, yeah, a couple big colonies.
Sure, it's good to me. Make it happen, Ben. Engage.
Engage.
Make it so.
ROV, go.
Go catch RV.
Go, go, ROV. What was that? Go, go, catch it. Remember that? Yeah, we've got some nice big ones over here. Really big Walteria sponge. And then also a nice large coral, probably looks to me like another bamboo coral. Oh, and a Bathypathes. And an Anthomastus and another larger coral.
I think we're gonna get cold. And actually,
Collect some of these too.
We're probably not gonna collect this one, but we will take a look at it.
And fire a Niskin.
Yes. Yeah, so if you are gonna park yourself, then we can fire a Niskin here. And then I'll take some zooms. This is a pretty fragile colony, so we just wanna do our best not to interact with it too much. Yeah, it's okay. Oh, Chrysic origin. Can I zoom?
Yeah.
Okay. This is another, look, Brian, where there's weird
Yeah.
branching and it's very strange, like brambly polyps. I've not seen that very often.
Look at that damage in that there's a little break in the tissue just distal to the
Yeah.
weird branchy polyp.
It looks like almost black too on the other side. See that?
Yeah.
I wonder if that's. I don't know. We'll look at this one up here. Most of these polyps are all retracted. It's cool with that sponge in the background.
Yeah.
Steve, if you're listening, have you seen that kind of strange
Answer is no. Steve's not seen that before.
Okay. Most of these polyps are all retracted.
There's
a
one squat lobster.
Oh, those There's a branch of open. He's very muddy for being up on this coral.
Steve is suggesting if we can get a piece of the bramble,
I don't see it.
but I don't see anything close to the tips.
Yeah, so I'm not.
Unless there.
Oh yeah, well, it's still, or down here actually. Let's see. Yeah, here we go. So Russ,
Yes.
we do want to collect a piece of this.
Yeah.
Even though
Go back in on that with me.
Huh?
Go zoom back in on that with me.
I will. Okay. We're gonna cut just like right here on this branch. See this weird clump? We want to include that in the sample. So I think
Yeah.
if we just
See what range we go.
Let me just- sorry.
Yeah, well, range, but,
So good.
Figure it out. What you know.
there's one question in the chat of if deep sea corals ever been observed releasing their young or like releasing gametes, and the answer is yes, in certain parts of the world. So up in Norway, in the fjords of Norway, there's a more known reproductive cycle where they do know that when certain coral species will spawn. Some of that is those corals are a bit more shallow than these corals, even though the water is still very cold.
Well, yeah, just be really careful. So, it comes to light.
But for the most part, we have not seen most deep sea corals. We have not seen them spawn or release their young in their natural habitat. That is something that if we ever saw while diving, I would absolutely freak out. We've also kept corals alive in aquariums for quite a while, a couple years, certain species of coral, and in that time, at least in the conditions we had them in, they seemed relatively healthy, but they never spawned over while we had them in the aquaria.
Take a good shower next one. I just wanna see the best place to cut in. So just cut in here, if that's okay?
Yep. I think just right in there.
Just, leave it just as is. It's okay, yeah?
Yeah, if we can come a little closer
Yeah,
to
I mean,
the.
if that's easier. Does it
is-
matter?
No? Okay.
Is that okay?
Yep.
Just that it's easier for cutting, is that
Yep,
right?
that sounds good.
Yeah, thanks.
Express.
Okay, right, can you, punch out a little more?
There's a question of if the ROV pilots have more views to look at than just the science cam, and the answer is yes.
No, we just did it because they're amazing.
They are amazing, but they use multiple cameras to really be able to. What before we move off the rock, we can fire a Niskin.
Right, okay. Haven't fired any yet, right?
We have not fired any.
Okay.
So either one
Large
through
one or a small one?
large
Large.
one.
Yeah, number one, so. So you back happy back there?
What's that?
You back?
What's that? And how we go.
One side. That's it. So one leg,
Are those numbered differently?
HR trail.
Oh, is it?
You get in the,
Yeah, one's fired.
One's fired, yeah.
Yep.
Yeah.
Okay. Okay, that's the arm skill. 14.
Three 44, right?
Yes.
Is there anything else you want to
I think not where we're parked, but if we continue to go around that wall, there was more to come.
Russ or Ben, there's a question about how the arms are controlled. Do you mind sharing a little bit about that?
Yeah.
Just Deb, can you use that microphone?
Yeah, just mute you guys
Okay.
while he's doing it, otherwise.
Yeah, so we had a question about the arms. So basically, on the ship we have a controller which kind of mimics the shape of the arm. So it's a miniature version, so we can actually manipulate it's kind of like a advanced joystick, kind of speak. And so what we do up in the top will actually mimic on the arm in the sea. So basically, we have got all the functions and all the joints, so we can move the top side, and whatever we do on the top will transpose into the arm and subsea. We can adjust settings to make things more sensitive or less sensitive depending on what we're doing. But yeah, that's basically how it's done. So yes, it's kind of like a small joystick on the top, and then it controls the controller in the subsea.
And there was a question about what are the containers with the numbers on the top when you see the porch come out. Those are sample containers, and the numbers are so that we know which container each sample goes into. I'm not sure what the red stuff is, but that's just a way for us to be able to record where each sample goes, because we have all the information from the vehicle associated with the sample. So all of the environmental data that the vehicle is collecting, and the location and the depth can all be linked back to a timestamp. This looks like a hemicorallium to me, like a corallid.
There's enough thruster wash flying around that I think
Yeah,
it'll
and
be
it's
moving.
not very, the polyps are not very dense. It looks like it's kind of struggling, actually. You can see the tips don't have polyps on them. So it might be a little bit too sedimenty here for that coral to really grow into a large colony.
Huh? Yeah. Yeah. I was just gonna show them all the cameras, Alexis.
Oh, okay.
But for us to write down.
Is that all you needed on that one, or do want to
Yeah,
close
I
it?
think that's fine.
Okay.
Yeah.
Yeah.
I think we can, kind of keep going around this feature, and then we'll come up just from where we are. We don't have to go back to the very specific track we were on before.
Back on.
Yeah, we're operating on 24 hours, so our dives are not necessarily, we're not going in the water at the same time every day. We're going in as soon as we can after we have enough mapping data and we have time to plan the dive. So some days that's going to be we're going in the water at noon, and some days that's going to be we're going in the water at 3 a.m. Just depends on how much transit and mapping we do in between the different sites. Oh, much more sediment here.
Am I seeing it? You guys aren't seeing it.
Yeah. Looks like a stalked crinoid.
Okay, Alexis, I'm gonna kick over, so that there was a question about cameras. This is now every camera that you could possibly see on the vehicle right now.
Okay, so Deb has put up a quad screen of all the different cameras that you can see from the ROV. So there's at least 4 different cameras used to be able to determine where we are in relation to what we're looking at, as well as where the arms are and what the back of the vehicle is seeing, or the bottom of the vehicle, really, I guess. So there's a lot of different cameras. It's definitely not just the science camera, which is what we focus on and stream most of the time. But the other cameras aren't necessarily as high definition because we're not using them for specific identification. But they're very helpful for, not running into things. Thanks, Deb. Another big bamboo coral, similar, I think the same type that we've been seeing. They seem to be doing very well in this environment. Yeah.
I had it on the quad and I was like, why do I have two and you don't have two? So I just turned it back on the mom again.
Awesome. My favorite HD stuff.
Yeah.
Was a scripted one 3 and 4, so it's
Yeah.
intended, but it's
If
my
you wanna
favorite.
try the other ones, you have to put them back.
Hold on a second. You just put,
Yeah.
see what you got. I'm just gonna be for a minute, so what about it? One still doesn't want to play.
You get your own faults, but
That's all.
We gotta go.
Yeah.
Well done.
That's good.
Yep. Yeah, it's 1 and 4 still.
I like two though. I agree, it's the easiest to like see where your arms are to the tray. Very helpful.
Percentage of the sea star.
They weren't like just
Is that that brambly
Let
sponge
me
we
see.
sampled the other day?
I'm gonna take a look at this right here, Ben. Just come in a little bit. Yeah, it does look like that, doesn't it?
Yeah, it's that branch.
It's very
sponge
weedy
thing
looking
we saw.
sponge, yeah. Is that good for you?
Right? Yeah, I'm pretty sure it's the same thing we sampled. so-
Okay.
what better with that with what better lighting?
That's great. We can keep going up. Yeah. Sorry.
That's easy.
Oh, it's not attached.
No.
That's cool.
Yeah, yeah, of course. It's cool, great.
Were you up for the end of yesterday's dive?
No.
Me neither. I was gonna ask if it changed from what we were seeing, but I think we both went back to bed.
Yeah.
Oh, here's looks like to the left is a fallen. Can we just take a look at this over on the left side? It's like a fallen over colony. I just want to take a little scan around it and see if it all looks completely dead yet.
No, one of my favorite parts about the 4 to 8 watch is my post-breakfast nap.
That's true. Yeah, so this looks like it just either the rock tumbled or the coral snapped off. You can see there's no live tissue left on the coral, but it's been starting to be colonized by hydroids, and look like there's a couple barnacles on there as well. Oh, there's a little sea pen right here. We're gonna take a look at it.
Oh, okay.
Oh, I see.
Oh, okay.
That'll be quicker.
This is probably something we could sit down for, because we are gonna try and
She
sample
got
it.
a stud in with those two pinnacles, yeah. That one there, that one there. That would probably be ideal. Sounds good as well.
This looks to be like the elusive, I don't know, runner sea pen, and that when you try and collect it, it suctions itself into the sediment. Like it retracts in.
It's a wee sticky.
It's-
It's good as a beastie.
It's a slippery little sucker.
Yeah.
This is related to the soccer team. I think, is it the first time?
So there's a couple. We have that push cord, right?
Yeah.
So you could try and do that, or you could try and suction it. I just don't know if if you're just only gonna get sediment,
I'm just
if you do
wondering
that?
about the push core, sediment wise, it may not be that deep,
Right.
but give it a shot, like.
Okay.
So-
Probably go in and then scoop the water you get or something. Okay.
For those of you watching at home, to catch you up, we've been attempting for multiple years to collect this little sea pen, but it has this very good escape mechanism. When it's disturbed, it sucks itself into the sediment,
You get this.
and it's become quite it's become quite the challenge to find a way to collect one of these. Steve just messaged in our Slack channel that he's going out on deck because he doesn't want to watch.
Shot that one and touch it that one as well.
Chris, I think, is somewhere. He's on watch right now too. The ROV. Oh, there you are. It's a very emotionally charged collection. Oh, we're still moving. Sorry, I'm sorry. I moved the camera.
Just now, touching that rock. Right there.
So you got this, Russ, you got this.
Am I good for Zoom? Okay.
Please be good away. I'm gonna core it.
Didn't we get one of these in summer? We did get a sea pen at some point.
We've gotten a couple sea pens, but not the
No, I mean a different expedition for it.
Not the rapid,
Oops. Hey. Got a good
Yep.
image. Okay.
How deep are the roots normally?
Don't really know. That's the part of the.
Right then, I think a core is the way to go then.
Yeah.
Even if it's like sand.
Maybe.
Yeah, you can kind of do your best to scoop.
I will get watching it. The sea pen.
Sea pen versus $5 million robot. Round 3. Score is score is currently sea pen 2, ROV 0.
I know, it's like, wow, it's like ROV 0.
I definitely agree with one of those.
Could have just said Chris 0.
Oh!
We don't know.
That was a little hard.
Yeah.
Yeah.
I think, Russ, you should let Ben try it.
He wants to.
I know, I mean, you need to get it.
He wants to.
I don't know who was flying in 2017 or who was.
There's a good chance it was me as well.
Uh-oh, our core is. Good thing we have this push core tube that we were joking about not needing.
Especially as 2 lines.
Do you want me to zoom out? No? Okay.
Just draw right on the green.
For about 6 inches.
Hey, Chris, you watching the core?
I'm watching the core. I'm watching everything, Ben.
The elbow?
You're fine. Yeah, you just need to come back.
Right?
Just spin, just rotate your shoulder in. You're good right there.
And that more.
Come down. You're good. You're on top of it.
Okay, stop that.
Okay.
Yep. Mike, there you go.
Okay. Sorry.
Looks like our little rings.
Oh, don't worry. We'll pick that up.
That's made from corn plastic.
Oh, it's made from corn? Okay.
That's irrelevant. We'll just go in here.
Just get this.
So while they're working on a question about what powers the ROV is attached to the surface, and so the cable, the tether, provides the electricity from the ship. So it's ultimately all diesel power, but the vehicle is electric driven with the long, and brings its long power cable along with it.
Yeah,
Okay, can we get set up?
essentially diesel powered because the ship is diesel powered, and all the power comes from the ship.
Why is it me on that? So, should bring the tree in. Camera out.
Do we have a quiver that's already open, or is it gonna go back in that?
That's the idea. It's got a button in the bottom.
So you go back in that tube.
Or we can open a cover and we can put it in there.
Can we open a quiver?
Yeah.
Because then we can close the quiver, whereas if we
No,
don't.
you can't close the
No,
quiver.
you
till
can't,
put
it's too
that
tall.
one in. It's too tall.
No, but you could dump it into
Yeah,
the
yeah,
quiver,
yeah.
right?
If he goes down deep enough, it's gonna cause,
works
it's
the
gonna
same.
create a
I
vacuum.
don't think we're gonna
No, I
get
mean
that
I get the way a push core works,
Yeah.
but I don't think the sediment's gonna be deep enough for you to
No, 'cause
actually
I'm gonna have
push
to go
core.
down and then just turn it upside down.
Yes.
And then I'll roll
Yes.
it into cover. I know what you want.
I know. Yeah.
Alright, what I'm gonna do is sit out
The
here.
issue with putting it in the push core quiver is that it's not. The coral's gonna be dead by the time it gets to the
Yeah,
surface.
yeah. Right, let's open it up.
Comment on that.
12. We'll get that shot. Okay, should we do that? Just punch out there, I can see.
Watching out.
Just double check that deep enough, I think.
Yeah, it's open.
Figure out.
These are attracting.
Yeah, I would just,
4 chunks?
Yeah, we'll take the sediment sample.
yeah, just,
4 chunks.
Yeah, please. Yeah, go back in and see. You don't attach that rock or anything, do you?
no. But that didn't really retract.
No,
right?
it didn't.
So
I didn't
I'm
see it
wondering if
really.
it's a different kind. There we go.
Well, it looks like it's going to be one of those cliffhangers on whether or not we got it or not.
I thought I saw something in there.
Yeah, I thought so too.
Yeah,
Something, but I don't
definitely saw
know.
something else. Something orangish red in the middle.
Oh, okay, that sounds promising.
Yeah.
I thought I saw it retract, I saw the hole.
Basically above the rock. Yes.
Yeah,
Like
right,
the scoops
yeah.
on- Unless you run around in sand.
No, I think we will
Hopefully.
look through the sediment when we get it to the surface and hopefully, and if it's not there, that's okay too.
We'll just update the scoreboard.
We'll
CPAN
update the
3,
scoreboard.
RV 0.
It's like the thing
No,
about I love these things.
That was quiver 13, correct?
Yep, cover 13, sample 345.
Yeah.
312, yeah.
Right, but it's labeled 13.
Yeah.
12:32 UTC, so
Okay.
5 hours ago. 5, 4.
That's FK 4345.
Yep.
Your guess is as good as mine, Adam.
We got Chris in the driver's seat.
We'll still get you.
Huh?
We'll still get you.
Oh, we don't have
Still gonna, I'm gonna grab the regular.
Oh, before you do. Oh yeah. Before you put that on there.
Yeah.
That's like
You just come back, or do you know the results? I don't know if we look back in the sediment.
I can't see that far. Portions of that.
Yeah.
I don't think it got it. We need the peninsula spatula.
How does a spatula work for that? Is that a good way to get them?
We used it on one cruise. There was we couldn't get them out, so we just dug them out with a spatula. Stainless steel spatula.
But did they stay on the spatula?
No, just to dislodge them and you slip them
Oh, gotcha.
because they're breathing fruit in there pretty well.
That was crazy though. That happened obviously to the one. Heavy.
Oh yeah.
You don't pick up that,
Okay, let's get the
fortunate.
There we go.
Ooh, can we reach that?
Not really. I'll just I'll throw the ring in the box and then
Alright, yeah, I'll, come down so you can.
just a wee bit.
You can jump in because I think super exciting.
See if these take 20 hours to check.
Plus, we don't really even see that, that would be unenvironmentally friendly. There you go, can you reach that? Hold on. Popped off. If these things were vertical instead of horizontal, it's the problem. I'll touch it.
Better late than never.
Yeah.
Okay, C-pen, hopefully, question mark. Okay, we got the
Anything to look at right here before we go?
No, I think we're good to keep moving on up.
these are good positions. We change, we start changing direction.
I feel like the setup is nice with that deep water.
I mean, the push core got down pretty far.
Yeah. Yeah, when he was putting that in.
I don't know a better way to get that.
We're in the deep water.
Take that,
The only other possible way is to get under it
And it's
first.
not like
Yeah.
cliffs, it's going up and up. So that's
Yeah, when he was, dumping that in.
Yeah.
I'm pretty confident I saw something
Good.
that looked reddish orange. And I would, I strive not to compliment him, so.
Yeah. Yeah. Yeah, so just so we've done this before, but like maybe on
So that green piece of plastic we picked up was part of the storage container for the push core that got knocked loose when we pulled the push core out. So we were just picking it back up.
I'll put some dark glasses on, put my hat down low.
Wait, do you want me to go get you a wig from the
Yeah.
answer box?
Go grab me the my COVID mask.
Just want the whole like horse's unicorn head.
Yeah, that'll work.
It's supposed to be a horse flying the RV.
Had that before. I'm a little nervous actually. Oh. The bullet, the full straight bullet. And I don't know how high this goes up.
Bridge, ROV. Just gonna get you to slide aft a little bit there. Thanks, Tony. Yeah, you're looking.
Yeah, we're gonna, this is gonna go.
Okay.
Yeah.
Yeah, a little bit. I was pulling a little bit, but it's straight up. Let's just go this way.
Reposition the ship a little bit. I'm trying to.
Okay. Looks like a hemicorallium. Corallidae. Big iridid gorge, yeah.
Brittle stars look kind of placed over here.
They do. I think that's what you sampled yesterday.
Those are brittle stars.
Yeah.
These are smaller spines, similar.
Totally thought those were percentages when I first saw them.
Can I zoom on one of those ovoids on the rock, or are we just?
No, we're I was just getting away from the face because our missiles is going
Okay,
a little
gotcha.
red right now. But yeah, you can do whatever you want.
Okay, thanks.
What do you want to look at?
One of these stars over here.
Okay.
I'll just come in a little bit and then I'll let you.
You want a close look at
No, I'll just do a snap zoom, if you will. I think they're similar, Steve. They just look, yeah. But from overall,
happy?
yeah, happy. Got a couple of Ortigorgia. like a black coral, small little stickopathy.
That makes sense. Over the right side. You're kind of, oh,
A lot of those are more brittle stars on rocks than we're used to. Did you answer that Facebook question?
It appears as though it's not
The Greenpeace one?
moving off.
The what?
actually.
The one about green, the green?
No, the Facebook one about
It might be going
pressure.
No.
Okay. There's a question on Facebook a little while ago about if the samples are ruined when they come to the surface because of the pressure, and not. No, they're not. Especially the invertebrates, which is mostly what we're collecting, seem to be okay with the pressure change. They don't have any internal gas chambers, whereas when you bring fish to the surface, they have swim bladders, so they do have there's a more need for actually having a decompression chamber to bring those specimens to the surface. But as far as the things that we're collecting here, they all seem to come up in decent condition, and we're not keeping any of the corals or sponges alive for any long amount of time. So we can generally preserve them or use them in a quick experiment before they start to degrade.
To give you a sense of the pressure, here we're at 2,010 meters subsurface, and the vehicle pressure gauge is reading 2,975 psi.
Okay, no worries. Looks like probably a little bamboo coral. Let me just come in a little bit. Let's see. Do you see any nodes?
Hints of one, not sure yet.
Yeah, the polyps look like bamboo polyps though. They come out a little bit.
Bathagorgia.
Athagorgia. Never even. That's great. Thanks, Chris. So, chrysogorgia? Sorry, we can come out. Thank you. And yeah, whatever you need to do with the bill, obviously. Okay. We're right around waypoint 1 now.
We made it to waypoint 1?
We made it to waypoint 1.
Is that
Look
it?
at us.
Waypoint 1.
Oh, hey, Deb. Hey.
If you switch to outreach channel and push talk on that. But this is gonna be next week.
Hi everybody, I'm just going to jump in and say hi real quick. My name is Deborah Smith. I'm a marine technician here on the research vessel Falkor. And we just want to shout out and say hi to Adam, who is one of our ROV pilots. His niece Emmy is watching with her class at the Mountain View Elementary School in Hinton, Alberta, Canada. So we want to welcome those classes that are watching and say hi and say please send in your questions on YouTube and Facebook. But we're just going to show you real quick where we are in the world so they have an idea of where Uncle Adam is currently working. But first I'm going to show you the control room so Adam can wave hi if he looks at the camera. So, good morning, Hinton, Alberta, Canada. Thank you for joining us. And I'm going to bring up Google Earth for us first. And so you can see we are very far from any land. Way out here in the middle of the Pacific Ocean. If I scroll the Earth to the east, we have the mainland United States of America and Canada there on North American continent. And then we have Hawaii out here in the Pacific. And if you keep going west, there's this little stretch of land called Baker and Howland Island here, these two little islands. And we're off to the west this morning on this newly mapped seamounts chain. There's two little seamounts here together. They're not quite so little. And if we zoom way in, we can see the Falkor's position, and the little dots here are where we're planning to dive with the ROV. So that just gives you a little bit of a sense of what we're trying to work on. And if we switch to our 3D view, you can see ROV Sebastian making the way up the side of the slope. And so that's what we're doing now. In the picture in the background is that there's a really steep slope, and Chris is flying the vehicle, and he is trying to keep us off the bottom and climb at the same time, while Adam is trying to position the ship to make sure that they move in tandem together. So we're going to be exploring this ridge for the next few hours, and we have Brian and Alexis with us in the science chairs running the control room, looking at all of the different organisms that they're trying to investigate, collect, and sample. So please feel free to reach out, send us your questions, and we'll try to answer them. Does Adam want to say hi real quick?
Yeah, good morning to my niece Emmy and her class in Hinton, Alberta. Just to further what Deb was saying here, we're presently at a depth of 2,000 meters subsea, and we're climbing up to a target depth of 1,200 meters this morning. So hopefully you guys stay tuned in while we climb this wall and look for samples.
There's another
The other thing is like it's
Ampelopathes, black coral, old bamboo stalk, Daguerreotarorgia with crinoids on it. I do think so. Brian and I were saying we've been a little sluggish this morning. We probably need to cover a little more ground. So
just
whenever you guys are, yeah, I don't want to.
Yeah, when you're sorted.
Yes.
But once you're back in a good position.
sluggish is like a meticulous, I think.
That isn't much fun. We've been meticulously scanning the seafloor this morning. We know people are watching from all different parts of the world. That's one of the great things about being able to stream these dives in real time. We're currently operating on the ship at Samoan time, American Samoan time. So it is 6:30 in the morning, roughly, on board. We went into the water for the around 1 a.m. So took a couple hours to get to the seafloor because we were going so deep, down to 2,300 meters, and then we'll work our way up this ridge on this seamount up until about 1,200 meters, like Deb said. Looks like Anthomastus with all its polyps pulled in. Mushroom coral.
see the question about bamboo corals getting their name, and the reason they got that name is they've got nodes up that look like the joints in bamboo.
Yeah, so the white part of a bamboo skeleton is made out of calcium carbonate, and it's hollow, and then the nodes, which are the brown part that you'll see when we're looking at a bamboo, are made out of more of a protein matrix, and it gives the coral skeleton more flexibility, which hopefully means that under high currents or under a high weight of the coral, they won't break or fall down as quickly as something that's more rigid.
Yeah, even to the northwest. Yeah. Hey Tony, Adam here. I'm gonna slowly start bumping you to the southwest there.
Yeah, sounds good.
We're sorry for
We're
you
not
to do west.
seeing quite as many.
We're just sorry for the reposition there, Tony.
We
just trying
saw
to get you off the wall a
yesterday
little
we
bit,
did a
just so
dive at
the
a
group quite
nearby
substantial.
Seamount around the same depth range, and we are seeing
Thanks, man.
somewhat different communities. Seeing more corals at deeper depths than we saw yesterday, but we also saw pretty consistent coral populations for the second half of yesterday's dive, or I guess middle of yesterday's dive. Most of our dives have been ranging between 8 and 12 hours, depending on depth, when it takes a little bit longer to get to the seafloor or back up to the surface. There's a Walteria sponge. I'll do a quick zoom on it, Chris. See if there's any, and a cup coral right next to it. Alright, that's good.
Okay. Go for it.
So on this expedition, we're exploring new areas, but we're also specifically looking at the coral and sponge communities, trying to get a better sense of their biogeography, which organisms live where, and then studying coral predation, corallivory, and spongivory. What are the predators of our filter feeders in the deep sea? And then we've got a microbiologist from Boston Children's, Harvard, on board, who is looking at innate immunity and the functioning of immune system and the interplay of coral immune systems with deep sea bacteria as well.
Looks like a. What's going on? I'm sorry. Should have run the camera. A bamboo coral. We've seen probably the most abundant coral we've seen so far. These really thinly branched bamboo corals, and then above here is a very different morphology, but still a type of bamboo coral. And a small, looks like paragorgid on the left too. Not many associates today. We've
Yeah,
seen a couple of brittle
that's-
stars, but only that one crab that was with the.
There is a crinoid there, but
Actually, there's something, Chris. I'm gonna look at this this lower left branch.
Okay. Oh, kind of like the one that's in the
Yeah, there's like, looks like there's a little green something on it.
little. Do we gonna set up for you so you can look at it?
Yeah, why don't we?
Do you think we're gonna sample?
I don't think we're gonna sample, no.
That'd be amazing for East to watch it.
Oh,
You know, I'm just wondering because I'm gonna pause the ship on this.
no, we're not gonna sample. Chris is trying to guilt trip me.
There's a little sea star eating that.
Is there?
Right in the lower right hand corner. There's a
Hold on.
Well, I'm letting. Yeah, I'm not.
see it there. There's stripped coral and then there's a
Oh, I don't
That's a
think.
brittle star,
I think it's a
though.
brittle. Yeah, I don't think it's actually eating it.
well, it just happens to be there.
You are correct. It does look like corallivory.
so yes, most fish are in shallower waters. There certainly are fish down here.
Okay.
We've seen one or two, but the shallower we get, the higher the density of fish gets. It's an old egg case.
Yeah, it looks like an old egg case. There's documentation of certain types of octopus laying their eggs on corals, so that possibly could be an old octopus egg case. There's also sharks that will do that, but their egg cases look quite different.
Yeah, you can't control it. Yeah, well you can, but it'll keep the group. Well, I've been trying to make sure I'm free of the cement before I rip off your.
It's a little pink ball.
Yeah.
I know, I don't know what that is. Little.
One way to find.
There's more of that weird branching morphology over here.
I'm just gonna keep it super close. Yes. Yeah, because I think it's out of this face of the depths.
What?
How high up is this? It looks.
It doesn't, like, it's weird. I don't know why, I don't even really see this face. This whole thing looks kind of like the same.
Well, the next thing is as soon as we get up to that ridge, but that ridge is still.
There's a nice gorget with an
This was
overlay.
600 meters away, and that slope is.
So bubblegum coral.
23 degrees. This is steeper.
Oh, there's the lasers. You can see the two green lasers right in the middle of the screen. Those are 10 centimeters apart. So they help to give us an idea of how big some of the colonies and other objects that we're seeing are. So this is quite a large coral colony. Yeah, somebody just asked that. Yeah, the lasers are 10 centimeters apart. All right, that's good. We can carry on.
That should be some. Sleep up. And then back. The odds of you having those both secure when you go back on deck increase. Me out here. See, that's good. Look how you look. Yeah. Yeah. Which means towards the ship. You're just on the edge, yeah. So that's what you're doing.
There's a fallen over colony, and looks like some sponges.
And then where the gorgia mixed in there.
Oh yeah,
Go to
that's weird sponginess, huh?
I'm convinced that's sponginess.
Oh really? Oh, you're right. That's a chrysogorgia.
With sponginess behind it.
A sponginess behind it.
You got some music parallel somewhere. Not the entire time. I put it in parallel while I was going. Is there one you want to look closer at? Or you just want to look at all of them?
I'm just gonna look at this one on the left
Okay.
first.
Oh, jump.
That Walteria looks like the internal
Yeah, it's dead.
structures are dead,
Agreed.
but the spiny parts look alive.
How's that?
That's good. Come in a little bit. This looks like that one that we saw. Remember where it was like we. What's he say?
You were flying away.
Oh.
So Steve says it's the biplanar Chrysogorgia, but not sure about the ID. He needs like a piece.
Is this something that we could sample?
Yeah, like, what,
Is that? Yeah.
yeah, what do you want to sample on?
We want to sample
The whole
that.
thing?
Not the whole thing. Just a snip. Just a branch of this.
Top to the bottom. The bottom.
Whichever works
I
for
don't
you.
think we care, yeah.
Yeah.
Alright, yeah, no problem. So I'm gonna set up on just the port side so that you can come up from underneath it.
Sounds good.
Where do you want to snip there?
Whichever is easiest for you. We just want a branch that's probably 20 centimeters.
Sure.
How's that for you? I'll do 8 so you don't have to all the way up. Hold on. Nope. There we go. What's that? That's something. I think we're a good spot there, and then we can just go straight down on that. Yeah, I'll go for that. You can go as far as you want. Yeah, I got plenty of space if you want.
What did he say it was? Chrysogorgia scintillans.
For one, for that
So just any little snip there?
Yeah. Are you gonna come up from the top? You think, or
Yeah, probably I need to come down on it like this.
okay? Then yeah, I would say just like maybe a little deeper in than that.
Sure.
Great.
That was wonderful, Adam.
That was
You did a
wonderful.
great job,
Adam, fantastic
Adam.
job
That was
with the
amazing.
arm.
If I was your nephew, I'd be so proud.
Niece.
Best
I'm not
best
gonna be his
collection
niece,
ever.
though.
Oh,
I'm his nephew.
you
I
are
was
right.
going for me.
Here comes the overseer.
So are we closer to where we were 4 years ago?
Yes, we're gonna be even closer on the next after the next transit.
Because this sort of is more where everyone
Yeah, yeah.
Did we cross the equator last night?
did we?
No.
No.
Never crossed it.
No, we shouldn't have.
We're gonna end up, well, we will cross, but we're not gonna do anything until we go home. We're doing it out the northern run.
Yeah, we need a couple of days to clean this up. Have like 1 day of suffering.
Alright, that was 346 in
346.
Quiver 11.
We weren't allowed to use utensils or sit in chairs.
When did you cross in person?
You guys.
Oh, really?
Yeah.
What do you mean? I used utensils and sat in a chair.
Not we had to like sit on their knees and then.
Are you telling us you didn't participate
I,
fully? 'Cause we
I
could-
did stop. Don't you dare.
Oh.
Don't you dare.
It's called cheating. Well, I'm glad we know this now.
I did all the things I was supposed to
Luckily
do.
for you,
Luckily for you,
we can fix any of
we're
the-
gonna recreate the experience.
No.
Oh, by the way, alright, so as we were talking about bamboo coral earlier.
Yeah, there's a good
Joints.
example. So you can see the white part is the skeleton, and then those brown sections are the nodes. So that's the protein part that we were talking about. And sometimes they can be much thicker. Just gives the skeleton some flexibility versus just being completely rigid. That's a good place to point that out, Chris. And that one looks like it's toppled over. It's starting to lose tissue, but there's still some live polyps on there.
Anything else while we're here?
No. You got good imagery on that one, right?
Of the Chrysler, yeah.
I was wondering if any of these Wolteria had spongivores on them.
I haven't seen.
Yeah, I mean, from that, since that first or second
Yeah.
dive, when we found that white star.
Sharks. No, I haven't. I sent Carly a picture of, I sent her the video of the HD of the sick on it. But I didn't have the, I didn't have that with some of the scenes.
I sent it a picture of those.
Alright, should we try and make some distance?
Yeah. Yeah, we're gonna just keep going. If we see anything different, we'll let you know. But if it's stuff we've seen, we're gonna try not to on it.
I'm still gonna like try to keep stuff with you though.
Oh, yes,
please.
yes, please.
Not.
Sorry, I didn't mean that.
Well, no, I'm not saying like keep your mind at zoo, but I still might go zigzagging just to.
Yeah, no,
see
that's
if
great.
it's,
6 and an 8. Okay.
Alright, but
Oh,
that's
there's
new.
that's new.
What's that?
This yellow one.
Okay.
So we'll take a look at that.
Yeah, like,
And there's a little.
collections of look or just.
I think just fly by look.
Of course there is, because there's video, but yeah, it's seven. I don't know.
It's a very large overlay on there.
I don't know what J-Rod or, Eric have put together so far from my lanes, but like, trying to tie
Yeah.
that on the shoulder.
Bring down just a little bit. No, that's the brakes.
Alright, drum roll. So what is it? Acanthorplex.
Just need more.
I would say
Collections?
no, not a collection. Two ophiroids, like one's got its arm wrapped around the other one.
More
Or around
interesting
the-
than the one Acanthidae.
No, they're both astroschema. They're just
Yeah?
different. Yeah.
Oh yeah, sorry, I was looking at the wrong
That's
side.
okay. Steve. Plexarid? Plexarid. I win.
There's a lot of good pictures of JIs.
Okay, we can keep going. It's our first Plexarid at 1,961 meters.
Oh, it's about where they started here. I think the deepest one we saw was 2100.
Okay.
Right, that's about right. The deepest we saw in 2017 was like 1700, so we've actually been making significant dips.
It was 2,700?
17.
Oh, okay, gotcha.
1700.
Oh, here's a different type of primnoid octocoral that has fallen over.
We don't have any primnoids for Randy yet.
Do you want to sample this?
Probably wouldn't be a bad idea.
It's already knocked over anyway, so it's not gonna. Yeah, let's set up to. Oh, a calyptophora? Interesting.
Something like that. That's how the posh people say it.
Posh people. There's a question about how do we tell if a polyp is live or not. It's more about if it seems like the coral is live or if it doesn't. So if there's still tissue and polyps on the coral skeleton, then we consider it to be alive, or at least is still partially alive. But if you see bare skeleton where there's no tissue or there's no polyps present, then that is considered a dead coral colony. So like this coral has fallen over, and at the base you can see there's no live tissue, but there's still a lot of live coral along the branches. Over time, this would probably be smothered by the sediment. So eventually, those polyps will no longer be able to feed or continue to grow. So it would die over time.
As well as its feeding ability is greatly reduced
Yeah.
on the seafloor like this as well.
Oh, there's some shrimp living in it. All good?
Can you come down on the brightness a touch?
Yeah. See this? L.
There's those shrimpies. Doesn't look like the big claw shrimp.
No.
I don't see eggs on either of them, so they don't look gravid, just hanging out on their coral. Okay, where do we want to snip? How much do you think?
I mean, I would say as much as fits in a quiver.
What about, is this possible? This branch?
Yeah, what we'll do is we'll grab it and then we'll stick it on the porch and grab it from the top and shut it down.
Okay. I mean, or wherever, if there's another spot that's easier for you, we just need a quiver amount.
It's a little bit of a reach. Is that something we can do on the other side, maybe?
Yeah,
Yeah.
for sure.
Yeah, no, totally your guys' choice. We just
It's just
a
we
quiver
need
full
a
of.
quiver amount of
A
this
quiver
coral.
amount? Okay.
Yeah.
So maybe even like,
Here.
yeah, like that.
Yes.
Right there.
Great.
Absolutely.
Okay. All right. Do you want it where that branches off?
No,
in
it's
the middle?
okay. Here.
No.
Oh.
Look at you see that? Yeah, like
Go.
yeah, like the branches off right there. So you bring down a little bit, or right above that branch.
It honestly, either way is
We're gonna
fine.
we're gonna dice this one up into lots of little pieces. So
It's that round ballish type thing at the bottom of it, so it's the end of the main branch.
Brown
You
ballish.
want that right there?
Yeah, like why, yeah, see how the main branch ends and it kind of just looks roundish?
Like a cat and knife.
Oh, this?
Yeah.
I think it's just starting to be overgrown by something else. Like that looks. Does look like a large. Yeah, it's just like a gall. I don't know if the skeleton grew over something else or if. Yeah, I don't know. Great.
I'm getting that into a quiver. I think it looked kind of pretty. Might just have to, does it have to be intact?
No, it could be
Figure
pieces.
it out. That's my power. The power of suggestion by just looking at my eyebrows go up, which comes up with. Oh yes, as far as I can. Yep. Well, I mean, if you want to stay where we are, I might be able to push it a little more, but that's the only thing that we're touching, so I can't really guarantee it's gonna happen. You rack a little? Yeah. I can porch out, I just don't know what's gonna happen. I think you're gonna have to. Alright. Sometimes it's just nice and slow, it should just, cause you got forward thrust on there too. Yeah, I'll just we'll figure it out and see what happens. I mean, it'll be the first time. Hold on. Okay.
No yawning. No yawning in the control room.
sorry. Adam. And then that's just the best collection out of
Just
Adam.
gotta
Great
use-
collection.
Use what you got.
Use what you got, which is more coral.
Alright, I'll drive it.
All right, so that's
That's your
sample
quiver,
347 in
Correct.
quiver 10.
If that fails, you okay with that? Ed?
Yes, absolutely. Hi, Mr. Tim.
Hello.
Did you get some sleep?
Got some?
Okay.
Everything's done.
Good. We have this many samples. We have not covered as much ground as we probably should have.
What was it? 10 on bottom.
Not till like 1 a.m.
Yeah.
Yeah.
No, I think even after that.
1:30.
2:30.
Sorry.
Gosh, Brian.
Brian's fault.
Whatever, 12:30, you can see whatever that is.
Yeah. Okay.
6 hours ago.
Yeah, oh, it's coral. Just coral popcorn.
No, that's
Yeah.
good. It's fine.
It's got
Those ones are.
that tight.
Seen some different corals,
Huh?
Seen some different corals from yesterday.
Yeah.
Yeah.
Really different.
And then the
Different-
density started deeper.
Yeah. Or at least like we started seeing,
The big,
Some.
healthy, fancy ones.
yeah, big fancy ones.
Fancy corals.
Are you happy?
What are you going for now?
Curious.
We're done. We just
Yeah.
snagged a big chunk of hemnoid for the respirometer.
Have you seen metallic gorgia?
None. 0.
Victor gorgia.
Nope.
Nope.
We did sample in a, what did he call it? Anthothella, which is similar to Victor gorgia. Now we can keep going.
2 to 7.
What's our
Slowly, very slowly.
what's our top depth?
I think it was
Our
like
top
1200-ish
yeah.
is the goal.
1200?
Yeah, 1250 or so.
Normal.
But the dive plan says it
We
should
gotta
be
go
12
faster.
hours of bottom time.
We got about
I mean,
100
it says
meters.
12 hours of bottom time.
No, we've done almost a little over 200
All right.
of vertical in
Yeah.
4 hours.
Well, what did we start at? Oh yeah, like 20. We started at 22.
Yeah.
Right, so almost 300.
Saw the biplanar chryso. That was cool.
Yeah.
You do you.
Oh no, it's a shadow. Never mind. I was like,
I know.
please stop.
Yeah. I think there was one to the left, Chris. A different, not the C pen, but there's something even further left.
Next one.
Right there. That's what we'll take a look at.
We'll be back shortly.
Wait, you want to look at this, Tim?
What is it?
It's this.
It's chryso.
It's not metallo, is it? It's on a.
Bring down the elevator.
There is a squat. That's like our first Europtychus of the day.
down on it, it's hard to tell.
I don't think it's Metallogorgia. I think it's a bushier Chrysogorgia.
It's nice. Oh, that's red face.
Right, that's red face.
That's red face.
Okay.
Yeah.
Okay, we can keep going. Sorry, you don't have to stay.
No,
I just wanted
that's
to-
good.
Rock or sand,
Rock.
it's striking the big thing.
Where?
Has Randy been around?
Nope, I haven't seen or heard of her at all.
Yeah, it's weird in the thick camera. Plus, it looks like the face is like kind of falling off. You guys want a rock?
Do you want another rock?
Since we've got such a long depth range, let's keep going. We got a good rock, and it's only been 300 meters.
Yeah, that's a good point. We did collect a rock earlier in the dive.
I'm good to late. Yeah, I'm good to late. Go eat.
How much stuff did you not eat since you woke up?
I had 90.
How do you do that?
Once you don't do it for a
I
while,
guess,
don't
yeah,
really
once you
think about it.
condition yourself.
Yeah.
Not me going downstairs and eating bread the second I wake up. It's a little chrysogorgid.
It's more rock on the side of the stuff, I think.
Big stocky thing. That's what we're always looking for.
Huge something.
Let's look at this little. There's like a very
Okay.
delicate stocky thing. There's huge ones and there's little ones. Yeah, that's a good observation. In the chat, there are a lot less crinoids than yesterday. We had a lot of crinoid diversity. Found you a Metallogorgia, sir. Tim. This is a Metallogorgia.
Where?
The one spiraling behind it.
I doubt Steve would like it, but no one is.
Well, is this something of interest for you?
I think we need to confirm. It's pretty small too. Yeah.
Want it?
Yeah, I do. I know.
Okay.
Thank you.
All of it?
Just the top part. We don't need. Yeah, but from here up with the associate. Yeah,
Later.
Box?
we can put it in the bottom box.
I think, I mean, I can try to put it in the river.
No, go ahead and put it in the bottom box.
Well, you're doing that, and you're double triple thing. I'm gonna cool call thing that I was actually supposed to.
Also, want to look at that little there. It's almost breakfast time. I know. I'm so hungry.
Yep. Oh, sorry.
Some RV?
grab a little board arm and use something to pull against.
There you go.
Just want to take that off. It's a full rotation. It's a good one.
Fortunately. Slow, pushing up the torch. Again, the same as before, drum those up as to have to do this. Nothing's in either, right?
No
Oh, bio
boxes.
boxes. I'm
No.
sorry. Yes, they're all open.
The G goes to the far one degree difficult.
No, it's the smallest. It's the smaller.
Nice. Well done.
Just leave that for
Yeah. In case, fathers listen to where we've
Yeah.
Alright, I'm gonna spin around.
Okay.
Come around from the far side.
Sounds good.
It's a little lumpy up there. Yeah.
You're real tall. Over to Gorgia.
No, I don't think I'd be very good at that. I barely fly right, but so what's your sister teach? Well, she ever watched us work before, so you know what she does. It's turned out fair play.
It's true. But we can nail that one.
For your living sea star. We never see those really thin ones on corals, or like doesn't seem like we see them
No.
predating.
Watch it just crawl down.
Yeah. Okay.
Check it out. I'm just gonna stare at this for a little
Yeah,
while.
yeah.
Make sure you're good.
Think I'm down on the camera.
Sorry? Yeah, there we go. That's much better.
You guys should be able to control from everything there as well, right?
Oh, really?
Yeah. Let's just see. It's in manuals, so.
This looks like Swiftia.
Let's have a look see there. I
Yeah.
think it's this one here. Yeah.
Oh.
Right?
Gosh, it only took 10 dives for us to.
It took 4 years.
Only
I
took
didn't know it.
Oh, yeah, there we go. Now I'm really in control.
It's okay. There's an auto heading. I never use the auto heading button. It's a soft button.
Yeah. Swiftia, so Buxarid.
EEZ?
We collected that yesterday. I don't think we need to collect it here. So we have this over to Gorgia. We'll just do a scan. Look for any jellies, because that's.
See, Ben trying to break up the surface up.
Crinoid. Okay.
It's a good picture.
Yeah?
It's like stars.
It does look like a star. It looks like outer space, doesn't it?
Yeah. The moon's getting
Not very much current, because all of that's just like sitting there.
the light one. Here. Oh yeah.
Okay, we can keep going. I'm gonna try really hard not to stop.
About 7 meters. I mean, when are we ever gonna be here again?
True. That's very true. Look at Adam putting us in perspective.
You will probably never see this coral.
Oh, you're
Ever
100% right. I'll never see that
So
exact
just
coral.
take it in because it's about to go away right.
No!
Hold on.
Now I'm attached to it. I
Let's
can't.
go back and land. Have another look. We'll,
It's a big bamboo whip.
like you're fading out. Yeah, into the oblivion.
Goodbye. Goodbye.
Okay. Try your iris now.
Oh, wrong way.
Yeah, I knew I should give you control.
That was a setup.
That's a cool.
Just play with it. Let when you.
Okay.
I mean, when we land out and stuff, and if it's not good, I can.
No, I think it is good. I just don't want to.
I can fine tune it over here too.
Okay. I don't want to like do anything when it comes to piloting. You know, I don't want to accidentally darken it up too much.
Have you seen us? We do that all the time.
Well.
We'll hit the auto buttons all of a sudden, just goes pitch black. We're like, whoops.
I think yours is more fine control. I have like step control, so I can.
Yeah, yours
do it
seems
quick.
like yeah. It's a big coral. Looks like a bamboo.
What I gotta do is I gotta get labels on the buttons. Because they don't even say they just say.
But that also, I think we used the iris control in 2017. Like
On there.
yeah.
Yeah.
That's what I mean. Well,
think
so
they used to be labeled. I don't think they're labeled right now.
it makes sense that we have it because I don't remember asking you guys every time to do it.
You got focus, yeah, you got DNR. So.
I wonder what B does.
I think that we need to be judicious with how much we let them know they can control. Not you specifically, but I'm not like science.
Yeah. There's an up-down control for the ROV.
On the way?
Yeah, you gotta be real careful. A couple of those buttons will actually take control of you start flying.
That is not true.
That's not true.
Okay.
I started buttons.
Brian was about to get real excited.
Yeah, I was about
He's
to
like,
start
I'm
flying.
gonna start controlling the computer.
I flew Sebastian in 2017. I'm a real ROV pilot.
There is actually
I feel like we should get certificates for that.
there's actually you
Oh,
can
yeah,
actually
it is.
take external control and nav. See this little button here? External control and nav. We've actually had it where we've had
was for like an autonomous thing?
Yeah, we were doing like transects.
Oh.
Speech program and like these grids and the ROV just go start flying.
We got Victor Gorgia and
Metallogorgia.
now more Metallogorgias. It's like we're definitely picking up in the. Can we? We don't need to set down. We can do it just right above here. Do it Papaniscan.
Sure.
I mean, yeah. There's a lot going on right here.
Yeah, but I think I had one of the
We popped number 1 already, but all the others are open.
Okay.
We'll use a big one.
yeah, 5 and 6 of the station themselves.
Yeah, it's because it's on the station. What it is the altimeter, and because you're on the edge, it's getting different readings, so it's floating around.
Alright.
We try to hold depth instead.
Hopefully no waste. Hold on. Yeah, but it's pretty good even. Alright, hold on. Let's keep an eye on it. Yeah, I don't like that. Okay, go for it. Great, that's fine.
Yeah, 2. Can you just write some of this
You must-
in about what was there? Because then we have it in the spreadsheet
It's
notes.
like 4 is kind of crap. See that? On the left?
That's the top.
Oh, that's the top of 5. Okay, yeah.
So that was 2.
Cool, thank you.
Let's make it to 1800 meters before we pass off.
Oh my gosh. yeah,
Not
that's so-
1800, not all the way to 1800. Sorry.
Oh, into the
1890,
18. Okay.
Into the 1800s.
Wait, there's a big picnic going
Oh
in.
yeah, wait, but I say that, but let's look at the picnic.
That's the best way to find things, is you say we have to cover more
I gotta go
ground.
somewhere.
I'm actually looking at this sea spider back here.
Okay, flip around. And the pink poofy thing?
Not the pink foofy thing.
The scary nightmare thing.
The skin.
Whatever we were looking at yesterday was 3 feet.
Was it a pic? Was it eating an
Yeah.
anemone?
Red.
Oh yeah, the sea
Thing.
spider with no eyes that look like eyes.
That's this.
That's this.
Oh,
It's
it's the
just
same
not.
thing?
Yeah, it's just not eating. It's just not feeding.
Yeah, that may be a problem.
Excellent question about coral reaction to predation. We are very much trying to figure that out on this cruise, and we're not sure, frankly, how the corals react to the damage or not. And that's one of the things. That's why we're kind of excited about this pycnogonid here, is they are a very, one of the more voracious predators of corals. So we're trying to get a better idea of their density, how they interact with the corals, what kind of damage they do they kill the entire colony, all those kinds of questions, and then part of that, understanding the immune response of the corals when they're attacked, how they respond.
I'm gonna sit down and games around with this.
Okay.
Switch this delta to 60 per
This one is not feeding. It's definitely just
Yeah, 6 to 7.
hanging out. Yesterday, I think they saw a pretty big pycnogonid
It was
feeding
dark,
on
It was dark red and just freaking.
feeding on an anemone, which is pretty common to see. We've seen one feeding on a sea cucumber during this cruise. We've collected one of them. Since we're over here, I might as well just look at some stuff.
Ben will be happy to hear that.
There's another Swiftia. Lots of hydroids growing on those. Really nice metallic gorgias. We haven't seen these so far, or we just collected one, but we hadn't really seen them so far. I don't know if we saw any of them yesterday, or maybe they were much deeper on yesterday's dive. Not much, but a couple hundred meters. Tim Sheng gets very interested in the ophoroid associate with metallic gorgias because it's pretty cosmopolitan. Anywhere you find a metallic gorgia, it often has one ophoroid associate and of the same kind of ophoroid every time. It's a nice little coral garden. No coral predation yet today.
Pycnogonid was the first predator I think we've seen.
I don't know if I've ever actually seen a pycnogonid eating a coral.
I have.
Oh, have you?
Yeah.
What kind of coral?
I don't recall. But I'm
That's the most important part, right? Looks like a plexaurid to me. Okay, onward. It's kind of funny, we are aboard the ship, so we have a pretty good understanding of what's seen in every dive, just because we're talking to each other, but we often, as the scientists on board and the ROV pilots on board, we don't actually see the full length of every dive. Wow, this looks huge. We don't see the full length of every dive, so sometimes the viewers actually who tune into a full dive see a more complete picture of it than we actually do, because we do have to get some sleep out here. These are pretty big bamboo colonies.
There have definitely been cruises that I've been so busy with the planning and logistics of the rest of the cruise that I don't watch pretty much any of the dive and end up having to watch the highlight reels to know what happened.
We can just fly by these, Chris, but I'm going to come in a little bit to look at some of this. If we have any associates, looks like crinoids, barnacles on there, but I don't see any predators actually. There's dead tissue, but
There's oafs.
gonna look at the damage margins. Yeah, sure. Thanks. That's down.
what you got, I guess. People keep telling me stuff, but I don't think it's true. Or I'm in denial.
I saw a used Bronco on Craigslist and I figured, why don't you just buy that? Yeah, it's true.
By the time I get my new Bronco, it'll be a classic Bronco. It's so long.
It's very strange nodes. Like they're not. They're very.
That.
Banana, no nut. Coffee. I did buy you a coffee. This one's got Solution milk and some
The little corkscrew
Soda.
cat.
chocolate.
Huh?
The little corkscrew.
Yeah, squid?
I don't know. So I'm trying to see. I just moved the wrong thing. It's the wrong knob.
Well, not home brew, but like,
Is it a little squid?
no.
So it looks like to me.
Yeah, I just think it was spoonful of chocolate. And you make
Our first squid of the expedition.
He's sleeping?
coffee, espresso.
Oh no, it's a little. It's something crawling. It has a foot. Do you see?
It doesn't, they don't fix the machine though. The black doesn't develop a freaking chocolate.
I think it's one of
I have
those
to put an espresso in that.
similar to a purple orb.
Black
Do you see this? This is like on the sediment.
Yeah, I see it looks like a foot. I'm with you there, but we don't know.
That's why we're down here in Texas.
Sometimes they do know. They know a lot.
Every once in a while.
Sometimes they don't.
Can you see this other one? No. Okay.
You want another one?
Nope. Let's keep going. Whoops.
Sure.
I'm positive. I think we're getting into a much more dense space.
Right?
Yeah.
You guys want to go to breakfast?
I do.
Go for it.
Okay,
But
I'll
do
be
you
back.
Don't you have a 7:30 meeting?
Oh, do you?
I'll blow it off.
Okay.
What's the spiral one?
I know what to do.
Course you're full.
I haven't seen any corallivories yet.
Not on a coral. We just saw a pictogonin a few seconds
yeah.
ago, but it was just on the seafloor, not eating.
It's in the famous rice board today.
Okay, I know.
But it's called an iridic gorge. I say that's still right.
All right.
What dog is that, mammal?
Ryan, do you want to go to breakfast? I don't mind sitting there.
I'm okay for a little bit longer.
Canine.
I'll take you up on it if my relief doesn't get here
She'll be here.
in a few minutes.
She just started breakfast.
That's
so
fine.
Okay. Quite a few samples already today.
I can push buttons on the computer.
Guys are making tracks. So 1927.
Good year for the Yankees.
Yeah, probably wouldn't be good. Yeah, last time Tim and I.
Yeah,
sat
so we
down
just
on
got up on this
microphone, we had
yeah.
on this little. It flattened out. We came up a vertical wall, and then we picked up a lot more life once we've been up on the top of the wall. But the current still seems to be pretty light.
Yeah.
Cornucopia.
Cool.
We've got one quiver full of primnoids for the respirometer,
Right.
and then we've gotten several new bamboos, one cup
Yeah.
coral, one
Yeah.
possible sea pen, and one niskin in a high density community.
Okay.
Actually, no, we should have two niskins.
You got Anthomastus, looks like.
We did not
Yeah.
collect it.
What is that?
That must have been
Anthothela?
That's Anthothela, yes, we
Yeah.
did it. But this, hold on, we got another niskin that didn't get looks like it recorded here.
Okay.
Because we announced our arrival, but it definitely.
talk with a lot of glass and barnacles to get into a groove. Let's see here. All right.
You can get a sense of the a little bit more rocky outcome.
So can you come out on the map on the scout map? Oh wow. So
Can you see the pin, Tim?
oh yeah, sorry. Should I go get it? One?
Oh, I didn't see that one.
That's
But it didn't want to go.
yeah.
Yeah, we're 1.2.
Goodness.
Hey, is that the seabird you guys got yesterday?
What's that?
Where?
Center left now.
You hold on a second, Chris. Center left.
Oh, there?
Sorry. Is that what you got?
Sit down and look.
I didn't see
I
the
don't-
seafloor photo.
I don't think so.
Bottom left corner.
Image that.
Sorry about that. A little bit cloudy.
That's right. It was this one, Steve, right?
Yeah.
Yeah.
Down.
There we go.
No, we got a big fleshy one and one with smaller polyps.
So this is different.
I think that's different than the two we got yesterday.
Yeah.
Is that the tracking one?
I don't know. I didn't see that yesterday.
We picked one up with the
Pretty amazing.
this morning. That was good.
Yeah, we think we got the sneaky one.
In the quiver? So you grabbed it?
In
Okay. You ready?
No.
yeah.
We push cord it and then dump the push core into the quiver.
Thank you. Yeah, okay. I thought you suction sample.
Oh no. Are we going friendly fire?
No.
We also learned today that that is iris control.
Excuse me.
I figured you guys could handle it now. There's also the one below it should be ISO.
They've been holding out on us.
Oh my God, that's me.
I mean,
Wow.
you see why.
Yeah, I know. Yeah. You don't want kids playing with things that can hurt them. All right, Steve, what do you think?
It's naturally, I don't know.
Yeah.
Yes, we agree with you.
Yeah.
No, like
You can
it's
sort of see the,
family.
yeah. You have an interest?
Is this the same we saw earlier or different?
Yeah, I do kind of.
Yeah. Well then, we're here. We're set up. How it?
I don't know if
Basically,
this.
if you see a sea pin, now you gotta collect it.
I don't know if this is a runner. So
What I'll do is
Yeah.
I'll get the soft jaws just around the base.
yeah,
So hopefully we can catch it before he.
yeah,
takes off.
really
Yeah.
quickly.
Yeah. And I think we've got 9 open so. Yeah, you can probably go grab it now before I. You ready? When you're set. We'll reach it. Yeah.
Don't know if I've ever seen that one.
Yeah, I have
don't know.
from 2017, but
Oh yeah.
uh,
Well then I must have seen it, but it's not
was,
registering. Yeah.
it was not well imaged, and I don't think we ever got an ID on it. Got it. You got it.
Nice.
I think it's really.
Yes, please.
Would it retract now if that you had it in your hand, if it was retracting?
I don't know. These pinaculas, they don't run away as much as the other one.
Are you driving, Chris?
They're a different family.
The camera?
entirely. There you go. That's really weird that it's alternating.
That's I've never seen that.
Somebody else?
Usually paired.
Is it focusing?
Branch.
No.
How about
Wait for it.
there we go.
Yeah. Okay, sample shot. Got it. Got it. Good to go.
Alright.
Thank you.
You reach that? Unfortunately. Yeah.
All right, this is 350, right?
350.
In quiver 9.
Aye, quiver 9.
Alright.
We also noticed that somehow these numbers got reversed. But,
So you could have been corrected here.
yeah.
Oh, I see. So quiver 9. Yeah, so what we've been in 5 hours on the bottom or so, and we've gone 300 meters.
Yeah.
Sorry.
Meticulous is the word.
Yes, meticulous, careful, methodical.
Morning, everyone.
Hey, good morning.
Have you guys all had a chance to eat breakfast?
I have not.
To go. Can you leave this here?
Yeah.
Yeah, thanks.
You guys are wearing matching shirts.
I'm gonna take over.
Hello.
Hello. How are you?
Okay, how are you?
Well,
Excellent.
how are you?
I'm okay.
Okay.
There's the, I'll pan down a little bit.
Single stalk, looks like a bamboo with a.
Is it almost center screen now?
Yeah.
Is it that little twig here? There? See that one?
Where is it?
Let's see this one.
No, it was lower.
Slowly start coming back up.
So it should be being NTP stuff, so something.
What did you see?
There's something here.
Yeah, it might have been. No, right there, above it.
Here.
Yeah, right there. See that crab?
That thing right here?
2/3 of the way up. Yep, on that.
Yeah.
That might be the Bethagorgia.
Good eye,
okay, good.
Steve.
See that.
Holy moly.
That's what I do.
I know, but that was like that was invisible. Except to everyone except for you.
All that's good now.
So what we see, so
Yep, that's it.
7 narwhals.
Just when Tim comes on.
But the other one had no associates.
No, I have no associates. I haven't seen a Bethagorgia with
And you
the,
collected it, right?
we didn't collect any.
Well,
The mermaid. The
Wanna collect this
mermaid.
I think that would be a good
I
idea.
did too.
As long as we check in, do some zooms. But I haven't seen any crabs. I've seen very few crabs in this dive so far. We saw one at the Thela, but it escaped. It was a Munidopsis, I think. Or no,
I'm sorry.
not Munidopsis. What are they called?
Eumunida. On what?
On Anthothela.
Oh, Anthothela. Yeah. That was 2017 too. We saw that.
That was really hard to spot. I'm really impressed. So is that a flexor? What have you been seeing?
This one?
Yeah. To the right. To the right,
yeah,
the pink one.
there's both Flexor or Gorgia and
You got two of those little babies.
Did you just bump into a wall?
There's 2 crabs on there.
Let me just frame this up.
Ben bumped into a wall.
Oh baby.
You want it?
is that Heteroptychus?
Not yet, but yeah.
Oh.
No. At least that was not. Just get a whole shot in focus.
Yeah.
At the base there.
Come again? Capital back, yeah.
We're gonna go to the bottom.
Oh, sorry.
It is.
Alright, I'm gonna go grab breakfast too. I'll be back. Oh, I wanted to make sure everyone.
We good. I was right. What is it? I thought I was wrong. What did you say? I said, alright.
isn't that where Carl's from?
Nah, he's from Stone.
No, I think he's from Mount
Steve, when I get sick, because I need this computer to see well my computer stuff, if you don't mind.
Yeah, I'll stay for a few minutes.
Wow, I don't know what that is.
The crab?
Yeah.
Yeah.
I don't know. Nice polyps though.
Mm-hmm.
Jimbo, did you make it to the,
Only meeting
on
yesterday.
time, like,
I did
for the
actually.
beginning?
I actually made it about 5 minutes before the beginning for Blue Miracle. At the yesterday.
Certainly a Carostara. Certainly a Europtychus. I don't know.
How far is it?
Beyond that.
Did you lose
Let's
where
see
you
a shot of this. Okay for the polyps?
Yeah. Awesome.
Okay.
So, do you mind doing low?
Yeah, that's whatever you need.
Yeah, right. Okay.
Let me
No,
Another.
this is likely a biobox sample. We like to black it.
I just took the first.
No, I appreciate
got the
that.
camera.
Thank you very much.
So,
Got open.
Thinking maybe a biobox, right?
No, box is fine. Yeah. I've been saving them.
I don't know. It's can't tell how long this is really.
What's the answer
I
number?
don't get that.
This will be number 351.
Thank you.
Yeah, right.
Oh, it's a lot closer.
Oh, there's a little.
Well, it's time.
Here we go.
That's
Oh.
Gaza.
It's a whirlybird.
Gaza. Gaza. I think is the name of that genus.
Yep.
Boom. Okay.
Well, that's one way to get around
Yeah.
your snail.
There's 2 crabs on there. Is that fine? For
Yeah.
both? Yeah.
Yeah. No, yeah, I mean, it's,
Yeah, I'm fine with whatever you need.
I love it.
It looks
Well, I
like
just-
bluish, right? The tissue.
It
looks
does look
almost
bluish.
yeah,
Has a
pearlescent
hue.
blue.
Right.
So are these crazy stuff?
Very, very,
Or what do
very
you want me to call them?
Style with it.
Well, just in case I'm messing around with this one. That's it. I'm gonna give you a city one.
Morning, Captain.
Hey, Captain. So this is the iris.
Yeah. I'll
I knew
play
that,
around
but
with it.
but it doesn't. Yeah, there's not a lot of dynamic range. Yeah, can we see if the crabs stay on here? Nice and easy, and they'll stay on.
Yeah, I'll just hold here to you. Portion of it.
It's falling over.
Hang on.
So, oh.
Let's talk to the
Are you
crab.
talking to me?
Let's talk to the crab. Sorry.
Oh,
Trying to trying to mind meld.
Hey,
Stay there.
Tim, we've got in bar box
Yeah.
A.
Yeah.
I think it's A on yours as well. Far left.
Metallic orgia. You can mix those if you want.
It's too big.
For what?
Spin it.
It's bendy. Just to save real estate, because you got still got quite a transect to go.
All right. That's fine with me. Wherever you think you can get it in, that's fine. Yeah, it's okay to mix those two.
If I don't,
I
Chris
think.
will have me.
I mean, I want you to get your collection. I want it to be good, but I still want to
Easy.
Is that bulbous butt crab?
No, I don't think so. I don't know. I don't recognize them. Yeah, that coral was sampled in 2017. I don't remember any crabs on it.
No.
I don't have any.
What is he paying? It's $100, right, Chris? It was 100 Canadian, so I think you got change for 5 US.
Doesn't even have to function.
Excellent.
What biobox is that?
A, yeah. It's with metallic gorget. 3, 5, 1.
Come on, hit. Here you go.
Good. That's good. Good precision sampling today. Like that. Good targets.
There's something in 10.
There's Tim.
Okay. There was a little bit of RNA later leaked out of 24, but we sealed it up.
I saw that. It's alright. I can't believe that much came out, right?
Yeah.
Russ had an issue earlier with another one like that.
Like, it looks like it should just go straight down.
Here you go.
It's weird. Well, it's something on the seal, right? What's in 10?
That's for respiration.
Yeah.
It's a big branch.
Yeah.
It was wasn't too big.
Good. Wow. You can see something there on the side, right? Oh, that's,
Yeah,
That
no.
shouldn't be preventing that from closing, though.
No, it shouldn't. But that's that's pretty the thickest part of the
Yeah.
of the segment.
Kind of coming up a little bit. See that? Yeah, the rubber.
Oh yeah, right.
Yeah, it's come off that plate. Oh, it's not like we're not gonna lose the sample, it's just not super.
There you go. Nice. All right. Ready to roll? All right. Good.
Seems like whenever I'm in the room, sampling testing occurs.
Huh.
In the effort of time,
That's like.
I'll take one for this.
Yeah, either the vehicle or just what he's gonna look at. Otherwise it's rock solid. Yeah. And what would we do? You know, just as a. Any cups to go down?
No, we're good.
Cup empty?
I'm one, but how are you doing, Van Hurst?
That's in Retigorgia. And bamboo coral below it. Typically we've seen Europtychus crabs in these long-armed crabs that sit around the second whorl of that coral. Saw a lot of those yesterday, but not seeing it today. And we're not that far from where we dove yesterday. We're just to the east. Hey, can we take a look at this one, Chris? Sorry.
Which-
This, the, yeah.
Wait, close look or just?
Yeah, I'm gonna sit.
Alright.
You see, I'll slack Steve. We were talking earlier about this coral, and I think he might want
I want to show Brian that sponge.
to. Yeah.
Yeah, let me just slide out real quick and then.
Yeah. I said that would be great. What was I doing?
Sure, I'm safely away from that one.
You're only the fifth person to do that.
Yeah, something's up with H3 too. It's flickering off like crazy. Good, I mean,
Can we look at the base of that sponge?
It's like Neptune's trident.
Just look, it's a half. Looks like it's half dead on the bottom. Do you mind if I grab this?
No, go. You're going for sponge?
Yeah, I want to look at the sponge
We've
first.
seen that sponge dead on the seafloor.
I know, but here it is,
Right.
half dead, half alive. Right? That's kind of interesting. Maybe,
So
maybe
you want-
it's.
He's almost.
Oh,
he's
sorry.
almost sitting down. So it was. Yeah, I was chatting with Steve. I think we want to image that coral on the left, and then.
no.
Oh yeah, we'll definitely
I'm
image them
looking
both.
at
Yeah.
H3 and
Oh, we got a little interaction, a little urchin interaction
I've
down
seen
here.
a lot of that this trip.
Those urchins in particular.
I don't think I remember seeing it before, but maybe,
No.
maybe I'm just
Must be this
misremembering.
time of year. That's all. They get together.
So you think there's a seasonality?
Nah, I don't know. There's quite a bit of seasonality in the deep sea.
I know. But I wouldn't. I'm surprised that these urchins feel it. There you go. You never know.
Tides are. You can feel tides way deeper than this. I mean, all
What?
the way. You can feel the tides, the
Mhm.
pressure change in tides. Much deeper than this.
I don't know. Is that a sponge at the bottom, or is that like a benthic ctenophore? something else.
I don't know, Holothurian maybe? I can't
can't
see
even
the
tell.
inside.
Yeah.
All right, I think we're good to go. If you want
Could
to
be a Holothurian.
image or anything.
All right, hang on. Let's just quickly answer this question.
Yeah, I'm
Holothurian, I think.
Okay.
What do you think, Tim? Yes. No.
Yes.
Yes?
Yep.
Okay.
Yep. Might even take off and start swimming.
Maybe.
Is that a Bathypathes or a sponge that's like
That's-
blending into the rock?
Oh my God, Anna, you are. That is a.
I don't actually know.
Black coral, no? What is that?
It's got an associate on it. Two of them.
It does. Looking for polyps.
Well, it's isn't that?
And
That's a
there-
crinoid.
There's a crinoid on the stalk. I just don't know.
But
if
see,
it's
there's
dead.
these are two dead stalks, and there's an oaf on, wait, an oaf there and an oaf there. No.
Yeah, absolutely. I don't know. Yeah, I just don't see any polyps. But to
Me
me,
either.
it's more like a hydroid.
You know, there's your question about structure versus. I don't know if it's the same oafs you'd find in a coral, but it's not a 3D printed
Yeah.
coral, but it's a
Right.
dead structure.
Yeah, it's
All
true.
right. Let's check out this sponge. And see if we can figure out why it's half dead. And then we'll talk to Steve about this coral. Oops. Almost, maybe.
We got the rock in the front. There we go.
There we go.
Well,
All right.
It's good.
This is wild.
Is that a different kind of sponge growing over the top and over?
I don't know. We've seen these dead morphology of dead sponges of this morphology at the bottom here, lying on the seafloor. I haven't seen this yet. I don't know if did you see this sponge before?
Not
Didn't
today,
know what?
or not near as. If we did, it was much smaller.
Yeah.
I'm just trying to get focus, but let's head out here.
There you go.
All right, there's a little crinoid on it. What else do we have on it?
Interesting.
It's beautiful. It's a hectactinella, some kind of glass sponge. Could you help me focus, Russ? Ben? I'm having focus problems.
There you go. You got to have the rest.
You got it. You got.
Oh, there it is. Thank you. I thought it was.
Oh, that's
Wow.
awesome.
Look at it. Look at the growth form on the top. You know, the small
Right, where the
budding
buds are coming
or
out.
whatever they
Yeah.
are. What do we call those? Is there a word for that? There must be a word for that. A sponge biologist would know the word for that.
Yeah.
I'm gonna call them buds.
That's it. That sounds good.
Wow. All right. Well, I don't know why it's half dead on the bottom, but I know that it's stunning on top. Let's just take another look at that.
Don't see any spongivores. I mean,
And
that's
there's the
nothing
lasers.
down there.
Perfect. No, I didn't see anything down there.
Right.
I mean, we could look at the other side when we're done over here, but let's look at this coral first.
Mm-hmm. Good.
All right. This is a bamboo coral. Have we heard from Steve?
No,
might
I
be
mean, I think this
asleep.
is a no.
I think his phone's right here, so that's a problem.
No, I think this is a morph that may be a, I don't know if it's a juvenile form or not, but it's.
All right, hang on. This might be a barnacle in the base. I just want to note it if it is. Come on.
You got it. Here it goes.
Or maybe not. An amphibod? You see, there's something on the base.
Wow.
What is that, Tim? You know, your
It's
three,
smooth like an apicophrin.
the three words
but-
that you like so much from me. What is that, Tim?
Yeah, right. You give me all the easy ones all the time. I don't know. It looks like it's a barnacle. We're looking look at the back side of a stalk, little stalk
Okay.
barnacle.
I will stop.
Could be a glyptolasma barnacle, something like that.
Okay. All right, let's go. Let's
Yeah.
traverse up this stalk. Let's take bamboo coral tour.
Looks nice and healthy. It's a pretty long central axis here.
Yeah. But I'm interested to get to that branch. Oh, there's some kind of little shrimp. Cute. And then here we go.
It's a
Yes,
nice
center
image of
this.
this. That's great. So this could be the isodella of some sort. When you get into
I'm trying to
isodellas, I
get
don't
it
know what,
framed perfectly.
there's Les Watling and Scott France been working on these, and they, have their clades. And, I don't know the latest from them. I think all the polyps are basically coming off of one side or off the sides, except that one.
No, there's two of
Or
them,
is it on
right?
the
It's
side
like
there?
every
Yeah,
third
you're right.
polyp it
Yep.
comes up into the page.
You're right.
On that, just on that one branch. On that branch, it's one into the page, and that one there's none. Right? It's very irregular.
Yep. It's really interesting up top. That's awesome. Great.
That's great.
We don't need to collect it. We just need to
But
image
I
it.
like looking at the apical tip, like the growing tip on this, because you can see it's interesting that it's not a polyp on the top. They're really growing axis 1st. And
Yeah.
okay.
That's great.
You know what else is cool? Sorry, just while we're here, look at the base of where it's branching. As the branches grow, you can see the new polyps coming out. You can really see the size differential.
Look at that mysid shrimp down there.
I know. He's like, I've got a whole coral to myself.
So I'm waiting for the tentacles to grab him. Oh. It's great. You can see right down into that
Les
polyp
is here.
too. Oh, there.
Hi, Les.
Plate
It's good to
14.
see you.
There we go.
We will, or
Great.
I4.
You want lasers on that? I think lasers are on.
Let's get some lasers on base for Les. So we've got Les Watling in the chat, which is a real treat. Thank you very much for joining us, Les. He is an expert coral taxonomist, specializing in many things, but
Bamboos.
including the Isididae.
Yeah.
And here we go.
Let's get lasers on the top too. I think he.
Oh, you want lasers on the top?
I think he would.
How do we get lasers on the top? It's just gonna bounce right.
Well, I don't know if we can get it or
You
not.
want to try it? All right.
We'll just go up and see. We'll just go up and see. I see. Here we go, Les.
All right. I trust your very big, your framing skills here. I don't think I can get the
We'll see. It's
lasers
gonna
on
be a
this.
little bit- All right, there's one.
Oh, you're
There's
so
two.
good, Tim.
There's two.
Look at that.
Bonsai. All right. Okay.
Yeah.
And I'm gonna zoom in as much as I can. Less.
Les, do you want a sample of this?
That would make like a great logo.
It looks like a
So
five-fingered
get
hand.
it's like a hand.
Exactly. Russ,
It looks
like
a
like,
mitt.
yeah, it's like a
It's
hand.
a
yeah.
mitt.
It's a cool. It's like a symbol though. It's got.
Monolith. It's like Jewish.
Yeah.
Nice to meet you. Okay. What do you want, Eric?
Just get center it. You had a really nice shot earlier, but you weren't centered.
After that, here we go.
Okay.
You're gonna give me a heart attack.
All right.
You're
We're
gonna
gonna.
give me a heart attack.
Okay, yeah, right there. A little back off a teeny
Yeah,
bit,
yeah,
just
yeah,
a teeny
yeah.
bit, and now slightly to the right.
Just so you can tell me.
Wow, slightly is hard.
That's all it is.
Slightly is hard.
Teeny
So
bit.
the lasers are
Yeah,
10
hang on.
centimeters apart. I'm
I
looking
mean, you
at the
could.
chat.
That's good right there. Just leave that for 10 seconds. Nature just creates its own
Amazing.
fascinating forms, right?
Amazing, amazing.
Amazing.
You got good focus, you think? Yeah.
It's great.
All right. Awesome.
Awesome.
All right.
Alrighty.
We're good.
That sponge is
Ready
really
to- ready to roll on?
amazing. Hang on.
Think about a sample here, Russ.
Yeah.
So we're thinking about like that. You think?
Yeah.
No,
Just to get it right?
I would think that even just the above
Just
the
the
branch.
one here?
Yeah, exactly.
Yeah. Okay. Well, I guess I don't know whether normally we try to include a branch node here.
Oh.
So for these things, so normally I don't know if
Okay.
unless you want to cut below one of the
All right,
first
no, that's good.
branches.
Okay. Good. Oh,
Yeah.
you're
we
Tim's
try
right.
to we try to take as little as possible, but still
These are
get.
just below the nose.
Yeah.
I think so. I think so. Right here.
Okay. See that?
It's gonna be pretty small, right? It's gonna be.
I know. All right.
So.
It's easier for getting access. I need to go to the camera for a minute. Must be.
Is this a definite pre-sample photo right now?
Yep. So Tim.
Yeah, it is amazing how much is grew before it branched. You got the camera. Sorry,
Exactly.
you got the camera. Sorry.
No, go ahead. Anna, did you have context?
Yeah.
I'm gonna
Or
Tim
I
and I
can get
are
another
gonna
RV.
have to organize a little bit as we swap to get our proposals in. So like maybe you and Tim and then me and you, and then so we can both get our stuff
Yeah,
in.
my computer wasn't plugged in, so it's charging right now, so I'll go grab it, it's been charging.
No, no rush. I'm just letting you know.
Just tell me when you want it.
Yeah, I'll talk to Tim about it in a second. So the conversation we've had about sampling, we always try to take as little as possible. We do have Les Watling in the chat, who's an expert taxonomist, who's really trying to resolve the taxonomic details of this clade. And this is a really good opportunity to honestly give this coral the name it deserves and give it and give it its place in biological knowledge. This will, go for taxonomic research, and parts of it will also end up in museum specimens and DNA archives that everybody can access. And it's really important to be able to resolve this, because if this is one species of bamboo, then and it's everywhere, then we know something about its distribution. It tells us something about the benthic ecology of the deep sea. If it's endemic to this region, that tells us something about population demographics and endemism in the deep sea. If it's specific to this seamount, if it, you know what I mean? So resolving who everybody is tells us something about, then allows us to talk about where they are, which enables us to talk about how they are where they are and why they are where they are, which is really how we get to the ecology of the deep sea.
Yeah, I think what's really interesting too is that when we see these isodelic corals, we often see the hand part of this thing start much lower on the central axis, and have much longer branches that extend out. This is sort of a different growth form. I don't know if that means I don't know if it's juvenile, if it's something else that in the life history or the life cycle of or they're totally different species.
So if it's a totally new species, then the morphology might just tell us something about its taxonomy. But if it's not a totally new species and it's got this really high branching, it probably tells us something about its physiology or its ecology. It had to grow this tall in order to be successful.
Or the environment it grew up in.
Yeah, exactly.
In respect to its physiology. Yeah.
So, there you go.
Don't normally see associates on these guys.
Oh, you have that little shrimp.
Little mysid, yeah. Yes. Yeah.
Just want to clarify, just below the nose.
Yeah.
Just below the fork.
Yeah,
Yep.
just under the fork, so you get a block piece, that's a joint.
Just that one branch point.
We get these subcounties a little bit.
Right.
Go get it. Yep. Go get it. Yep.
You gotta get them right away. You're looking at 6 seconds, tons of. Zoom in on this set as well.
But over here, Tim, are you happy with that?
Yeah, just a little lower maybe. Make sure we get that node, that branch point. Oh, I see it there. Yeah. If you're below it, you're good.
Blades right in the actual blades. It's like the centimeter. There you go. Set. Set. Perfect shot.
Nice. This should be 352, right?
Roger, yeah.
Mm-hmm.
Isidella? Is that what
Yeah,
it's called?
yeah, it's a bamboo. Should have bamboo there somewhere. Or Isididae. There you go. Nice, Ben. Nice job. All right.
Okay, camera's yours.
All right. All right. Let's keep on going down the line. We're headed at waypoint 2. We've been in the water for about 5 hours.
When you just said that, I was like,
Waypoint 2.
uh-huh.
We have a long way to go. See if I can iron something a little bit.
That's not the easiest job.
I don't know if it's better for you guys to control the iris or not than over here. I think
Yeah, I usually just use the
I'll just leave this alone, I think. All right.
along the sides, just the one that.
Okay.
Is that an Anthomastus?
I don't know where you're looking. Oh yeah, sorry. Yep. It's the other side. Okay. Okay.
Yeah, so, I don't know. When's the last time we white balanced?
Just thinking about that.
You do it right now, you want?
We had that sponge in our face. We could have done that. Let's go on the sediment. The white balance. Let's go right here.
It doesn't look too bad now.
Yeah. Go for it. Wait.
It's not really too bad. That's too low. Just give us a sec, man. It's called the station. Okay, try that.
Much better. Thank you.
Okay.
Lovely.
All right. We're at 1887 meters, heading up to I think 1250 is our goal. 1267. And we've got 20 more meters elevation to our next waypoint. We're just following up a slope before we come up on top of a ridge, and then we'll take a left turn and go down the ridge. Seen a couple of stalks. A lot of these stalks with crinoids on them.
It's like a sea pen? No.
No.
That was just the angle. It's definitely like a paracord or a hemicron.
Yeah. Just trying to see if it's a corallium or not. No. We're okay. We're good. Keep going. Good.
Oh, cute.
Another Anthomastus and another bamboo. skeleton, really large holdfast. Must have been a really large colony before it snapped off, and you have an ophiacanthid ophiuroid living on that bare skeleton. Yeah, I think we're nearing a really steep part of the track. That's right. Do you have an underlay on it? Right.
No, I do, but it's just,
It doesn't do anything?
we're not at this.
Oh, I see. Yeah, sorry.
Over here.
Yeah. We're just, yeah.
It's very bland.
Yes, it is. It's always a bit slow going for us at the beginning. I shouldn't say slow, but it's just that we see so many things we want to confirm our identifications on. We can keep going there. We've seen that, I think. So we like to do a lot of collecting for representative taxa, and get those in good collections, and then we tend to move more toward the middle of the dive so that we can make tracks and see the distribution of the animals that we're observing. Lots of sediment, but still lots of rocky outcrops. It's also hump day on the ship. Did you know that? Today's hump day for the cruise. Yeah.
Never heard of something this.
No, it's the middle of the. It's the day that marks the middle of the 34-day cruise we have here.
I thought it was on holiday.
Another hold fast. Bamboo. We have some really large colonies on this dive that we did not see yesterday on a neighboring feature that was at about the same depth, almost exactly the same depth that we're surveying today.
That's Dr. Crino again.
Yeah, haven't seen as many of those today as we saw yesterday. Pops right around. There's another bamboo whip on the right. In addition to the coral composition and the animals that live on them, the ophiuroids and the crabs, and we're also looking for predatory events. When we see something that's predating on a coral, we will stop.
Look at that whorl skeleton.
Yeah, that's a Rudogorgia
It's huge, though.
skeleton. Sort of fallen over onto the rock. So we will stop and look at any type of corallivory feeding on the corals. So identifying who's feeding and how they're feeding and what the wounds are like. And then we look at the microbiology of the wounds, and the areas. We look at the microbiology of everything, frankly. Everything that comes up, we look for the microbiology. There's another.
Microbiology, the seawater,
Yeah.
the microbiomes, what's culture. Wow. Oh, it's.
Yeah,
it's
we're okay.
And,
Look at those ophs on the right there, all in a line. Well, we just passed a Metallogorgia coral, which has an ophroid living in the central area of the branching, and they seem to be much more flattened or more planar today than we've seen previously on the cruise. Like not as many branches, so it's really interesting to see that. We did get a collection of that to look at that and confirm that with its ophiuroid. Can we take a look at this coral over here? Please. Yeah. Just want to double check. It looks really bushy. I don't know if it's a.
oh, there's that sea
Might be.
star. Kind of
Where?
that.
Where you see? Where you see?
Right in the middle.
Oh, the purple star there. I don't know. We'll get a snap zoom on that too.
Yeah. It's quite fluffy.
So we'll see if that's a planar chrysa. Looks pretty thick.
see it?
It does.
Yeah. Inside.
Okay, it's all yours.
Thank you.
Justice there.
What did it say? Right there in the center? Is there
Sharp.
something? Yeah. You can see its eyes.
Kinda.
There's two screens different. You see it.
Yeah, it's the
Very distinct.
call it the red face. Let's go in here.
I think it's the seconds.
Oh, and there's its legs moving.
Yeah, there's a cow-style crab in there. On this must be the planar chrysogorgia, but it really has thick.
What's on the right
I don't
there?
know.
The other red eyes? You see that?
That's the way.
Yeah.
Is that another one?
You mean?
Same thing, or?
You mean over here? Oh, down here. Yeah, there's two in there.
Yeah.
Yeah, that's right. All right. Great. Just want to get a good image of this, and we can go.
Take a beauty shot really quick. Hold it.
No.
Well, not hold it. Right, got it.
All right. Okay, we're great. I'm going. There is a.
Sea star, yeah.
Yeah.
We're gonna go have a look.
Yeah. It can be a quick look or a flying look.
You can all over it.
Roger that.
Very pretty.
Stand the camera over there.
All the way up.
Okay.
Yeah. Just gonna
I'll
take a
fly
quick.
off yours now.
Roger. I'm gonna go part zoom and you can fly it in a little bit. Something like that.
Yeah.
Yeah,
That's
kinda.
rock.
Yeah. Come in and do a little bit of this. All right.
It's on the move.
He is on the move. I was just noticing that. Let's go. Let's get some focus here. There we go. It's really on the move.
Not as slow as you think. Yeah.
Kind of resembles a hymenaster, a slime star. It is.
Oh, wow. So cool.
Cranking in the deep sea. On the run. Right?
Well, it's fascinating, right?
While you're getting gas in your car, this thing's on the run on the rock in the deep sea, right? All right, that's great. I think we got decent zoom on that. Let's see. Can do that.
Oh, wow. Good job.
All right. So
It's on top of
we often,
it.
what's that? We often watch them respire through that. That'll open up
Oh,
that
okay.
central area there. And it'll respire. So. It's fun to watch it. This is
I
why we
didn't
want
even
to.
notice he was moving at first.
Yeah, I
and
know.
then it like really picked up.
All right, that's great. That's,
Just me. I got an email.
oh, hey.
They
Yeah.
put in that north up symbol in Freedom House.
Did they really?
Version 4 in the new one, yeah.
Got to download the next version then.
Download the latest version.
Oh man! All right, you know, just gonna straighten up a little bit. Roger.
Oh, that's his number.
Yeah.
Hi, Les.
Sorry, just reading the chat here to see what we got. little bit. Just gonna angle that. Can we turn the ISO on just some more lighting or something? Just a little more. Just a little, like a click, you know, just a little. Oh, there we go. There we go. That's great. Now
Flabella Chrysologia, usually more
What?
of a double fan.
over there.
Yeah, unless it's got a in the middle. Oh no, that's the sperm ones.
Yeah. Another metallic gorgia. They were prevalent at about 100 meters ago in depth. So about 1950 we saw a lot of this metallic gorgia. Now we're seeing them again.
Is it astroschema or the
Which?
Ophiocanthidae that are more common?
Oh, the astroschema
Okay.
type. Yeah. Any current today?
Not an awful lot.
No,
Not this depth.
not seeing much.
Coming down, there was a couple of bands.
Mm. She's up in the cabin, I think. So we're just running a transect upslope here, looking for corals and sponges, and looking at the animals that live on these corals and sponges, seeing their associations and how specific they are, and also looking for predatory events or predation events. This seamount that we just mapped, I keep saying overnight, but I don't know when night is now, so it was a few hours ago, I guess, 12 hours ago.
Yeah, I think we got in the water at around 12:31.
Yeah. And we put a dive plan together, put a dive track down to try to maximize the slope angles.
The input.
Yeah.
Well, you guys can click on
And
buttons. Watch change.
so we saw 45 degree slopes here that allowed us to run the transect we're doing right now, and then maybe another, I don't know, solid hour or two before we get up to the top of the ridge, and then we can run along the ridge.
Almost.
We know that these animals, sponges and corals, really like to feed using on particles that come through the currents that are brought here, and the currents hit the slope and they come up the slope and they whirl around. It's probably quite complex how these things work. Are there some cup corals down there? No. Sorry, I thought I saw 2 cups.
Count minutes. Every minute's a minute.
So yeah, so we know from experience that they like to have these areas with currents, and so we're often asking the pilots about what they're feeling for currents. We have a Doppler, acoustic Doppler profiler that can show us the currents beneath the ship, so we use that as well. But we also know that any current that we observe here can change. Certainly, there's a can be a strong tidal influence here at this depth of the deep sea. We're at 1,837 meters. And we also know that the distribution of corals and sponges can be patchy, like we're seeing now. We haven't really seen that much in the last couple of minutes. Earlier on the dive, it was hard to see a video frame that didn't have a coral in it, I feel like. And we're still on the lookout for rocks. I'm just noticing some of these rocks
Yeah.
may be loose. So Russ, if you ever see something like looks loose, you could go for
You know
it.
me, I'll fill that basket up.
Nah, I didn't know
There you
that.
go.
That's why I didn't mention it until now, until I really saw something. So,
Is that a Victor?
oh
It's
yeah,
purple.
get just a short,
Yeah.
quick zoom on that. You can look at rocks while we're.
That's a big one.
Yeah, so this is something that we did see before too, I think. You're flying it?
I'll never
I
get
don't
over
wanna
that
move it.
color
Try
though.
to fly it. What's that?
I said I
purple coral.
can't get over that color, like their pigmentation in the deep sea and why it's so interesting.
Yeah.
I feel like that's still the.
It's funny, most purple corals that have any kind of branches in it, they're calling it. Yeah. You want me to do it?
Yep.
Something like that.
This isn't the new one, is it? This is just regular.
I don't know what you mean. I mean, we have.
Steve said they had identified a new family, Victor Gorgia, that's like clear.
Oh yeah. Well,
Clearish.
yeah.
Let's see if he says anything.
I'm familiar
I'm sure
with.
it's normal. It looks like.
Yeah, I thought we had seen this one before, but another morph that grows really closer to the bottom and really sort of shorter branches and sits low. All right, that's great. I think thanks for doing that, and we'll still stay on watch. We have a collaboration with folks at USGS that want to determine the chemical composition of the rock. They want to look at the manganese crust. These seamounts are so old, between 90 and 120 million years old, that there's been a chemical alteration of the crust. The surface of the basalt has got a lot of manganese coating on it. It can be several centimeters thick. It's really slow to accumulate, but it does over that time. Been collecting rocks and looking at the width or depth of that crust. So don't know if that one.
I know. That's what I'm looking
Yeah, I know you're looking at that. We like the dark ones, you know, that have that crust on them. And it looks like it. You know, a lot of this is like pavement in place, just with a sediment drape over it. But the one that Russ is eyeing up right now might be loose.
In a minute, then. It's going to be attached to that crane. That's what's going to happen. Yeah. Oh, don't fail me now, camera.
These pilots are great. They love a challenge. You know.
Flying planes. That's what I want. It's cramping my hands. That's it.
Oh yeah, I gotta keep slack up so we can. All right.
Then, man. Make it so.
Make it so. So we have a pilot that flies us into these things, and we have another pilot that actually operates the manipulator, starboard manipulator. So they work in tandem to get us these great samples. So we've got Russ at flying pilot, and we've got Ben at sampling pilot. You hear them chatting. Ben has like 50 years experience in deep submergence assets, and
He's very patient.
he's very patient. He's extremely methodical and experienced on the stick. So.
Sorry, what's that camera?
Oh yes.
She's a mover. Oh, I gotta press something. Too much attention.
You got them dry sink lights. Let's see that. Oh, nice one, lights anyway.
It's a nice size rock there.
You're okay? Yes, clear.
That's not a small rock.
Thank you.
There you go.
353.
Oh, you can go all over right there, eh?
Yep. That's fantastic. Take a little zoom here.
Maybe two.
Get a sample shot of that. We image and we take frame grabs of all of our samples in situ so that we know what they look like when they come to the surface. That's great. It's a beauty. Nice job.
Straight.
It's yours. Yep.
That's excellent. Excellent.
So we like to get rocks that are in place that we know didn't fall down from hundreds of meters up
Those are the
slope.
bogs,
So we can, they can equate depth with the sample. So you see a lot of this rock in the background that's cemented in. There's no way we can sample that. So we have to look for loose rocks that we consider to be relatively native to this area locally. So
yeah, you can swing it a bit further. And should be times your wrist. Okay, bombs away. It's only. Yeah.
arms away. There we
Oh,
go.
there's
So
space we got. There's space there.
yeah. So that's 353.
Yes.
Okay, 21, 353. Nice job. All right. There's Anthomastus on the left.
I got it earlier.
Right. Mm-hmm. And we always should start thinking about making tracks too.
Oh, it's good we got a rock. That's a good rock.
No, that's a good rock. It's great.
Not yet. We're gonna make Randy's decision even harder if we don't make tracks.
Yeah. True that. Right, that is very true. Yeah, that's right. Okay. We have lots of patches of sediment, and our colleague Les Watling is chiming in again that most likely the sediment is derived from planktonic animals and plants. Absolutely. This could be pelagic sediment over hundreds of thousands of years. And depth is 1828. So there are some dips on our track and places that are more flattened rather than slopes. And when we come across those, we often see these ponds of sediment. Sometimes with the ripples you see there that are indicative of the energy that the motion of the water that created them. Don't see a lot of bioturbation. Don't see a lot of worms that reworked or other animals that reworked the sediment surface. Don't see any tracks or trails. Oh, here's an edge. Sometimes you pick up animals there on the edge. The flow comes across the sediment.
These whips are stick up handles.
Yeah, I think that one is there. And I don't know if they imaged these earlier in the dive or not. Or if they're.
We'll just keep going.
We're seeing quite a few here. A lot of small whips here.
Ooh, look at that metallic gorge.
Yeah, it is. There's that sponge you've seen quite a bit. We saw one live. We saw some of them have been lying on the seafloor. Lots of animals on there. You can see there's ophiuroids. Yep, that's fine. Can we look at this for a second? Just curious of what this is. I don't know if I've seen this. Sorry. We're good. Yeah, all right. Oh, sorry. Sorry. Yep, I'm all stopped. That's all right. We can keep going up if we. Yeah. That's fine.
Whoa,
That's good.
down the left there.
Yeah. It's gonna really. Yeah, we've seen a lot of large holdfasts today that I think are going to resemble the one we're coming up on here. It shows you how large these colonies can be. This is a massive bamboo. It's an icidid coral. See some focus from here.
Sit down for a second.
Yeah, that's fine. Sounds good.
Look at all the associates.
Yeah, I'm not going to touch them. Let him fly it. And yeah, there are ophiuroids all over this thing. There's barnacles here too. They get on the bare part of the skeleton and colonize.
Okay.
Yep. All right. I'm going to come out for a second. Make sure I'm all the way out. Get a sense of the scale of this colony.
It's just not clear. See it?
Pretty massive. I see the lasers there drifting. Yeah, these glyptolasmid barnacles love these large bamboos.
Trying to write it in. Oh, they're
Wow, look at that.
hydroids. I thought it was.
Well, there's commatulate crinoids here, stalkless crinoids on this. But I'm also noting some of the weirdness in the shape of the skeleton. The larger parts, some are inflated.
Yeah.
This is a seriously old colony that's seen a lot of stuff. These barnacles are really, I'll just say good at colonizing areas like this. They take high advantage of these places. They get off the seafloor. Not much of a stalk to them. It's just all about feeding. You see the ciria of the barnacles out feeding right now. You see the top one on the top right going in. It's taking in particles just like the coral is. Yeah. But I don't know.
Yeah.
Right.
Looks like an arm was moving. That's why I was.
Oh, is that right? Yeah. These bamboos are really fantastic. It's like a massive fishing net for particles. Look at that, right?
Oh my gosh, yeah.
There's crinoids everywhere. All right, I love looking at these things. Okay.
All right.
Nodal branchings are probably clade J3. Not sure which subclade, however. That's tough. Thank you, Les. Thanks, Russ. That was great. 1819. Back on our tracks. What's that?
So you just have less zoom in. Pretty much writing most of it.
Very hard to say how old, most likely more than 1,000 years. Thank you, Les. Yeah. It's just amazing how well they can do. And so you wonder how old those holdfasts are that have, where the coral's no longer there, could be really thousands of years.
We have no idea how quickly they degrade over time, right? Really?
Like the dissolution rate of a holdfast?
Yeah.
Clearly it would depend on water property, right? Temperature, acidity, that kind of thing. But yeah, I don't. Yeah, I see the sea fin. Is that one we've seen before? I'd have missed the first couple. Unless I see something, we'll keep going. another bamboo whip.
Yep.
We've seen quite a few of these.
It's cool to get see something that's living, that's over 1,000 years old and is that big. I
Yeah.
don't know. You're still, even though bamboos are relatively common in this area, it's always a treat. There's a mobile urchin.
This is just reminds me of the image we got from the multi-beam mapping that we did of this. It was sort of an undulating series of ridges or depressions with local highs. And so as we dip down and go low and come back up again, I feel like I know it's different scales, but it really seems to be the feel for this seamount that we're on.
Guessing this. Place of origin.
Yep. And you can tell that we're keen to get upslope, cover depth, and have an idea of the distribution of these corals, transition zones, and same for the associates, the animals that are living on the corals. It's one thing I really want to see if these animals are really specific to their hosts, and so they'll just change with the corals as they change their distribution, or will they follow their own depth patterns independent of the coral hosts? All right, Russ, cue the whale shark. Have to keep in mind too that our swath of vision here with our lights is several meters. You never know if that's indicative of the seamount or anywhere else. If we took a different pattern, different track up, we might see something differently. We likely would. Might be something big over there. I don't know what that was.
Sponge.
You calling it a sponge? Yeah.
See it again. It's a dead well.
Yep. Yep. It's true.
Seems like the dead ones have more associates than the.
Yeah, and that can be the sponges can be full of chemicals and whatnot.
What
so it
is
could be
hanging off that whip?
That's a crinoid over there.
Oh.
Yeah.
They're definitely both the same species.
I'm not going to say that. I don't know. It's like one.
I know. It does look like another one stuck on.
I agree.
Yeah, that's kind of what I was thinking.
You don't know how that morphology turns into that morphology,
exactly.
right? It's a gross. So I think you're right. We got
It
an
does
old
look
dead
like a
sponge here
bridge.
with a different sponge on it.
One just made the benefit of
Yeah.
it's already created a tower, so it just jump on top.
But we don't see that white sponge on anything else though,
No.
which is amazing, right? So it likes that old sponge.
It's weird.
It is. So next time we see one of those stichopathies like whips, we should stop and look at that. I want to stop and make sure. Yeah.
So you think some of them are black corals and some of them are bamboo?
No, well, for sure. I think yeah.
There's
I'm
two.
just trying to confirm that those black corals are
That it is actually
what we think it is.
stichopathies, yeah.
Just document it. Yeah.
He wrote here. Is that a sea pen? On the right bottom?
Yeah,
right?
it's Umbellula.
Sorry.
Is that the same as we had
See,
before?
but yeah, I think so. Again, I didn't see the first two. Yeah, it's fine with me. I don't know if you've seen it or not. So Russ,
Not that one.
Pretty
No,
sure
the
it's
little red one we tried
the little stalk, right?
Yeah.
Yeah. Okay. Let's take a look at this. I think it's Umbellula, but we will take an image here so that we can record that it was here at 1793 meters depth.
Yeah.
Yeah, okay. Thank you. There's one laser. There's two lasers. All right. Now let's see.
Hang here for a second while I reboot the video volume system.
Whoops. Excuse me. Sorry, Deb?
Can we just hang here for a minute while I reboot the video volume computer?
Oh yeah, absolutely. Let me just get in here and we'll hold it.
It's still being, like, the secondary one is still running, it's just,
Roger that. Okay.
it's me capturing. It's coming up now.
You gonna try?
You gotta click on the draw, you see. It's bad.
You got the rock in the back. There.
It's a lighting line as well, so just,
10 seconds. Just
Okay.
did
Oh.
it. Good job.
yeah, it froze it.
Okay.
Back.
All right. So we're rebooting the video system here. So we're gonna hold for just a minute.
I'm good.
You're good?
Yep.
Okay.
We're good on 10 seconds.
All
So
right.
this pink stalk in the center,
That's-
clear, that's the main,
That's the main axis. Yeah, that's coming up through. But then that's all that. All the rest is just fleshy bits,
yeah.
right? And so if there were eggs, you'd see them in here,
Right.
in here. So not much there, but you know, it's massive polyps. Sit there and feed.
Yeah.
It is amazing. You're not bored by it? I see it all the time.
You always see something new.
You do. Love it. All right.
It's the closest we get to an alien
We're
world.
gonna square up again. And something like that.
Glass sponge up there.
Is that me or you? All right. There we go. Just make a quick note. Let's see that. It's a bit hot. Try to keep the camera pointed up so we have some time to see things.
Yeah, I think Steve also specifically requested that if he was going to be analyzing
Yeah,
the video,
right.
it makes it
No.
a lot easier for them.
Yeah. There's always an area that's just hard to, try to quantify things on the edges of the image.
For sure. Oh, there's another one right in front of our eyes. We don't need to zoom in on
Yep,
it.
no, that's right. 1787.
You know, a bit more light.
Yeah, a little bit more would be good. I tried to take it up a little bit. A little bit more, Ben, that'd be great. These rocks are so dark, especially in the corners, we don't really get to see everything. That's great.
Oh, there's that stick
Yeah.
thing. Did a few.
You mean that little, yeah, it's a small whip, you're right. Let's see if we should,
See there.
Oh.
no, yeah. No, it's metallic gorge again. I don't,
Is that a A sponge
Really
on the
small
side of the
ones.
rock coming out? Oh no, that's just the rock face.
Yeah. Nice. See, they're flattened.
Go ahead, Donna.
Yep, but I just sort of lost my heading up here. I'm gonna try to come back around to starboard.
Yeah, roger, no problem.
Another small whip there. All right, let's take a look at this one,
Yeah.
please. That's what I was looking for. Just want to confirm document. There's always this itch to grab the thing and try to zoom in right away. Where Russ does the best, you know.
I know, they're better at it
You know,
than
I could
us.
just say, I mean,
Looks like we're gonna overshoot this waypoint.
That's fine.
Roger.
right, so this looks to me to be a black coral sticopathes. We'll take a zoom in here and look.
Okay, you are clear to engage.
Clear to engage. Roger that. Going for enhancement.
Just dump it in.
You are clear to engage.
That's it. Here we go.
Like that.
It's blowing away from us. That is a nice specimen.
It's beautiful.
I'm just gonna go in and not able to control that zoom very well.
Get a marker right.
A bit more.
So are stigopathes an octocoral?
They are a black coral.
Which is different than an
Yeah.
octocoral. They don't
Right,
have
so
the like
so
8
the,
things.
so the polyps here, there's 6 tentacles to the mouth rather than 8. Octocorals 8.
Okay.
Black corals have 6.
Thank
I
you.
just saw this part here that
Bridge.
right. So that's
Go ahead, Donna.
Yep, got her back. Slowly working my way back to starboard.
Roger that, thank you.
ten count. All right. Perfect. All right. Thank you. It's interesting that we don't see associates on those things. Not until the colony becomes bare in some spot. All right. I didn't check Slack.
What is that?
What's this?
Yeah.
I don't know what it is. Let's take a look at that. It's
Yeah.
Looks like a heart.
I think it's a Venus flytrap anemone with stuff on it.
Enemy and
Oh, that's cool.
Yeah,
Yeah, you're right.
it is. With
it's
crinoids
like
on
probably
it.
not, what is it? No, was it? No, how's it? Horrors it was.
Little house
No,
of
no,
horrors.
shop
Yeah.
horrors, that's what
Little
it
shop. That's right. That's right. Anyway, yeah, that's what that is.
Cool, thanks.
It's a Venus flytrap anemone with a bunch of stuff on it.
Barnacles. Crinoids.
Yeah, right.
It's pretty.
It's funny how that landed there. And yeah, I don't know how that landed there and colonized that.
That's
I
amazing.
guess it's not. Yeah, it's amazing how it wraps around that branch. Yeah.
It's just attached to that coral.
That's right. It may be actually eating something. It's sort of pulled in, but I don't know. Yeah. Pretty neat.
It has like a little hat that it's attached to.
Actinoscypha. Yeah. You want to see more, Russ? It's, hang on a second. We're good. You can take off.
Cool.
I just thought I saw serpulid on there, Glyptolasma barnacles.
Is it
Massive
on a
crinoids.
stigopathes? Or is that its stock?
I think so. That's good. I think so.
Okay.
I think it's probably an older colony. All right. Crinoid.
Whatever's off to the right there.
Yellow plexaurid like coral. We've seen that one several times. Good old bamboo skeleton. Not seeing a lot of coral rubble. These are like individual colonies that don't build up. I haven't seen any colonial hard corals today.
No.
Right? Now
We've
it's
been pretty
Zambia.
deep though, right? I mean,
Yeah.
I guess it doesn't always mean we won't
No.
see them, but
Or cup corals. I haven't seen cup corals, but maybe someone else did.
I think that's accurate. Yeah, we haven't seen any on our watch. There's a colony over there. It's probably just another. Got one of those Luxorets. Where's that at? I can't the gorge.
Thanks, Russ. It's nice coming around here.
I think it's a Luxor.
Yeah.
It's some big astrocytoma.
You can see the slope that we're on with this. Astroschematid with a interesting branching pattern. Take a closer look. Good? Oh,
I'm just gonna have to hold it.
yeah.
Yeah, they're good.
You can see the bottom half of the colony has its polyps pulled in. Looks like. Good. Are you? All right. I think we've seen that before. I think that's a.
Yeah, we're good.
Yeah, I think we're okay on that one.
Okay.
Russ. Sorry, I'm trying to get it squared up here. Thank you. He's right down the line. Got about 50 more meters to the next waypoint, waypoint 3. Where the depths are here. 71 meters.
There's another one of those weird looking corals. I think it's a black coral.
Should be
Keep the ship ready, so we can-
coming up. Look at all this habitat for corals and sponges. See more of a draped sediment here though. They like those rocky hard bottoms. That are often current swept, right? So you tend not to see
Yeah, don't pick up.
things when it's draped with sediment like this. I often think about the things we have not seen when I'm on a dive like this. That maybe there's an octopus we haven't seen. I haven't seen a lot of fish, I don't think.
No, that is
No?
true. And we saw a lot of fish yesterday.
Yeah. So I think we're facing more to the east on this dive, and yesterday we were facing more toward the north.
I'm always game to see an octopus. So
Another Stichopathes. Maybe I can turn up a little bit more here too. Something over there.
sponge. The stocked crino.
Yep.
Some of it's,
See, there's that similar sponge as we've seen before, big stalked. Is it Chrysocrinus? I don't know. Yet. that same sponge with a very small base of the older sponge looks like. So it's a pro-isocrinus branch. Another little black
Stegopathys.
coral whip, yeah.
And no, sorry. What's his name? Oh, we got a few things.
Little chrysogorgian there behind it. Another vase sponge back there. A lot of little stalks here.
It's a good little face. It's weird that it's
We'll go halfway in?
Don't look at
Yeah.
Keep flying.
its polyps are all retracted.
Yeah.
Strange.
Large asteroschemata in there, yeah. That's good. So you can see that in general these ophiuroids will hunker down in the middle of the colony and stretch their arms out and wrap them around. They'll maintain that position for a long time. Looks like an asteroschema intactum to me, based on the segments on the arms. But these will feed, we think, on the particles that are in the water column. I think that they're taking things off the corals. The corals are trapping particles, and then these ophiuroids will go and take particles off the corals. So it's not a parasitic relationship or anything like that. It's a mutual relationship, I think. So I'm going to zoom in a little more on the disc here. Give us some ID.
This does not look particularly. I mean, it looks okay.
It's a funky looking disc, isn't it? But I think it's still. There's ossicles, there's bones essentially are all around the central disc that you need to look at to identify species. But
Yeah, it's like very well defined, those
Yeah.
bones there.
that's great. Thank you. It's another chryso with a crab in it. All the way back out again. Okay. I feel like there's more sediment here.
Than yesterday, you mean, or
Just
in
during the
dive?
course of the dive, just noting that it's more, these are more low bait type flows, low relief flows. Maybe a little bit flatter here.
Is there an input delay on controlling the robot? Question from YouTube.
No.
Not with the controller. Sometimes the video feeds have got a slight delay, but not on our main pilot.
Cool.
It's obviously milliseconds, you know, but not enough for us to notice.
Actually, yeah, comprehend it.
Sorry.
Because essentially the communications is done on fiber, which is the speed of light. So. But then the electronics have delay to convert. So that could be 10 milliseconds, maybe less.
Oh, it was like the red stabs, like you
Yeah,
put
Debs.
it. I was
They have
like,
answers right
whoa.
in the YouTube. Yeah. Right. We used to type answers, right?
The scientists?
Yeah. Remember
Oh.
scientists used to type.
I think they might have taken away those privileges
Probably
after
did.
issues.
I don't know.
Not with us, with.
Come ahead. Yeah, come ahead. Okay.
Can we get a little more light, perhaps?
Yeah.
I want to tilt up, but I think it's going to be. It may blow out the center, but it just seems like it's so dark. It's just. There we go. Thank you.
Wonderful.
Hey Rich, we're pushing you forward.
A small plexar there. It's a red. All right.
Yeah, that's why I asked you to go to answer.
I'm gonna keep going unless something's obvious that we need to look at here.
Sure. Yeah, I think we've seen a lot of this.
I mean,
before.
otherwise we're not gonna make it.
Internal traffic, he said yes, I was able to make sure that this stuff.
I think Randy may come on soon, and I know she likes to zoom in and look at things and
Well, you've got to
curiosities, but
do your stuff soon too, right?
yeah. Another stalk crinoid. There's a crinoid, the stalkless crinoid, comatulid, is big. Yeah. You know, really don't see any crabs on the, in the the little holes, and not seeing a lot of crabs, not seeing a lot of sea stars, not seeing a lot of the, most of the things that are up off the bottom. Urchin there, I guess. I don't see much in the way of shrimp in the water. As soon as I say it, I see one, but. Yeah.
Oh, is that a fish?
As soon as we say we don't see fish, we don't see $1 million. Let's see if it'll show up. Yeah. Maybe a halosaur or a Synaphobranchidial. I think it's a halosaur. Got it.
I'll see if I can just fly for a little bit.
Yeah. Really not much current here. That could be a large part of what we're seeing, why we're seeing what we're seeing.
Really have to have like a happy medium, huh?
Yeah. Getting a sense too that the corals are oriented in different directions. Sometimes you can get a feel whether they put their polyps into the current or they're just the opposite. And I feel like they're all different positions with the heading of the ROV. Yep. Sediments, not even a lot of sea pens. Well, I did not see one that was, I guess, yesterday that retracted itself into the sediment really quickly. I don't think I've ever seen that before. I'm hoping we find one. The small Stichopathes on that rock.
A small something on this rock too. Or no, it's just
Yeah, no.
just a shadow.
Urchin.
Eyes playing tricks.
Crinoid. So we're out over sediment. Let's see here.
Making tracks.
2021. Scattered rocks.
There's Tinapathes, mostly green. There's a fall, or is it growing out, or did it fall? I think it fell.
It just fell over.
Yeah. It's like a Paragorgia.
Just quick zoom. I think so.
That's still
Alive.
Some living polyps there.
go from up there.
Yeah, so,
Yeah, the polyps are out, stretched, feeding. It's really amazing.
still alive and kicking, eh?
Yeah. I'll take a look at the base here and see what.
There. Although, no, I think that's different.
There's barnacles at the base there.
Yeah.
I'm assuming that's that base, or no? I think this is the
Yeah.
base here.
Sorry?
You can't blame me.
No, it's up, I don't know.
Don't know if that holdfast there is against the rock or whether it came off up top. I think
It's hard to say.
it is hard to say.
The way it fell, or the way it's growing makes me think it's a fall because there's a broken piece
Yeah.
off the top.
Let's get going in a second here because the barnacles are here
I think
too.
it's that one closer to the bottom that it was attached to.
'Cause it's like angled like the.
Look at this again. Nice tall barnacles. Yeah, that's what I'm thinking too. I don't know why a paragorgia would grow there and then grow into the sediment, which doesn't make sense to me either. So it's broken in two again. Like something ran into it.
Just be a
You know,
fish.
big fish. Yeah. Stickopathes over here on the other side. Nice. Interesting.
Yeah, you don't get to see usually a recently fallen coral. Yes. It's more rare than certain other events.
It's funky to keep good light on because you got the sediments that are blinding and the rocks that are black. But here we go. Another stickopathes
Pushing
off to the
forward,
right.
Bridge.
New waypoint, thank you.
4, 17, 11. Yeah. It's a bamboo. Let's see if something's on it.
There's shrimp next to it.
Hey, Tim, do you want to stop at that waypoint?
No. What? You mean waypoint 4 or do you mean waypoint?
3.
No. We'll keep going down the line. Yeah. She's working on it. Yeah, let's take a look at this. This is a branched
Yeah, nice.
black coral, I think. I thought it was just a bare skeleton, but it's got a bunch of stuff in it too. It's got a couple of crabs, I think. Barnacle. This is it. This is interesting. Alright, thank you. Let's check this out. These black corals, identifying them can be really tough sometimes because their branching patterns can be so different. Oh man, look at this. So we'll go and look at the polyps. So the question is, how do these crabs find this particular coral? Wow, because we don't see haven't seen this one today or anywhere else. This crab, and it must love these black corals. If I go in a further zoom or not, that's perfect. Okay.
Midnight.
What?
Midnight.
Okay.
Wow. It's a movement.
So you see the branching here. I don't know what this is.
It's
It's a black coral.
Taxipathes, maybe. Hoping someone else will chime in, but I don't know if folks are around. But
Steve's around, maybe.
I mean, I'd love to sample these crabs.
He might have
Let's
found.
move up here a little bit. There's the branch points there. It's a broken branch there, it seems. Also looking for polychaetes. Polychaete worms live on the central axis of this. There's one there. Do you see that? That's actually our branch. I'll come back to that. Let me get this crab. This is amazing.
Hey,
There we go.
I got one in, but I didn't get the second one in, but we have till midnight, so why don't you go work on it and I'll figure it out.
Yeah. Do you know if Steve's in the library?
I
I was
don't
curious.
think he is. I think
Okay.
everyone's asleep, so go ahead.
Okay. Well, I just want to have him look at this coral, but he'll look at it later. Sorry. There's a worm right here. No. I just had really good focus and I lost it. Here we go.
Oh, that's nice.
I think I'm out.
You collecting this?
I'd like to. This is a black coral that has a branching pattern I'm not totally familiar with. That can be really tricky. There it is. And certainly I have not seen these crabs before.
Okay.
So there's a polychaete there too. So I don't know where, where this would go.
Well, if you want the polychaete,
What we can
take
do.
a small piece, put it in the quiver, sure.
And then suction crabs.
Yeah.
What do you think?
Perfect.
I mean, we've been making pretty good tracks, I think,
I don't
about
know where we are,
1,700.
sorry. Waypoint 4 out of 7 waypoints? Or 8?
I don't know exactly.
Water one here, right?
We can back out and look.
1, 2, 3, 4, 8 waypoints. So we're about halfway there.
Yeah, we've been trying to really know.
It's good.
For right upstairs.
How much more have you got to do
I
on
am
yours?
like 10 minutes away from submitting, but it's like if this cover sheet keeps crashing on me. And so, like,
Go
I
do
feel
that
like
now
I'm gonna know.
while we sample.
Are you sure?
Yeah, it's going to be 10 minutes.
And then
Yeah.
we have until midnight. I just every time I
Okay.
fill it out and I try to save it, it saves empty, and I'm like refilling it out. It's endlessly frustrating.
You can't save it to your.
So I finally
The
figured it out. That I
attachment?
so I finally printed it as a PDF, and it was fine.
Yeah,
So
all right.
when
Okay.
you do it, just
All right,
so you know,
great.
I'm like massive.
All right, thanks, Randy. Okay, we've been imaging this black coral we came across here at 1705 meters. That's one that some people may recognize, but I do not. But I do know it has these interesting crabs that I would really love to conduct research on. There's also a polychaete worm on here too, so we're going to go for a snippet of this, and then I think we're going to try to collect the crabs. This is the importance. We see something new, we want to be able to document it, identify it, and provide that data about biodiversity and life to the public, the world public. Oh, I thought you had some news.
No.
From the library or something.
No, sorry.
Yeah. So we're going to sample some of this, see how it goes. And I'm
Cool.
wondering if
Let's get two.
my stick went. Yeah.
Sorry.
So if it's
Do you want that lower branch?
so here, right? Well, isn't that where it's on here, right? So
Yeah.
you could do that if it's easier, it's closer to you.
We could do that.
Might be a good angle. I don't know.
I think that will really work.
Yeah.
These are stickier than really, aren't
They are sticky. You're exactly right. So I thought more might be better because it would heavier, perhaps. You know.
The crab is like moving away from the arm, like no.
Yeah, they could.
Your
They could.
crabs, you're looking for 10.
What's that? Yes. It's reminiscent of bulbous.
So sample and C2.
Yeah. No. So this will be 354, I think.
I thought we had it. Was the.
Uh oh. 353 was the rock.
Oh, word. Okay. Then you're right.
Oh, you know what? Oh, I didn't write that in here.
I just told you.
This one? Yeah.
Yep.
Yeah, 353 is the. Yeah, you're right. 351.
Okay, we're headed for number 2? Yeah. No, yeah. There it goes.
You're not going back for a crab, or you're gonna slurp one?
Slurp one.
Okay.
So we're gonna get a separate number.
You wanna do that?
Separate number.
Okay. Then we can do 5 or do that.
Great.
Do you know what jar you're gonna be going into, Russ?
Number 2.
Number 2?
No, sorry,
No,
no,
jar.
jar.
First section.
Just so I.
One. That'd be number 1.
We're still
Okay.
number 1. Yeah, we don't have
Just
anything
so
in
I
there.
associate them in the notes.
We used jar 5. We used jar 5 for a cup coral, so they did see a cup coral. They did sample it. Okay.
Right.
Nice.
Let's get this done.
All right, we're gonna go for jar 1.
The one going, the arm.
What do you think, Deb? You seen these crabs before?
Huh?
Have you seen these crabs before?
It's crabs?
Yeah.
Is that what we were collecting?
Never mind.
No, I'm just.
Have a nice time
I
down there.
didn't-
Have a nice time over there. That's nice.
I didn't get a close
Sorry.
enough look at
It's
it.
okay.
I had to step out, but you know,
All right. All right. Well,
if you zoom in one more time, I
We'll-
can tell you if I've seen it.
we'll do that.
I haven't done a lot of ROV dives.
I just did it 10 minutes ago, and it was like, I'm not gonna say skeleton, but I'll do it again after this.
Okay.
Just gonna get a sample.
I wanna get the top one first.
You want the top one first?
Yeah.
Yeah. Okay.
Hopefully
This may get pretty wavy, you know.
Did you take that and put it in an RNA?
Joey to come in.
So did you put that in a special?
No. Don't need it in the special quiver.
But you're
No.
taking two different samples?
One sample, yeah.
Sample.
Q2 and jar 1 is what we're gonna do. Separate numbers.
I think it's easier when you can do that beforehand if
Well,
possible.
Yeah, it gets confusing
When they're linked so far away.
yeah. These will be one after the other. Yeah.
We did that once yesterday too.
Pan down.
Looks like they're still going. Yeah.
Repeat you on some.
No, way too much. So 92. Alright, we're good.
So we'll look at the morphology of these this coral and the crabs, and we'll do DNA barcoding on them, and we'll do other kinds of analyses. But here we go.
Just give us a second to get set up. I can't see. Okay, two views now. You see the little machines now.
Whoa, nicely done.
Like flipped it right in.
Wow. Off.
Oh, I'm in the washing
Off.
machine eagles.
Okay, so
There's no happy carbide. Look at that.
We're going for the second one, same
Same
time.
shit. Nice. Same technique.
yeah, that's fine. Yep, same thing.
Fantastic.
Ready to go.
No, just- Wait. Good. Come on, no, branch.
Let's call it 355 to 356
Okay.
if it goes.
This is 55 stuff.
What we do is you get it off.
Come on now.
It's tough. I'm not like,
Hang on, Ben, so hold on.
You're stronger. There you
Hold
go.
down and I'll keep posting.
Yeah.
No.
You almost go back for it a little bit. And then fight this one.
Wow. Those back legs are modified to hold on to that coral. They're not.
snip it.
Wow.
Some precision to work with.
I can get close to the nozzle. Pan down a little bit, please. Somebody. We'll probably. Can I do it?
Don't let him get you. Don't. Wait, hold on a second. No. Don't let him do it. It's a matter of will.
Yes, it's tough.
Let's come run all day, he can.
Wow.
Slowly moving.
This guy's like, man.
Tough crab. Wow.
Thanks, good, again.
Okay, yeah.
So, I don't
Nice.
Snip the coral with them.
I'll get off of this. Steven said that we'll take a look at it.
We'll do a little snip snip.
I think so.
Perfect.
Just amazing. Watch this battle royale. No one's backing down.
Freeze. I'm sorry. Somebody zoom.
Me to zoom? Where do you want? Down lower?
Oh, actually, zoom back out again. So.
Yeah, yeah. Oh.
Can you do that? Do that, man? Okay. Up.
Oh my God! You gotta go in with the finger.
Hold on. Okay.
Just stay still on the screen. I'll turn it over. I can see it.
There you go.
Just leave it there. This is
Yeah, you might be able to scrape it off now.
yeah. Freeze your arm. Is there suction?
It's full. You can fix plastic as quality
Yeah.
Excellent.
Wait for it. Come on.
Who's more stubborn, the crab
Did you see
or
it?
the slurper?
War of will.
Mm-mm.
You see it yet?
No.
Tenacious one.
I'll leave this up until
Keeping an eye on that.
Is that it? Like up at the top?
Oh, maybe right there at the.
that's the one that
The
really
edge?
hit you in.
Oh, is that right? Man, they are tenacious. Yeah, everyone. So we have suction sampled these two crabs, and we have a camera on the collection container, and we can watch them drop into the collection container. We're still waiting to see the first crab is on the edge of the jar, ready to go in, and haven't seen the second one yet. they're tough. They have these little hooks on the end of their legs that really allow them to hold on, like you just demonstrated. Right, so it was 354, 5, and 56.
Mm-hmm. Exactly.
All right. Wow, nice job. That was fun. Well, I wanted to watch how you did that. You see it?
I'm in.
All right. We're just finishing up
I sent you the link. It is super straightforward.
to make it a PDF though.
Making the PDF is not, but writing the checking the boxes is really easy.
Yeah.
I don't know why
But I
had
thought
a
it
hard
wanted,
time generating the
it wanted
PDF.
you had the ship time, day rate, and all that. Sorry, I'll just admit this.
No. I'll do it. I'll email it to you right now. Hi, everyone. Sorry about the radio silence for a minute. We are changing watches, changing shift leads. Russ, you need to give me 1 sec as I send Tim something by email instead of Slack. And then... I'm not. I'm sorry. I didn't... It's my fault. Slack is not working on my computer very well, so I'm going to try to find another way to send it so that I don't have to get stuck.
I don't even like getting documents on Slack. Slack's for talking. I can't. If it's on my phone, I need it. Slack is talking.
Different people.
Exactly.
Okay.
Okay, moving ahead.
Thank you. We are moving ahead. So I am gonna catch up here. So I am.
Yeah, we just did 3 separate collections. The black coral that we don't know exactly what it is, and
Yep.
then the 2 chirostylic crabs. Each were assigned a different sample number, so now we're just making headway again. because-
Perfect. Yeah, makes sense. Okay. Well, hi everyone. I'm Randy Virgin. I'm a faculty member at Boston University and by hook or by crook, the chief scientist of this expedition. I am here with Anna Gauthier, who is a PhD student shared and co-mentored between my lab and the Kagan lab at Harvard University and Boston University. And we are in the room with our fantastic ROV pilots, Russ and Ben, if you guys want to say hi.
Hello.
As requested. And Deb's our amazing marine technician who does all the things, who's waved, but you can't see her. But I promise you, she says hola. Okay. So we are here, and we are making tracks on the bottom of the sea. All right. Where are we? Where is here, by the way? I forgot to tell you. We are in the US EEZ, in the sort of in the area surrounding Howland and Baker, but we're pretty close to the international dateline and the equator, but not quite on either one. But we will cross the equator probably later on today or tomorrow. And we are. Oh, have you guys been seeing these book corals?
Yeah.
Okay. We're exploring an unexplored, unmapped, unnamed seamount, although it is sort of mapped now because we mapped it. Mostly Deb's mapped it, and so gets a ton of credit for that. James mapped it. The ship mapped it. You can see it takes many hands make one map. Let's look at this dead sponge for a minute.
Happy World Hydrography Day.
Is it World Hydrography Day?
Yeah.
Hey, happy World Hydrography. Oh, okay, hair in my mouth. Hydrography Day. And, okay, we can keep going. Thank you. And we are hiking up the side of a mountain at night with nothing but a flashlight, trying to see what we see.
Shout out to Jan.
Shout out to Jan Whitting. Mm-hmm. So, yeah. The focus of this expedition is on characterizing unexplored deep sea coral and sponge communities, but also looking at associates of deep sea corals and sponges, and specifically looking at corallivores, or predators of deep sea corals and sponges, looking at the damage they cause, the wounds they leave, the potential wound healing, and the innate immune systems of these very ancient, very early multicellular basal metazoans. Okay, this I think is a halosaur. Cute. It sounds like you guys have been seeing a bunch of them.
This is only the second fish Tim and I saw in
Oh.
the whole
Well, then
morning.
let's take a good snap zoom on it, and then we'll keep going.
Usually the center on fire.
That's all right. I think we've got enough so that somebody will be able to identify it. Okay, we are. Oh, and he's gonna follow us. See? So fun. So it looks like you guys are finding tons of corals, and then I get on, and suddenly it's just rocks and sand.
No, we've had patches of this happen where it's like sediment, and then we'll come up to, a rocky outcropping, and there'll be.
We're almost at the top of the ridge, so.
Got it. Oh, okay. I see where we are.
Yeah, the boat's passing waypoint 4, but
That's
we just passed waypoint 3. The ship, excuse me.
Seeing the occasional sparse but cute urchin. I'm gonna snap zooms here and there. Is it okay as we're flying?
I'm using this one.
Okay. Just making sure I'm not making you crazy.
Otherwise, it's gonna headache.
What?
Just make sure you hold it for 2 seconds or more, or you won't get a frame grab of it.
Good point. Thank you. I was just sort of checking in to see what it was.
Yeah.
Mhm.
So you know, I can stop and slow down.
No. I mean, we're just trying to go until we find something, but just as we go, I might sort of zoom in on things. What? Is that okay?
Yeah, it's good.
Since while we are making tracks here, we are happy to answer questions. So if you have them, please let us know. I'm going to zoom in on this a little bit. Just take a quick look. Okay, thank you. Urchin again. A lot of them.
Yeah, there's been quite a few.
And have you guys been looking at this already? Has this been characterized? It's dead. So they're hydroids. Okay, never mind. There were a couple of oafs on one earlier. What was have we collected rocks? We've collected lots of rocks. Okay.
Just 2. We could get one
Yeah,
more.
We have
right?
3.
Let's get more, because the basket's so empty.
I think this seems like a good place to collect a good
Wow,
rock.
it's like, oh, this is what's the choice for?
I think it's pilot's choice. Would you like to pick a round,
Like that.
crusty crust rock?
Crusty rock, crusty rock. What's the
Sure.
square?
No rush, though.
Triangular. Round. Let's go and see what we got.
So we are collecting rocks in collaboration with our colleagues at the U.S. Geological Survey, who are using these rocks to help to characterize the age, the mineralogy, the geological history of these seamounts, which, again, were previously unmapped, uncharacterized, unlooked at, totally unexplored, and so
By humans,
by
anyway.
any humans, that's right. Certainly the brittle stars, the crinoids, the corals have been all over this place for a while. We are that's your rock, 1,675 meters below the surface of the sea here in the U.S. EEZ of the Howland and Baker area in the central Pacific, equatorial Pacific, and we are collecting this rock. Could you take, yeah.
Come on, do it.
There's nothing dialed in except a tiny bit dialed in that. That's what I said. Other than that,
There you go.
Happy?
it's pretty good. And we've
Hey.
got lots of rocks.
Lots of rocks?
Yeah, rocks are
Because
priority.
then you won't need to trim down.
Crack.
You're fine.
Ooh, that is a crusty crust rock.
You may have to adjust the trim actually.
Oh,
That's right.
It's soft.
It's just crust.
Well, there's always a
A nugget.
nugget inside, exactly, that sort of catalyzes the crust.
Is this for GS?
Yeah. Yep. So you might want to pick a.
It's wrong shape.
That one will work. We can if we can pick it up. We can get it. Or this is a captain's choice, pilot's choice rock collection. What?
You want to go for a different one? That's fine.
Don't forget. Okay. That light. Okay.
Oh, I won't.
Okay. So we've just had a shift change in our ROV pilot, so now we are flying with the fabulous Adam and Chris. Hello.
no.
Hello.
That's,
What?
It wasn't a mistake then.
I said you were amazing.
No.
Amazing.
Step down from fabulous.
No, I was just picking different adjectives. My goodness. Picky room. We have 4 amazing and fabulous ROV pilots in the room right now. We've just switched from 2
And we
to
have
the other
Ben,
2.
And also we have him.
And we have the most amazing Ben in the entire world.
Is that an operating procedure or is it documentation?
In regards to.
Alright, that's broken already, so you can't blame me for that right there.
No, yeah, it is.
It is your choice. So if you're looking, you can see these lasers, which are 10 centimeters apart, and they help us to gauge the size of the rock to make sure that we can pick it up and put it in our basket.
We'll put it in the discussion tab. but I
Keep
would
thinking
say
Steve's gonna come in.
No, that was the amazing, fabulous Ben.
section.
Alright, we need to fly away.
What I need to do is porch over the touch it. Very slow.
Yes. Alright, that should be good.
That's good. Did you get a good?
Look down.
Yeah, I got several pre-sample photos.
Okay. So we've gotten some good photos already and good imagery. So we just need to get in the box. Perfect.
Oh, hey. That's still there. It's one right.
Excellent. We are.
We'll do another rock wall in here.
I think we're good. We have successfully collected a rock, put it next to the wiffle ball of science in our push board, which are hanging out, waiting for some use. Wiffle ball of science is for photomosaic size scaling.
Depth.
The depth is 1675.
Okay.
And the sample number is
357.
357. Okay. And so we're gonna make some tracks. Okay. So that's the lot. All right. Today is hump day on the ship, which means it is our halfway point. Isn't that amazing? I feel like we just started, don't you? But we're 10 dives in. Correct. So
9 dives left?
maybe 10.
10. Oh, then that's halfway.
That's what I'm saying. So we're 10 dives into this cruise. Which is actually pretty good, given the fact that we've had a really long transit all the way from Hawaii to get down here. It is true. So I'm pretty happy with our progress. We've got stocked crinoid, dead hexactinellid sponge going on. Another one of these very tall bamboos. Let's just get a quick estimate of size here. We can just kind of take a look. Oh, actually, is that just sorry, can we take a quick look at that?
Hop over.
I don't think we need to set up in stock. Just
No,
kind of just a fly up the top to
pause.
see how big it is. And I'm looking for associates. This is very tall. They've been really tall here. Woof.
Hello.
There we go. Okay, there we go.
It's about 4.5 meters.
4.5 meters. That was huge. Thank you. Did you mark that? Could you?
Do you want me to put 4.5 meters?
Yeah.
Okay.
So somebody is asking us whether or not the coral larvae settle better on ferromanganese crust, or do they need gaps in the crust to colonize successfully? It's an excellent question. If you take a look at these corals, they are settling directly on the crust. However, what makes some crust good for settlement versus other spots good for settlement is an unknown question. So we're gonna, but part of the, it could be the crust itself, it could be the microbiome, it could be something else, like a biofilm or another organism that helps to facilitate settlement. And really, there isn't a huge amount known about deep sea coral larval recruitment for obvious reasons, right? It's really hard to study. And then even if you see recruits on the seafloor, and we do sometimes, the answer is or the question really, I should say, is just because you see recruitment now, does that recruitment going to lead to a successful adult coral? And that is a question that often takes decades, if not, over 100 years to answer. So the timeline to answer these questions is really outside the scope of a typical project, but there are other ways to get at those questions, and a lot of our colleagues are really interested in experimentally trying to determine what makes good larval settlement ground. I'm going to sort of snap zoom as we fly. Is that okay?
Yeah, yeah.
Okay. Ooh, no, I don't have control. Yeah, now I do. Okay. And then I'm going to take a look up here. We just have a crinoid. A lot of this just seems to be sort of dead stuff. I'm going to take a look at this bamboo colonized with... Is it a crinoid or those hydroids? And an ophroid.
It's up top there.
Yeah. So for example, if you take a look here, this is no longer a living bamboo, but you can look at the base and it's clearly attached to the rock. This white spot over here is another base of a former colony. And then there's a sea cucumber in the background. All right, I'm good. Thanks. So it's really interesting how some of these dead sponges are still providing enough structure, even though they're no longer alive, to be able to support a number of organisms, mostly ophroids or crinoids or hydroids, which will hang out on them. We can keep going. I'm just going to kind of zoom as we go. This is unusual. It's got like a little bit of a yellow tint to the yellow pinky tint. Can we actually, sorry, I lied. Can we scroll? No, just scroll down. Scroll down. Just look down. I'm just going to see if anyone on Slack has any interest in this. And our team. Well, let's at least get some good imagery. Oh, look at that. See how it's got a spiral?
What is it?
It's, I think it might be a sea pen. Like
Yeah,
a very
'cause-
long sea pen that's whirling around. So I don't really think I'm going to collect it because the whole thing will come out. But I think this is a sea pen. I'm like, you see how it's spinning around the base? No, not the base, the stalk. It's got the polyps are, oh Jesus, I gotta get better at flying here. It's not consistently in one direction. Take a look down and it's gonna start to spin right around here as it whirls around. That's a very long sea pen. Let me just get some lasers on base. And you see sea pens settle on sand or in sediments more so than on rocks. They have a peduncle that they can use to sort of anchor themselves into sandy bottoms. So it's a very different, it is a coral, but it's a very different habitat for settlement than, for example, the hard rock substrates we were just talking about with the other deep sea corals. So sea pens are, it's a good thing we were just talking about this, an exception. Thank you, that's a very tall sea pen that caught me off guard. Woof. 1.5 meters, 2 meters? Something like that.
Oh, yeah.
Okay.
Not more than 2 meters, maybe 1.5 meters.
Yep. Did you get that? There's an Anthomastus mushroom coral. Another dead stalk, but always providing habitat. Anytime I think you can get up, out on top of the substrate and gets a little elevation, I think it provides an advantage to food capture. and flow, and so I think that's why they provide so much structure. Here's another excellent example of a dead sponge providing structure, vertical structure that these ophroids, these brittle stars, are taking advantage of. Yeah. Shaggy but useful. Thanks, Adam. Little sponge recruitment. You can kind of see all these little white spots. Oh, actually, sorry. Can we go back to that sea star that just is down? Yep. I'm going to ask you to collect that after I get a good look at it. So what we're looking at here is a putative corallivore that I've seen on other corals, but we haven't really seen it so much on, on the seafloor. And one of the experiments or projects that we're doing is looking at stable isotopic signatures of predators and prey, trying to link them. And so it's really nice to get a couple of examples of the predators that are not directly on their prey, so we can see if their signatures match. So it's funny, we're coral and sponge people playing with all kinds of interesting organisms that are preying on corals and sponges. Also, I'm kind of wondering if when we pull this off, we're going to see a sponge base right underneath it. Want to take a bet?
I don't think we will.
It's possible.
No, I know, but
Yeah.
you asked me if I wanted to bet.
Oh, there you go. Well, then I think yes, and you think no.
Perfect.
Let's see what we get. So can I get a good zoom on it before we pick it up?
Yep.
Great. I don't know if this is the same kind of star actually, but it looks. I think it is. Hang on, let's. Tim Shank, if you're in the library, could you confirm, please?
See in the dry lab.
Can you read? I think it.
It's
Yeah.
one of the two. It's got this undulation thing going, and that textured dorsal surface makes me wonder.
So I think it's
But
Hipposteris.
But it's closely related.
Yeah.
It's-
So I think it'd be a good candidate for
yeah.
isotopes without
yeah,
being on a
it's
coral.
not Cerciaster,
Yeah,
not
yeah.
the other ones that we've seen.
Okay.
Yeah.
Thanks, Tim.
Yeah, I'd collect it, I think.
Yeah. All right. Let's collect it, please. And while you collect it, can we also see if there's a sponge underneath it that it might be eating?
Underneath it?
Mm-hmm. So like I don't want to lose sight of the spot where it was.
Okay.
Thank you.
4. Even though it's 5, but it's 4.
Maybe we'll switch the lids because I think there's.
No, we edited it.
Oh, you didn't? Okay.
Yep. Nice. So there's a question about pressure. So there is absolutely crushing pressure at these depths. If you have seen any of our styrofoam cups going down as we descend, you will see what happens with pressure as you descend. You basically compress all the air escapes. And so organisms that go up and down through tremendous pressures either can't handle it or they have incredible adaptations to handle it. Yep. Oh, there we go. The pressure is 2,405 pounds per square inch right now at 1,638 meters below the surface, just to give you guys a sense of perspective. Adam, our awesome ROV pilot, just pulled up that screen for us so we could give you the exact numbers. Thank you, Adam.
Just put the overlay up for them at home.
Oh, great. But as you guys have also said, organisms that have sort of air-filled cavities are going to be much more sensitive to pressure differences than those that don't. So something like a fish, which has a swim bladder, which is basically a air-filled balloon that they can use countercurrent exchange to inflate or deflate across different pressures. They have to move slowly through major pressure gradients or else it will rupture. They need time to adjust the air bladder to the ambient pressure in the area. Some organisms, if you've ever seen people go deep sea fishing and they bring it up too quickly, you can see a ruptured swim bladder. It's a really common thing. Yeah. So one of the other questions I see is how come, and yes, we can keep corals alive for 2 or 3 days in the lab. We can keep them cold, we can keep them happy, and we see their polyps out. They do all the corally things in the lab, so we can keep them alive for a couple of days after collection. The next question I see is, why don't we use machine learning assistance in real time for species classification? Ooh, there are so many reasons. Oh, sponge underneath, maybe.
I got my eye on this spot.
Okay.
We're going to cover over.
Yeah, so once this is collected, I'll answer your question. Perfect.
I'm gonna try to get the coral inside of there as well, same spot, or just
No, I just want to look at it.
Okay, we'll put it
Very
there.
close. Yeah. Focus on the view the camera.
Mm-hmm.
What did Tim say was the potential?
Hypastria. All right, let's take a look at where it was feeding. You know the spot?
See if I can remember it. Now that we've zoomed away. Yeah. It is right there where the sponge base is.
This one?
No.
Up here, right? No, up here. It was here.
It was absolutely that blue spot.
I think it was this spot
Blue
right
spot?
here.
Yeah.
Can we?
Yeah, right there.
Yep. So I think this thing was feeding on. Oh, I can't get any closer, but this right here. Don't you? Maybe?
Yeah, I mean,
was
it's
underneath it.
Within the realm of possibility, certainly.
So let's call that a possible. Could you hit spongivory and then write possible spongivory interaction? We've seen those eating sponges before. That's great. So you can see that this is an odd shape for a sponge base, which either means it's growing or it means it's been partially munched. Okay, so what was the question? Oh yeah, the question was, all right, we can make tracks, we're good. Thanks. Yeah. So the question was about AI. So first of all, there are a lot of people interested in trying to do this. It's a really good question, and there's a lot of people interested in it. However, there are a number of issues with it. So number one, the software and the technology are being developed, but they aren't quite there, so we can't just use it. It's all in development now. Number two is that a lot of these organisms are undescribed, and so we can maybe get them to family level with something like that, but getting them to the level where we could really describe them accurately is going to require more traditional taxonomy first. And then some of the characters are so slight that, that AI training would be absolutely tremendous. So there's an awful lot of work to do before we get there, but gosh, that would be wonderful to be able to have that level of identification accurately completed in real time. But I think we're still a ways away. Okay. Can we take a quick look at whatever that is to the right? Just want to see what it is so we can document it. You can see the bamboo whip.
New species too.
Yeah.
In every trip still, which means we haven't reached saturation.
Okay, so we have seen this before. I'm just going to take a quick snap zoom on the associates here for Tim. This looks like it's either a plexaurid. Given how bendy it looks, I'm just going to tentatively call this a plexaurid, but it could be Acanthogorgia too. It looks like there's a bunch of ophs, crinoid, several crinoids.
Hydroids.
Okay, we're good. Thank you very much. So you can see
that base.
And then yep, that was that's right. Another bamboo whip. I'm just gonna kind of try to snap ID on these as we go. Looks like this is another plexaurid. No ophs on that one, which is there was an oph at the end. Yep. Another anthomastus coral, another dead coral base, another bamboo whip coral. I'm just gonna take a quick look at this just to make sure we know what it is. Another bamboo whip. This looks like it's probably the same plexaurid with crinoids and ophs. We've got this lateral view, but that's good. Okay, thank you. Perfect. What blue thing? Little sponge in the back? Yeah. Ooh, very big dead sponge, hosting lots of life. So it's a glass sponge. It's dead hexactinellid that has a whole lot of structure left for crinoids, hydroids, ophoroids. And then in the back we have our plexaurid again, more bamboo whips. An oph is an ophoroid. It's a brittle star. As opposed to an asteroid, which is sort of the more traditional sea star shape, which is what we just collected in that putative Capastria. Can I take a look at this one, please? Okay. It looks a little funny. It's all tucked in.
Yeah, we saw that earlier on one too. It didn't have as many associates though.
No, it's got a lot of associates. It is very tucked. All right. I am good, thank you. We've just passed waypoint 4, so let me write that down.
Stickopathies and a bamboo whip. Right.
Thank you. 21, 26, 16, 12. Okay, there's a chrysogorgid, I think. Yep.
Yeah, you're right.
Another bamboo whip, I think. Or is that Stickopathies?
It's
No. Okay, Stickopathies. We're just traversing up this slope here. Oop, we're gonna have to stop and look at that. There we go.
There we go.
That's deep for a madrepora.
Is it madrepora or an Alexandria? How can you tell?
I can't. Well, so we need Alexis for sure, but well, let's take a look. So what we're looking at here is a scleractinian hard coral living at 1,607 meters down. So scleractinians are calcifying. They calcify with an aragonite skeleton. And it's amazing for them to calcify this deep because the aragonite saturation state is not typically favorable for these. So I'm going to
try to just get you a little bit off the side
Yeah, exactly. I can't quite get a good read on the polyps with this lateral view. Or I need a more lateral view. This is a really... Okay. So I don't know, but I think this is Madrepora because of the zigzaggy branchy pattern that we see on the polyps, but it could be an Alipsamia. It's really... I'm going to try to get a better zoom here. But this is... We're going to get... let's get some lasers on base. Here are the lasers. So let's see how big it is. Let's first get a sense of that. So we're taking a little data here. I need to move it over so I can get a sense of it. Okay? The lasers are 10 centimeters apart, so it gives us a sense of scale. And then I'm going to do some zooming. What is going on here? I don't know what that is.
Tim is also confused, so is that just
He did. I think it's a Madrepora, but I'm not really.
It's thick like an Alipsamia.
I know, but it's zigzaggy like a Madrepora.
Yeah. Here.
Maybe it's a hybrid.
Oh, the first hybrid.
No. So these look like a little colony of anemones that's kind of set up right here. You see this?
Those aren't. They also. Yeah, barnacles. I know. I see the tentacles, and I'm like.
I'm not sure. Uh-oh. Let me see if I can get a better look. Okay, let me see if I can focus here. Working on it. Could you?
Oh, you want to focus it?
Yeah, I'm trying to focus this, and I think I don't know if I actually have manual.
Now just like kind of in and out.
Oh. Perfect. That's right. I don't mind going manual. I just want to make sure I actually have it. Okay. I'm not sure if I'm making it better or worse. Let's go the other way.
There you go.
There we go. I don't know.
Yeah,
You're
I'll
right.
take a
They do
frame
look
grab.
more like... Yeah. Well, let's just take a frame grab and we can figure it out afterwards. But either way, that's a very cool associate. It's clearly set up and colonizing the top of this. I'm going to take a little tour to colony here. There we go.
Barnacles, Tim said.
Barnacles. All right. Look at this beautiful base. Wow. And this very... Oh, look, and it got broken off. This is interesting, you see? It got broken off and there's a new polyp forming on top.
It's pretty.
Can
It's
you
a nice
note
frame
that?
grab.
Can you actually call it a frame grab and actually write wound healing? Maybe click the ivory button and hit wound healing too, just so it pops up for us. I'm not sure if somebody ate it or if it just broke off, but either way, this is a nice example of wound healing. And it looks like it broke off in two places, right? Can you see that? Up here and down here, and in both spots, it looks like there's polyp recovery, which is really interesting. It's one of the things that we're thinking about in this cruise. Okay. Fascinating. This is why a close look is always worth it. Mm-hmm. I am not seeing any other associates except for this crinoid on the top.
Holding on for dear life.
I want to see if I can... You see, it's definitely making some abrasions right on the side of the colony.
Yeah. Very.
Okay. Cool. I just want to get one more look at that wound healing.
It's pretty cool.
It's pretty, that's very clear, I think.
Like an actual picture.
Yeah, you know what? I think I will too, because I think I'd like to. Okay. All right, that was really informative. That was awesome.
Did you want to take a little sample of that stuff?
I'm thinking about it. I think.
Are you asking me to go make you a pre-workout now?
Yes. It's extremely brittle. So the trick is to try to take a sample without breaking too much. So maybe down here, it's gonna break off like.
And then try to do it like, you see how it comes up, and then there's like a little gap behind it, and we try to do that to get like the,
You'll have to show me. I don't know what you mean.
go like right there.
Yeah, I think that's probably the way to go. So we're gonna sample this putative madrepora. And I feel much better about sampling about this now that we have evidence that they can heal from broken wounds. Which is what we've always presumed, but here it is, very clear. So this is going to be 2134, 1607.
This is like, yeah. Let's do like this.
This is going to be sample FK359.
Keep thinking someone's gonna barge in. Alexis must be asleep though.
Yeah, I'm sure she is.
Go for it. Yep. Oh, I missed. Alright, we're back up. That was just a practice run. Alright, go for it.
Nice. This might need to go into a bio box.
B and C are open.
Clear. That was the most stressful thing I've done all day. Other than fine. Stressful?
Bio box 3,
Bio box 3. Got it.
or B.
B. Sorry. Yep.
You did.
It's really good. That's great. Thank you very much. Okay, yeah, let's go.
Did you want that in there?
Sure. Waste not, want not. What's up?
Is this it?
Yes. See, because it was exploration and not OER.
Yeah, but this is it. 'Cause it's got all this detailed information that I'm not going to have. Right?
What do you mean?
Maybe clever.
It's a busy day on ship. It's hump day where it's our halfway point, but it's also proposal deadline day and like more diving day and sample processing day and round 17 million of an experiment day and everybody trying to work around the clock day. So a lot of things happening. Always busy
For
ship.
sure. Yeah, it's been a busy couple of days again for us.
Okay, and it's right next to that plexorid again and another dead hexactinella. I'm gonna zoom out and see if I can get a wider view. Is that the only one in sight?
There's a Sticopathes too.
What?
Oh, you want to zoom in or zoom out?
And just zoom out. I was just trying to see if that was the only. There we go. Ritigorgia. If that was the only scleractinian in sight. So if you just happen to see another one of those white ones in your
Okay.
cam, would you let me know?
I can just pop it down and take a look.
No, it's alright. That's great. I think we'll see more as we go up. If there's one, there's probably more. Okay, so beautiful Ritigorgia. I was just doing a quick scan looking for this little jelly that I've been looking for this entire time that I haven't seen. We saw it a lot in 2017, and so far it's been elusive this trip. We haven't seen it once. Another dead Walteria hexactinellid. Another one. Lots of little sponge recruits all over this rock. No, a half sponge. Is that a cup coral? Right there, that white stick. Can we take a look at
Right
that?
there in the middle of the right.
Yep. Can we take?
Down to the right.
Yes. Can we take a quick look at that, please? Oh no, I think it might be broken bamboo stalk from
Yeah, it is.
what's ahead. No, I'm good.
Broken bamboo stalk in a cup
That-
coral shape.
That's what it is. Thank you.
Exactly.
Excellent.
So we are traversing the seafloor at 1,597 meters below the surface of the sea on a previously unmapped, unexplored, unknown seamount in the United States exclusive economic zone in the Howland and Baker region near the dateline and the equator. And we mapped this feature last night and are diving it for the very first time today. And so you are joining us for an absolutely brand new first eyes to ever see this piece of seafloor experience. And the only difference between you and us is that I think we're seeing it a solid 60 seconds before you do. It's another plexorid. Wait, so can we take a quick look just right here, all these little white spots before we jump over this ridge?
On this like ledge of rock.
Yeah, see, there's all this stuff. I just want to, it's a recruitment event of some kind, and I just want to take a quick look.
You take a look long.
And then I think we're going to have a bunch of stuff right on top. So I just want to look at all those little white spots in the bottom.
And then what I'll do is
Yep.
I'll land and you can look down.
Sounds great.
That's rough.
Mm-hmm.
It's like Jimbo this morning.
You're not supposed to laugh, that just encourages it.
Jimbo, everybody laughs at that because it's so true.
That's not the point. Try that, right?
Okay, thanks. I'm going to zoom down. Nope. Porch is in the way. Nope.
Oh, I thought
I
it
was,
was on this rock. Okay.
no, I was trying to look at the this side of this rock. Sorry.
Probably things down.
See, right rock. Different angle.
Where else are you looking around?
So I'm gonna zoom up. So I want to look at actually almost basically right where your foot is, or anywhere along this way. I can probably work with what I've got right here. I just want to look at like this spot and this spot and this spot and this spot and this spot, so I can take a look. Is it okay? Can I zoom in?
Yeah, you happy there?
Yeah, good. So whenever you see these little spots, all the same size, they could be a recruitment event of something, and I was just wanted to get a look to see if it was sponge or coral or something else, and they look spongy to me. But they're actually all different sizes, so I don't know if they all recruited at the same time and are growing at different rates, or if there have been multiple recruitment events, but regardless of which, you can see an awful lot of little sponges right here in this crevicey section of this ledge. And I mean, we're not talking like one or two here, right? We're talking about quite a large number. And so we don't often take a moment to just look at these things, but I think it's really important to document when we see this, as we try to get evidence of what recruitment events in the deep sea might look like. All right, that was really helpful. Thank you. I know we have a lot to explore right on the top of this little bit. Where to start? How about the sea star straight ahead? Well, I think it might be eating a sponge. So yes, that's exactly what happened. Oh no, it's eating a coral, or I can't see.
It looks like a Chrysogorgia, but I cannot.
I know, it's very bizarre.
Which we don't see a lot.
No. So I'm gonna be pretty surprised if that's what's going on.
Yeah.
But I've been surprised before.
Yeah.
Yeah. So yes, there's larval animals that will settle onto rocks, and so recruitment events are lots of larval animals settling together. This is legit on a Chrysogorgia.
Interesting.
Is it?
Yeah.
Really? No.
Unless there's like zoanthids on it, but I don't think so.
What are you doing? I'm just trying to get, yep. Really? I'm going to see if I can zoom in on that foot and get a sense of this.
It'll be the last thousand heads I ever bought.
Wow.
Oh, which ones did you get? I meant to ask.
All clad copper core.
I have those. They're good.
What brand?
Goniasterid sea star. He had some ideas on lower level classification.
They like everything always gets all stuck around.
Okay, well this Goniasterid sea star is on a Chrysogorgid, and we're going to collect it.
The star or the whole thing?
Not the whole thing, but we're going to collect the star and a piece of the tissue on the top. Okay. And again, when we pull the star off, I want to take a look at underneath it. So if you could pull it off, but then hold it in your claw while we zoom right in and look at it right after, that would be amazing. Thank you.
Yeah,
It's true that the small sponges growing in a rock could also be a fully grown small species. But I think we saw bigger ones that looked just like it. I'm not talking about huge, but maybe fist size as opposed to pinky nail size. So it's always good to see that mix of sizes there. Did you write Ivory?
Yeah.
Actually, nope, it's okay. Keep your hand there. It's all right. I'm good. It's also not working, so never mind. Just do your thing, chicken wing. Okay.
Rain crab. Yeah. I want a DVRO.
You want a free frame grab?
Rain crab. In DVRO. Thanks.
Okay, now I'm going to zoom in before you take him too far away. Okay. Whoa. Those are definitely denuded chrysogorgid stalks.
Yep.
Okay. Well, as we keep track of who's eating who on the seafloor, I have to admit that's a new one for me.
What's behind the denuded stalks in the bright red?
It is some kind of associate
Okay.
that I cannot get a read on. So we'll probably ask
We
you
can
to.
come back
We
to
can
it.
come back to it, but they've got a sea star in their hands and those little holothurians on the side. Okay. Did you get a good pic? Did you get a frame grab of the damage? Sorry.
Got it.
Okay, we're good.
Do you want to look at the underside of the star?
Yeah, that'd be awesome. Can you turn it over for me?
Camera splitters.
What?
Camera splitters.
Okay. Let's take a look. Oops. Look at this chrysogorgid eating monster. Hey. You're eating corals.
Oh, I just put in
Oh.
on your frame.
Go here. I'll,
Try now. There you go.
well, they say you are what you eat. Stable isotopes will tell us. That's really cool. Thank you. And we're going to collect a little bit of the coral too and put it in the same
Same spot?
river. Yep.
We're going to quiver 6.
Mm-hmm.
This is sample number 360. Same sample number?
Yep,
The
it's
star?
going to be FK360.
Okay.
And we're going to put, I see you're going to mash it down.
I didn't want to do that.
My kids are calling at this exact moment, but they're going to have to wait.
They can go live with you.
That's where you collection. Oh my god, children.
Go for it.
Oh, it's Jeff.
That's what's up.
You want to talk to him? All right.
Speakerphone?
You really want to? All right. Hang
No,
on.
don't do that.
Let me do some outreach.
You want to do marriage?
It's like, no, Adam is not being serious.
So my husband and kids just called as they are driving from Boston to New York, and they're bored in the car, so they call me while we're driving.
So call him back.
They sent me pictures this morning.
Did they?
Yeah.
Ooh, is that your garden?
Yeah.
Wow.
No. Isn't that crazy?
Beauty.
Better enough, you think?
Yeah, that is perfect.
It's pretty
Can
good.
you see?
What's the last one you showed me?
I have 82 tomato plants of 31 different heirloom varieties.
That got huge while you were
We did it
gone.
from seed. I thought they were gonna be dead.
Looks so lush.
Yeah.
It's really good.
I missed the color green out here. That's a great collection. Thank you guys. And then we have one more to-do list on this coral before we come up, which is to try to figure out the associate side on them.
I can. The problem is as soon as I go out, then it changes. It's either or. Say it again, sorry.
Is it one more thing on our to-do list before we leave this coral is to zoom in and see if we can see what the associate is inside for Tim.
Sure.
Well, I lost some. What was it?
You said that was in your product.
I think you have a lot of it left, so
Yeah, most of it when it just really doesn't count.
I think we're fine. So the name of the coral is some kind of, it looks like a bottle brush crassicordid, and it's some kind of goniasterid sea star. We'll have to identify, confirm our ID. Ooh.
Like sticky, whatever it was.
Mhm.
We got some of it though. Still with
Yep. But I have not seen that kind of star eating that kind of coral before, so this is a really important collection for our evaluation of corallivory on the Seamount floor.
I don't know if we've seen corallivory of a crassicordid
No.
before.
Oh, we have for little jellies and things like that, but not for a
Not of a
not sea star predation.
At least in our 1.5 cruises so far in this area.
Yeah.
I'll leave that right before we get off. Okay.
Is that my favorite part?
Yeah, dinner doesn't matter.
Okay. It's not fun when the lid drops down.
Yeah. Been in that situation a couple times.
All right. Let's see if we can figure out what this associate is for Tim. So far, little pink blob is what I got. Looks like it's probably a crab because I think I see an arm coming up.
Mm-hmm.
That's all I got.
We can try to like. Oh, that's pretty squishy, isn't it, Chris?
What's that?
That coral?
It was not, I mean, it wasn't brittle. It was sticky and light.
Mm-hmm.
Do you think you could like move that little branch to see what the associate is? Just like,
What are those things to the left again? Sorry.
Those are sea cucumbers.
What
So what's interesting here is one of the questions we've had is whether or not when corals have an associate, whether or not the associate is capable of deterring predation.
Yeah.
My guess is
And
not.
I
but-
like to convince myself they make an attempt. Especially with the astroschema. Like
Mhm.
on that paragorgia the other day that we
Oh.
collected where it was like
Just peer in, yeah. And if not, we'll just move on and it's okay. But we'll try. Because I think it's an important observation that there is
well, their
an
working
associate president.
hypothesis
Yeah,
could be.
exactly. But I'd like to know what kind of associate we've got. Yeah, it is.
Yes.
Little chirostylid.
Yep.
Did you see it?
Yep, we did.
Yeah.
Thank you. That was perfect. All right. Thank you very much. So, all right.
Do you want to look around anymore before we go off?
Just to
Yep.
verify, what quiver did I put that last?
That was quiver 6.
Yep.
Okay.
So I think if we can just do a quick, like, look-see just to snap identify stuff, and then we'll keep moving on. I don't see things that we haven't seen before. Like, there's our plexaurid again. There was another bamboo whip behind us.
I got that already. Looks like there's stuff in the background over there, but I can't quite.
Yeah.
Probably just a sponge base from the
Yeah.
shape of it.
That's what it looks like. Okay, I think we can move up.
Yeah.
Thanks, Adam. Thanks, Chris. Thanks, Jimbo.
That was a really good collection.
Mm-hmm.
Yay.
Great. Looks like some stickopathes and another dead sponge with ophoroids. Coralline shrimp. That is stickopathes again and again. Can we get some more light?
Yeah.
Thank you.
Yeah.
Thank you very much.
That's fun. Luxorian. Industrial Seamount.
Yep, a lot of these dead sponges.
It's
Covered.
weird how many, and that they're host to so many things. It's not always a dead sponge have to host things.
It's true. You have to wonder if the conditions changed such that it just wasn't good for these sponges anymore. But it's clearly still a great habitat for lots of other things, like another bamboo whip, another plexaurid with ophs, and wait, is it a pycnogonid? It's a pycnogonid. We're gonna have to take a look at that. This is a sea spider. Looks like it's feeding on this coral. Can we kind of flip around the other side so we can get? Yeah. Thank you. Can you hit the corallivory button again, please?
What's up?
Man, I just think sometimes you get what you're looking for on the seafloor. Like, there wasn't very much corallivory at all, and then I walk in the room, and all of a sudden, boom. Maybe that's not true. Maybe there's a ton of corallivory before I walked in.
No. There definitely wasn't. Yeah, we just decided to ignore it. No.
No.
That was sarcasm.
Oh, thanks. I'm not really sarcastic.
I know. So I was like, I should clarify.
Thanks for the clarification. I needed that. Oh my God. Look at that. It's eating like half the coral. We're going to have to take a really good look at this. Tell me when I can zoom.
Is it eating the coral or is it eating something on the coral?
I have my thoughts, but let's take a look.
That other one
Yeah, so yesterday's pycnogonid was feeding on an anemone. This one is feeding on this. I think it's a plexaurid. Could be acanthogorgid, but I think it's
And it looks
plexaurid.
pretty stripped, yeah.
Well, I can't tell if it's stripped or if it's just the pycnogonid legs.
Base is stripped.
Oh my goodness. Or is it eating an anemone on the coral? So hang on. There's too many things. It might just be eating an anemone on the... I'm working on it. I'm just going the wrong way, as usual. Yep, I think it's eating an anemone on the coral. But look at that. Its proboscis is out, and it's just chomping. It's just I think of them as like the Dementors from Harry Potter. So I think that these aplacophorans are actually the culprit.
Of why it's...
Or some of these little snails.
Yeah, oh yeah, look at the...
Or...
So the sea spider's sucking out the polyp, is it?
It's sucking out the anemone that's living on the dead section of the coral, which probably is dead either due to a placophoran corallivory, and then there are these weird snails that I haven't seen before that are further stripping the tissue off of the stalk of the coral, which is also inhabited by an ophoroid, just for good measure, with a couple of barnacles on the side. So there's a lot going on here. So I'm going to try to get some more imagery of this and then of these snails. What did Tim just say?
A lot of aplacophorans.
Yes. to ask him if he knows what the snails
He also
is.
said anemones will take out coral tissue.
So anemones could just be wrapping them around, but that's an awful lot of coral tissue gone. Beep. All right, let's see if I can take a look at these.
Oh, it's like moving. Or maybe that was you.
I think
It
that's
might
me.
have moved its mouth.
So, Adam.
Yes.
I don't know how I'm going to ask for this. Let's see. What I want is going to be hard to get. I kind of want this coral piece cut right here where I can get these two snails and but a little bit of the base, which might still be alive.
Okay.
And I think I want good imagery, which I can get right now, of the coral from the base to the top.
Yeah. How much do you want? You want that little back branch?
And then these aplecopherans, I'm not going to try to collect, but because I'd have to kill the whole coral to do it. But I will just note them. Could you note them?
You want the...
Yep.
Do you want the branch too, or do you want to try the suction sampler?
I want the branch
This
too.
branch
Do you think you can cut
okay.
this right here?
Yeah. Alright, so just that last, not the one off of the main one.
Yeah.
Okay.
So like you see these two little snails here,
Yeah.
and you see how it looks like to me. Just so you know what I'm looking for. You see how this almost looks like even stripped even more, like down to the skeleton, but there might be like a little sheath overgrowing it. So if you cut here, I can see the sheath, and I can see the strip, and I can get the snails.
Do you recognize the snails?
Nope.
So they might be a new species.
I wouldn't go that far. They might be, but I'm
Well, I'd be excited. You're gonna sell it to me.
I'm not a molluscan expert here, but it's very possible it's a new species. It's always possible.
I think we should name it after Jimbo.
Jimbo Gorgia?
Ooh, Jimbo Gorgia has a nice ring to it.
I'm just gonna let you know that if the spider thing attacks the arm, I'm going to scream
You can do it.
Deep sea arachnophobia
Yeah.
with a robot.
I mean, it is
Just
like.
be glad it's not force feedback. Touch feedback.
It won't attack you if you don't cut its leg off.
Just see what it does when you touch it.
That's right, it was my display of dominance.
Okay, but now leave it alone and see what it starts to eat on afterwards.
That'll be interesting.
Yeah, a little behavior.
Might be too nervous to eat now.
He might have ROV phobia.
It's going back to its original prey.
Actually,
I
can we
think.
just watch this for a minute to see if it goes back to its original prey? Although we have to get the sample too.
Try your there you go.
So gross.
So he almost, because he was
Not
on
gross.
that
It's
little
amazing.
nubbin in the middle there, right?
But it definitely like.
Like he was latched on right here.
He was eating that anemone,
So it
and I
looks like
think
he's
he's
going
coming
back
right
to
back
it.
to it. I agree.
So how does he know where it is?
I don't know. That's why I want to watch his behavior. And then we'll disrupt him again and get the snails. But let's see if he can latch right back on to where he was. Or she.
I think that
No.
the presence of the...
The ROV, I think she was
Yeah.
checking if it was still there.
Yeah, I think he did. All right, you want to just snap this little branch this dead branch while we're at it, and then we'll let him go. We'll move the RV back and let him do what he does.
Oh, it's so funny. He or she keeps like trying to feel if the ROV's there already. It's gonna touch the ROV and it's gonna be like, fuck.
ROV phobia. Chris, I'm not sure who's scared of who here.
Can I just take a bigger piece?
Yeah.
Put in there.
Yeah, you can.
Yeah, go for it. Come in a little further. Hold on. Ready?
Oh my God, amazing.
Alright.
Yeah, you can get that little piece in there too, if that helps. Perfect.
Ready? Go, get.
Amazing. We ask the impossible on the seafloor, and yet it still manages to happen. Thank you very much.
Now she's like, alright, it's leaving.
They'll just swim away.
That's right. We still have one.
Okay, Porch coming up. Yeah. Yeah, that's much better. Thank you.
No, it's still in there. I can see it. Unless it's just a like.
No, it's there.
That's quiver 3.
Yeah. This is gonna be
361.
P361.
Depth is 15.
Yeah, it's in there.
It's just kind of mashed in there. I don't care if it's mashed, but
It's now.
all right.
Don't know what's gonna be in that, see.
That's all right. We'll see. We'll try. Thank you for trying. I really appreciate it. Oh, there it is. It just. That's okay.
Right
See?
there. Hold on. Where is it?
Falling.
It's right there. Refresh. Fell.
That's all right. No worries. Meanwhile, what this really is a distraction to see what the pygnogonid has done while we're not paying attention. let's take one look and see what the pygmy gonid has done while we're not paying attention. It has not left the coral, and it still has its proboscis out, ready to strike. Okay. I think we can move. Thank you very much. That was some great observation and some great behavioral footage, which we really don't get very often, and that was a real treat. Nope. Nice stocked crinoid. What?
Your day will come. They make a musical I want to see that you go to theater.
That's another chrysogorgid, which just does not have a giant sea star on it. A couple more stocked crinoids, more of this plexaurid, more of these whip corals, stichopathes, as we're moving up the slope. We're trying to get to waypoint 5 out of 8. Well, we'll try to get to 8 out of 8, but we're moving up. It's a pretty busy part of the seafloor. There's lots of things to see. And it looks like here we have a baby Iridogorgia amidst the whip corals. Whoops. Yep, that's exactly what we have. Some kind of fish. More of these dead Walteria sponges. Another more stichopathes. There's a Victorgorgia, just to snap zoom. And another plexaurid. Let's see. We've got another sea pen, another one of those tall sea pens, but it's shorter. Urchin. Another tall sea pen. My goodness, it's cool. Really interesting to see them living amidst the rocks, amidst the sand, right? It's like everybody's, instead of having their hanging out, everyone's hanging out together. Look at this, ophoroid covered, brittle star covered, stalked crinoid. And it literally looks like a curlicue forest of what I think are black whip corals. So let's take a look and make sure that these are really Antipitherians. They indeed are all around us, so probably stichopathes. And then we can take a look at this crinoid while we're going, which looks like it has something living inside of it. Maybe an anemone? I don't know. Alright, thank you. Whoa. Maybe just part of the crinoid. Okay, let's keep going. This is great. What cool habitat. We see these large sea pens now sticking in the sand to the right in between these cracks. And a bunch of these. This is definitely the habitat for stichopathes. They're cool.
Yeah, for sure.
And we just saw that one scleractinian. We haven't seen another one since.
I know.
It was big too.
Yeah. I guess, well, we've only gone 30 meters.
I know.
In the up direction.
Which is what I would have expected to see a lot more. And just as I say this, is that what this is in the distance? what do we have here?
I think so.
What is? Yeah. Can we take a look at that, Adam? The white one. I should have asked again for something much. It is.
I know. Oh my gosh.
Why do I
How
not
do you do that?
I don't know.
It's so weird.
I have. It's my whole life that's been a thing. Can we take a look at that coral?
Yeah.
Thank you. Oh, and there's another one up there. See, we got him. And there's a Victor Gorgia right next to it, to the left, with a big giant crinoid on it. All right. I'm gonna need the ISO down, please. Ooh, flying ophroid. Oops, I went the wrong way. All right, little hermit crab. Here we go. Well, there it is. I still think it looks like madrepora from some of these zigzags, but I agree it's very thick. I'm trying to get focus here. All right, that's really great. Thank you. I do not see any associates, but there's another one up ahead, so we can go take a look at this one right up here, please. Which is going to be found right next to Jimbo's narwhal. I'm just saying I asked for hard corals and I got them, so I was just trying to
Wish it into being.
wish it into being, exactly. So here we have Victor Gorgia with a crinoid on it, and we have the Scleractinia, and you can see this is really common, some of these dead branches that have fallen as they get heavy and grow. The branches fall, but I think this still has quite a lot of live tissue on it. It does. And I want to see if we can see any evidence of wound healing again. So if you wouldn't mind.
Coming to the side there.
Yep, perfect. I think I'm going to have a chance here. Let's see. Beautiful. And now, so there's the break.
Yeah, and there's, well, it's hard to say if that's
I
growing
think
on
it's just-
top or.
I don't think I've started yet. Maybe it looks like it's almost like this. Hello, Tim. Look at this.
Want to sit?
I can. I got
Do you have a question about the deep sea or about? Oh, wait. Okay.
No, that was an awesome whole scene of the spider and the.
Oh,
Yeah.
touching the ROV.
That was really fantastic. Well done, just to see how big pod's probably doing most
Right.
of the damage.
So this is the last
That was really
sample.
great.
That was what?
That's the last sample.
Okay, great. I think we're good here, unless you want to collect this, Tim. We already collected the big. Is this
Well,
madrepora?
This is, I think it's a nalp semi.
You do, even though with the
because of
zigzag.
No, the position of the polyps, the way they.
I know.
The way they turn. There's some zig, but it's really not sharp like madrepora.
Mm.
The way they come off. Each
Yeah,
other,
I can
looks
see
like
that.
a nalp semi to me.
All right.
I don't know if that's something that Alexis would.
Well, we collected
You
one
did?
down earlier.
You did? Oh,
Yes, we
you
did.
did. I saw it,
Mhm.
right.
Yeah. Okay,
Good to
a
go.
nice big one, so we could use it for experiments too.
Good to
All right,
go.
let's keep going. Thank you very much.
should be here, right? Instructions.
No, that's maybe, but it's not.
No?
So that's it.
I thought it was at the bottom here. Anyway,
Here, I'm writing it down for you. Can we go check that out up there, please?
anyway, looks like I'm guessing a bamboo, so it's fallen off.
I don't know.
Guessing. Thank you.
You're welcome.
Can do that later.
You got it.
Oh, she took the sea log away.
That's all right. You can just go switch with her. It's all right.
Pretty quick. She was pretty quick.
So you made your PDFs. You're ready to go. All right. Oh, sorry.
Yep.
Right. No,
Okay.
you don't. You don't. You can send them separately.
Okay.
Yeah.
Okay.
So
By the way,
listen, there is some damage on this.
some big amphipods here. We haven't seen a lot of amphipods.
I know.
That's what I'm saying. I think I'm seeing there.
But I don't see the culprit of what's caused all the damage. I just
No.
see the damage.
Sorry, I misspoke. Again, we haven't not. No, those are barnacles.
Oh, they are barnacles. Yep.
I thought I was. It's good focus.
Mm-hmm.
Not sure what that is down there. Is that a
I think it's a
barnacle or is it?
gall. Yep. It's a little
Yeah.
coral gall.
Yeah.
All right. Well, this is
Okay, wait a minute.
really great. Thank you. It's very tall. These bamboo corals are getting huge. What are the depth records for Alchamia and
3 meters tall.
again? We're seeing them at like 1,700 meters. Felt deep to me, but probably not within the range, not out of the range of reason.
Another sea star down there?
Where?
Looks more like Suriaster at the very bottom of the screen.
Oh yeah, good call, Tim. Can we take a look at the star right here, please? That's not the same one of the.
Okay, I don't think so.
I don't think it's a
know.
corallivore. Is it?
Don't know.
I don't know. Let's take a look.
Yeah.
We're gonna see if that star is eating anything. So I don't necessarily want to collect it. I want to pick it up and look at what's underneath it. Yep, that white.
Oh, yeah.
Yeah, right here.
You want me to pick it up and see what's going on with it?
Yep, I just want to pick it up and see what's underneath it. Did you see this? The star eating Chrysogorgia before?
I did. So it's pretty cool. I have to go back and look and see if we have any records of that.
I don't think we do.
remember that.
I was pretty surprised.
Asteroid here?
What?
Nothing.
I don't know.
You alright? Steve. On XC2. Steve? Yeah. Dirty Bilkwood? What's this, sorry? Dirty Bilkwood?
No, I think it is. I think it might be.
Same one.
Hipposterius.
Yeah.
Is he commenting or something, or is he talking to you?
Who?
All right. Yes.
Yes,
Oh
it
my
is.
goodness, you have such a good eye, Tim. I
I
would
think
have
it's-
totally
I
missed
think
that. I
it's-
wouldn't have seen it at all, nor would I have caught that it was the same thing. It looked sort of different because it was
Yeah.
curled up. Good job. So I think the last one's on a sponge. Let's see if this one is too.
Just a few seconds. I got that.
22, 28. This is 1556.
Yeah.
It's another Hipastria.
Goniastrid.
On the rocks.
Yeah. I have to go back and look
So
for
if
a
it's
second.
if they're really eating all these little bits on the floor, then they might be clearing an area of recruits too, or of just small
Maybe
sponges.
on the rock here, you mean?
Mhm.
Yeah. Let's see if I've got a note here.
So somebody's asking a question: isn't this the same starfish as before? And the answer is we think so. And so we're trying to figure out if it's really a spongivore or a corallivore. So I'm not going to collect this. I just want to pull it off of pull it off the rock and see what's underneath it. And then we'll let him go back to his business.
What are you gonna do? Flip them over?
Just flip him over.
You're gonna go flipping stuff. Huh.
sea star somersault?
Small.
What are you on top of? Nothing. Can I zoom in on the area where it was?
Yes, okay.
Just walking around.
Just, oh wait, no, what's that?
We got a little purple deal.
Wait a minute.
He was trying to eat a bug.
No, I don't think it was trying to eat that. It's still very alive. Why can't I focus on it?
I'm zooming out.
I don't see anything underneath it. I think it was just wandering around. Okay, we can let him go about his day.
That's it. So you can should focus see if there's anything else. Yeah.
Oh yeah, we can turn him over.
Got a weird angle. Kind of at a weird angle.
Robot versus sea star. Can we turn the ISO down a little bit? Oops. Yep. Well, his stomach was. Oof.
I don't know.
Yeah, it was out. It was extended. He was trying to eat something. Yep. See what Chris Ma has to say about
I
this.
can also compare to what I have right here.
All right. Thank you very much. That was really helpful. Good imagery. We'll just let him go, and we'll go too.
I think maybe ego plus over. This is it mysterious.
Maybe that is an evil plasoma. Did you get your stuff in?
No, I will.
Okay.
Right
I just
now.
don't.
After this.
No, I just want to make sure you're okay.
Yeah, I'm good.
Okay.
Yeah, everybody's good.
I might have saved a small little purple amphipod or something from immediate doom. Oh, look at this. Oof. What's going on? Are we just hitting the ridge? Or are we? I'm confused
Yeah,
at where
looks
we
like we're
are.
on the top of the ridge here.
Okay.
So just follow it back over straight.
Perfect.
More
Back
corals.
up a second, I thought I saw a Mopacopher.
You saw a what?
Mopacopher. Oh, cool.
Alright.
Well, let's go back. Can we go backwards a little bit?
It's just a little bump we passed.
Little bump.
If you did, I.
Left or right, you think?
No, just right behind us.
Right
Straight
behind-
past.
Right
Straight
behind
ahead.
us, just a literal backup.
Sorry, I was looking at.
I didn't even
stars.
see it. I was looking at
It's a
the
small
or the
shell.
analpsamia in the distance.
It's a small shell on a little outcrop. Yeah, it's in the down camera right now.
I was looking for one of those because I think they might be a great candidate for.
Take a closer zoom in on that head.
On right here.
Yeah. No, I thought it was that little
Up there.
discoloration. It doesn't look
Okay.
like it. Okay.
You sure? Okay.
Yeah, false alarm, but thought I.
Wait, while you're here,
No.
could you settle a little scleractinian debate for us? Thank
Yes.
you, Adam. We're going to move forward. Can you tell us if this up here is analpsamia or madrepora? We've been seeing a lot of it, but it's got it. I agree. It looks thick and analpsamia-like, but it's got a couple little zigzaggies.
It does. It's
So
zigzags.
I just want to make sure I know what I'm doing here.
There's something over there.
I mean, I'll tell you what I would call it, and
We're just going to take a look at any of those three, please.
we can settle the debate by doing a tight on the polyps.
Okay.
If we want to do
Are you
conclusive.
gonna need to bring
Yeah.
your heading arm to the south, or are you okay? up
We
there?
did a collection
I think
too.
that you might have both here from what I've seen just over the past 15 minutes or so.
Whatever works for you. It's not whatever you need to do. Just let us know. We'll do
What's
it.
the depth zone on those collections, on these corals?
Shallower than 1,600 meters, but usually shallower than, more common shallower than 1,500 meters.
So the first
You're
one we
right
saw
at the
was
bottom
right at the
there.
bottom, exactly. The very first one we saw was 1607.
Yeah, that's pretty much the deepest. And they're all about this size. They don't get much bigger until
Right.
you get up into the
Right.
around 1,000.
I'm gonna go to the next one just because this is
Good idea. That sounds great. Do you see these sea pens that keep showing
Yeah.
up in the?
Yeah.
they're all
and
around.
All of the sediment is actually, if we could, before we zoom off of here, get an image of that one on the base of that one to the right, make sure it's in the sediment and not on the rock. That'd be
Sure.
kind of neat.
Yeah.
Because
Can I run it
that's
for you to zoom
Ellipterus.
and move?
Yeah.
Okay.
I think the other ones are on sediment that I saw. But
Oops.
Yeah, that's perfect.
Okay,
So-
let me first take a look at this coral since we're here, Steve, if that's okay.
The fallen material might be the best to zoom on because I can get a
the profile.
profile on the polyp.
Yeah.
I'm working on it.
If I was analyzing the video and I saw this, I would say Analopsemia because of the directionality of the polyps. They all are a little zigzaggy in some respects, but if the Analopsemia usually has the polyps facing all one side of the column.
Right.
Usually
And that's what we've seen consistently.
Yeah.
However, it's that ziggy-zaggy look that's been kind of throwing me a little. I think the sea pen is clearly in sediment. I don't know if you
Yeah.
got a good look at it.
Yeah,
but,
I think the other ones were. That's all too.
Yeah, those pockets could be pretty deep, I guess.
yeah.
Mm.
But you're right, it's very
There's
right up
also
against
some
it.
curvature I feel like I'm seeing here that I don't see on
Curvature,
the report.
yeah.
Yeah.
Of
Thank you.
the base of the polyp.
All right, let's get a look. That's what I've got for you, Steve.
Can you go close on the broken material at the bottom?
Trying. Yep.
So with Analopsemia, you can see on each of the calyces, there's a rostrum
Yep.
where the species names come from, Rostrata. You can see it most clearly on the farthest right one.
Yep, I see it.
Where there's a little kind of point that juts out around the calyx. That's Rostrata.
Okay.
Yeah.
That's really helpful. Thank you. It's amazing to have so much expertise on board. Steve is an expert coral taxonomist, and I learn more from you every day. It's amazing. And then let's see if we can get a look at. I don't think we can get a look at that sea pen from here. I'm trying, but I think the base is just.
Right there.
Yep. Okay. So I think we're ready to go, though.
We sampled some of that at Kingman Reef. We had a forest of them. It must have been like a sandy patch with probably 50 colonies, just like super dense.
Wow.
But why don't we
Sandy
fire
patch no bigger
a
than this.
Niskin somewhere around here, given that it's pretty diverse?
That would be great.
Are you coming on shift on watch?
Yeah, I'm gonna go grab some lunch and then I'll be back up. in a few
Perfect.
minutes.
I am dying to take a shower. There's our little Iridogorgia, I think. Let me take a quick look at it. I'm just gonna snap zoom. Okay, I'm ready to keep flying.
I'm gonna do this.
Oh yeah, the Niskin. I forgot about that. Sorry. No. I was in a. I forgot what I was doing.
What else is around here besides Analopsemia, sea pen?
There was a lot of Stichopathes. We saw Victor Gorgia like just before. Hang on, 223815552.
That was 3.
Niskin number 3, and that would make the sample number
Number
361. Perfect. Thank you.
Number 3.
Yep. Thanks, Tim.
This one fired?
Yeah, it's fired.
Bam. Bam.
You have to hit water.
Oh, duh. Oh, bam.
Yeah, it's done.
Didn't type in the depth. Damn it. Okay.
Go buttons,
Magnus-
apply
little
buttons,
Magnus-
apply buttons.
Spiralis.
Yep. There's been a lot of corallivory. You noticed? It's been
Has been.
interesting.
What's amazing is those Pentagonus seem to prefer those anemones,
I know.
right? And it's almost identical to the one we saw earlier.
There's been a lot of these dead sponges too, covered in oats. It's
Yeah.
very typical.
Yeah.
A bunch of crinoids you could list. Soft crinoids.
Small battlebrush chrysogorgids with your chirostylid crabs inside. Do you want to snap zoom on the crab, or are you good? They always have a
I'm
crab.
good. Yeah, I know.
Okay.
I know what it is.
Actually, that one, yeah, it does have a crab.
There's a red thing there.
Okay. Brazingids. It's a very diverse area. Here we have another Iridogorgia magnispiralis with a crinoid on it. Here, going up the slope.
Here's your ROV. It's been fun for you.
Can we take a look at this? Half,
pretty
looks
cool.
like half damaged, half
Yeah, it is.
more sea pens, more stocked crinoids. Sorry, we're tired. Not trying to yawn.
What's going on over there? Some damn little sea star.
Yeah, I'm yawning again.
Oh my goodness, Randy.
I'm so tired.
You're putting me to sleep.
I don't want to put you to sleep. I just want to put myself to sleep.
It's like Ecology with.
Is it? I wasn't sure if it was just
I
a
don't know.
Plexaurid or an
It may be
Acanthogorgia. I'm having a
Plexaurid.
tough time.
You gotta zoom and look for the spikes.
I know, but it's really hard to look for the spikes when the polyps are open. It's easier when the polyps
No,
are
I
closed.
know. You just have to.
There's an
Okay.
Aplacophorine on the bottom.
Yeah. All right. So we got a coral here.
I don't think those snails are eating the
Let's
coral
make- let's make
polyps.
sure.
I think they might be eating the sheath on the base. Hang on, let me zoom here. I mean focus. I mean, you know what I mean.
You gotta go left for closer. That's closer to you.
Now I'm trying to go further away, so I'm
You're
going.
going. You're going.
I'm failing.
Come on back. No, you're not failing. You're winning. You're winning. Keep going. You're winning.
All right, I think that's good. Let's see if we can. Oh no.
Here. Can Matt just rail on it?
Oh,
Okay,
you
so
got
that's
it.
that.
Okay, you see the little hydroids.
The hydroids.
So you see why I think the snail's doing that? Look, I think the snail's keeping all of this sheath clean.
Yeah. So what coral is that?
That bridge is we're just seeing the shadow down here.
I don't know. See, can you see the spikes when the polyps are open? I sure can't. Oh wait, here's some closed polyps in the top. Let's see if we can tell. I do not see any spikes. So I think this is a plexaurid.
That's what I have.
Okay.
Cool, thanks.
All right, that was great. Thank you very much. But did you note the Aplacophorine on the bottom?
I did.
Thank you.
Yes, I did. I didn't hit corallivory though.
I don't know if it was.
I know.
I didn't see it.
Oh my God.
I'm so tired, Tim.
There's no oxygen in the room.
Ah.
Yawning. Come on now.
I got a little bit of sleep, and now I have like 2 weeks to catch up on. It's not going to happen. It's straight on magnispiralis on the right. Looks like a giant
I think so.
dandelion.
Bamboo stalk.
Bamboo stock. What's this purple thing? Nope, there's a star for you. Slime star?
We saw one earlier.
This is my
Yeah,
first.
sort of like that.
But yeah, slime star.
Little sponges here. What are the white things on the right?
There's
The gray
tons
things?
of little sponges. There's been a ton of them,
Encrusted
and all
sponges.
different sizes, from like itsy bitsy to like these bigger like. Sorry guys, I can see in the chat that everyone's feeling my fatigue.
They need some coffee beans.
I know.
Are those sponges, you think?
Yeah, they are. I zoomed in on earlier. I'll do it again.
Okay, no.
No,
You got
look,
'em.
look.
I'm looking. What's that?
That was.
Goodness.
I have a communications check-in meeting in 15 minutes, so I will have to leave you on the seafloor.
Where are sponges?
I don't know. They are all sponges. These massive amounts of little baby sponges. Or maybe they're full grown, but never get big sponges. But there's massive amounts of them regardless. I think there's a mix. See, this is like the max size of them, and then there's a bunch of little itsy bitsy baby ones.
Yeah.
A lot of Analexamia. It is amazing. Just look around. I keep.
Still seeing stickopathes.
Coral. This just looks like a really productive area. Just a lot growing. This is not barren at all. Oh, here we go. The landscape shot behind.
Massive.
Climb to the top of the rock and you get the view.
Rock climber, go over the top of it.
What?
I don't know what you were saying. You're
You
climbing
climb
rocks?
the rock and now you get the view. You get the vista.
Ah, the vista. This is the vista rock. Mm-hmm.
Just like hiking.
Mm-hmm.
Oh. Hannah, no, I'm all good.
So
I'm sorry.
We're at 15. Ha. 1537 meters depth. And
Yep.
what's our final waypoint target? 12
We
something,
have
right?
8 waypoints, and we are most of the way to waypoint. Well, I don't know where we are in relation to waypoint 5. Could you show me, please?
Go ahead, Rich.
Hey, are you waiting on me?
Nope. No, you're good. Yeah, just got a waypoint there.
Okay, just checking it. And now it's slowed down.
I know, it's just...
Sideways is a bit slower for me than put up ahead, but just let me know.
Perfect. Thanks.
Could you show me where waypoint 5 is on the screen, please? What?
My relief is coming soon.
Oh yeah, same here.
Oh, I see where it is. Okay.
That's why they call it relief.
Oh, let's talk about that. Great. I think we're on track for.
Crinoids
and that's the last one,
with
or-
lots of small white ophiuroids on them. Common. See that? Another Analexamia. Haven't really seen associates on the Analexamia, the hard corals, but still seeing stickopathes, black coral.
No, I think we want to try to be
Almost
on
throughout
deck
the
by
dive now.
30 so that we have enough time to process our samples so that we have enough time to do the thing. So 3:30 would be like the latest. 3 would be it. So I'm trying to get
Sure,
us.
so off bottom 230? Because I think by the time we get to the top there, you probably get about 1,000. That's an hour of search.
I think off deck by 2 would be. Okay. I would love to be off bottom by 1:30. But I don't want to. But I want to finish our tracks. This is exciting. So I'm balancing. All right, so we're making some tracks because we think we have captured and characterized what's in the area. I'm not seeing too much stuff that's brand new, but we're looking for new corals we haven't seen before, new associates we haven't seen before, any evidence of corallivory or spongivory, and we've collected some good rocks. We've collected some great water. We've collected some good lots of things, and now we're just looking for new records, but I feel like the characterization of the seafloor here is mostly under control. So if you are just joining us, you are joining us on a very cool seamount in the U.S. exclusive economic zone. We are far west within the U.S. EEZ in equatorial waters in the Pacific. We are on a seamount that's unnamed, and that as of last night was unmapped. And so we mapped it last night. We're diving it for the first time ever. We're the first people to see this piece of seafloor, and you're here with us, so you are too. With about a 60-second delay at most, so you're really 1 minute away from being the first people to see this on the seafloor. Which is pretty cool, and nobody's ever been here before. And we dove the sister seamount to this within the past 24 hours. I don't even know.
It's hard to know.
I'm so confused. And we have been exploring over here for multiple reasons. One, the unknown is always exciting, so unmapped, unexplored checks those boxes. Number two, this is a really nice latitudinal position for us. It is roughly the same latitude as Howland Island, where we were a few days ago. And so this, but it's off of the Tokelau Ridge, so it gives us a chance to look at a seamount that's probably in the same east-west current flow, but off of the mountain chain. So we get a chance to see what things look like in this part of at this longitude, at this latitude. And it also gives us an opportunity to work in depths without an island on top of us. So we were working with Helen and Baker, and now we're kind of comparing the same latitude with and without these sort of surface features to look and see what the deep sea looks like. So we're working at the same depths we were working at on Howland Island in order to make that biogeographical comparison. We are really honored and lucky to be working with the U.S. Fish and Wildlife Service and NOAA in the Pacific Remote Islands Marine National Monument, which are to the east of us, still inside the U.S. EEZ. We will be back there again tomorrow, roughly 24 hours from now. I don't exactly know, but we'll get, because I don't know what time we'll leave the seafloor, but ish, to go to Tie Top Seamount, which is also within the monument. And then we'll be working our way down the Tokelau chain to the edge of the U.S. border, where we'll be then headed to some more ABNJ sites, or areas beyond national jurisdiction in the high seas, which are not owned by any country and are really interesting places to explore. So more unnamed, unmapped, unseen seafloor. All these plexurids are setting up here. We still see some bases, but in the time it took me to give that little spiel, our landscape has really changed from sort of bouldery, rocky outcrops with pitted sand in the middle and a very sort of steady stream of corals to now a much flatter, more
pavement or
pavementy area with a bit more of a sediment drape, and we've changed our coral community to almost exclusively these plexurids. So interesting, isn't it? How you can turn a corner and everything's different.
Yeah.
I grew up in near New York City. It's the same thing. You turn a corner and everything's different.
Right.
It's true. One neighborhood to the next.
Yeah.
So maybe we'll call this, I don't know, Manhattan? Nah.
Yeah, lots of ophiuroids and all these plexarids. A little more sediment here on top of the pavement
Yeah,
than we've seen.
for sure.
Depth is 1523.
Well, I'm going to have to go in about a minute, so I'm hoping Steve or somebody comes in, maybe Anna. We've got another one of these stichopathes, and we've got some sponges with, dead Walteria. Yeah, we seem to be in dead sponge territory again. There's a lot of them right ahead. So my name is Rady Rojan. I am faculty at Boston University and the chief scientist of this expedition, and I'm joined by my co-lead, Tim Schenk from the Woods Hole Oceanographic Institution. And we are here in the room with some awesome ROV pilots. We've got Adam and Chris flying today. You guys say hi?
Hello.
They say hello. And we have Debs and James, our two awesome marine technicians in the room. There are 39 people on this ship, but it takes all 39 of us to make this happen. everybody from the captain to the chefs to the deck crew. I mean, everybody, the whole thing, and tons of people at home. I'm watching Hannah was in here at least for a little bit earlier, but she's one of the communications experts at Schmidt, and there's so many people on the ground. So 39 people on ship, and I don't know how many off, who are helping to make this expedition happen, to help us get to these really far-flung places. I mean, we are really in the middle of nowhere, in these equatorial waters, and to make sure that we can see these new spots and stream them live to you on the seafloor, which is amazing. I never get sick of thinking about that, right? Like, it's just incredible to think about the fact that this is even possible. So thank you very much to the Schmidt Ocean Institute and to the people who have helped us to get here, like Conservation International,
Yep.
NOAA Deep Sea Coral Research and Technology Program,
Yep.
A private foundation who has very generously helped us out in a time of COVID, to our friends and colleagues in Kiribati who are battling COVID right now, and so we're staying in U.S. waters in order to respect their closure boundaries due to COVID. What a mess this pandemic has been for everybody.
Truly.
But we are wishing our dear friends and colleagues in Kiribati a serious Maori hello from across the equator. Literally, we'll look across the line and wave. We miss you. We're very lucky to have a Kiribati national on board with us, so we're thinking about you all the time. We have Anamere Tanaba. And we are working in the U.S. Phoenix Islands, which is really nice to have this archipelago split across 2 countries and across 2 different marine protected areas, which really showcases this really important biogeographic space in the ocean. Right. I
Very
am
good.
going to see if anyone can come up.
It's Anna Murray and Steve, yes.
I think so.
Yeah. 1,511 meters. Getting there.
Yeah, we are getting there.
So you're thinking 130?
Wait, someone's coming in. Perfect. I was about to slap him. I'm thinking. Okay, thanks. I'm turning you over to Tim and Steve. We'll take it from here, and I'll see you later. Hannah, give me 2 minutes.
Alrighty, thanks.
I'm trying to poke around for one more rock before we leave.
You going this way?
Oh, no, we do have a few rocks actually.
We do have, yeah.
Oh, okay. There's one.
Some really
One pretty
cool, nice
recently.
ones, yeah.
Yeah, maybe we'll wait a little bit.
Here, you want to?
No, I've got it. Perfect. Plenty of space here.
Those are big dead sponges. We've been seeing them quite a bit. Haven't seen very many live ones though from that in this type of habitat.
Oh, I think I have it, Steve.
Oh, you got it?
There's so much.
Some little sponge.
Got some corals there. Some black corals.
Like
Black corals called stichopathes, as well as mostly yellow plexaurids. Let's see. Some of the sandy patches have Polypterus sea-pens in them, but not many here. Yeah, I'll work on it.
I thought I had them, but they're all shape files. It'll take forever. Okay. Anyway, we can sit down today. Just and then
You looking for anything in particular?
No, we're just kind of cruising. It's been pretty similar and consistent. I think we just want to move through this, but maintaining a consistent altitude. Lasers and just getting some quantitative data.
Is this too low? You want to get a little bit higher?
Yeah, just like a quarter meter, or something even less. It's really good the way it is. Let me make sure we're full out, and I'm gonna tilt up a little bit to get some distance. Wait, can we actually zoom in on that yellow fan on the upper right? There's a bunch of things around it and on it. See if there's any predation. So we've got a pretty ragged looking plexaurid fan here. Mostly with hydroids. Hydroids, several Asteroschema brittle stars, and then this curious hermit crab. So the hydroids are where the hermit crab is right now. They're these thin, wispy cnidarians. They are experts at colonizing pretty much any available substrate where they don't have to compete too hard. And then they'll usually gradually take over a coral colony since they reproduce faster and are excellent competitors for space. But I think this one's just hanging out. I don't see any evidence of grazing right now. However, a couple of the sea stars looks like they're missing arms or might have missing appendages. But looks like we've got one Ophiacanthid plus 3 Asteroschema
Is that a bamboo
and a
coral?
crinoid. It's a plexaurid coral. So that's this category. Yeah. All right, coming out and keep going. Yeah, good. Thanks.
So anyway, and the next dive into the spreadsheet is eliminate.
Turn on one of them, just a camera. Knock it out. I turned that camera.
So we. And then I haven't, I'm assuming it's gonna be 9,600. That's still. And then those two things will start. 12-hour drive. This RV drive is actually.
Is the camera roughly centered? Is there a way to check that without having to hit a button? Roughly to the left of it. Yeah. Okay. Yeah, I'm gonna go like that. It'd be cool to have a center button.
Yeah, I've been asking for that one for a while. Actually, and also like some of the RVs I've been on.
Yeah.
I've
Yeah.
You can set it to like so.
Yeah. Yeah, I like that. Transect mode, full wide, center, lasers.
Time-wise, so it's really, people are putting them in. Just right here. Ship visual.
Yep, go ahead. You can see that in this type of terrain, anytime where you have this complexity, where you have changes in terrain, you get different communities. So you'll start to get more bamboo corals, and in the ruts you'll see where there's sediment, the sea pens.
This is a very tall coral.
Yeah, these bamboo whips can grow extraordinarily tall, sometimes several meters, and they're usually unbranched, entirely unbranched. They just keep growing and growing and growing, and yeah, it's not. I'm not clear, what might stop them. I guess probably the weight of the colony on the base that they're on, the rock that they're on, might result in them tipping over eventually, but. They're not that heavy in reality. They're pretty light, and when you're in the water and you have a little bit of buoyancy, that probably is the reason why we see so many huge coral colonies. Can you go back to the right? I'm sorry, just out of frame. I was hesitated a little bit too long. There's a red something kind of hanging about half a meter up off the bottom on a very, very fine something. See that right there? It's a dandelion. These are cool.
Wait, is that a jellyfish?
It is a type of jellyfish. It's not a medusa. It's not a medusa. It's an animal called a dandelion siphonophore. So this is related to those siphonophores we find up in the midwater. I'll go in a little bit more, and this one in particular is anchored to the bottom. It has these long lures that used to anchor to the bottom, and also it uses them to feed. If you actually watch, say there was movement of particles on these lures, sometimes you see like particles moving up and down from the action of mucus. This one is a.
And they can suck those things in to change their altitude off the bottom.
Exactly, and it's pretty fast. Yeah.
Pretty awesome. You want a little bit darker?
No, it's all right. I mean, we got some good imagery way earlier in the cruise, but I just wanted to get a close look for. Yeah, this one's not exactly the most photogenic specimen. It looks like it's been through the wash a few times.
Nice color.
What's it called?
The current's kind of pretty strong up over this.
Yeah, it's called the dandelion siphonophore.
See the current here? Is that
Yeah.
coming up?
Up and over. Yeah. So what's the directionality on it? It's north
So that'd be
to
going from north to south.
north to
And
south.
also going up.
Up and over current. Yeah. All right. All right. I'm gonna pull out and keep going. So yeah, it's like a jellyfish in that it stings things that swim past it, but it has specialized zooids, which some parts of the animal are just made for reproduction, some parts are just made for eating and digesting, some parts are for movement only. So they are made up of all these different types of animals.
Do you think he ate the snow, the marine snow as well? Like,
Marine snow, or even bigger things. Sometimes in these siphonophores you see big things. Can we take a rock here? This looks like a good spot. I figure we have better pickings here since it looks a little... we might need to...
In this rubble area?
Yeah, we might need to check first the box. Can I...
I can see it right here. What do you want to see?
You have plenty of space for...
Yeah.
I was just seeing I had 4 rock collections, so I want to make sure I have more.
Yeah, there's plenty of space in this.
Yeah, let's do a rock. 10-15 centimeters. Kind of the most lumpy looking rock. Not particularly angular, and maybe something on top of a pile than below it. I'm sure most of these are probably loose, since they seem to have just collapsed here. But I've been wrong before about rocks. I'll stick to corals. Yeah. Another sea spider. This one, I don't know if it's preying on anything. I think it's moving, actually. Very slowly. Yeah, really interesting. No grazing for this one, but yeah, we've seen them eating all sorts of things. Sea cucumbers was a new one for this cruise that I've never seen before. Yeah, anemones, yes. Corals, yes, but never sea cucumbers. Alright, I'm gonna go poke some rocks. Yeah, these are the right type of shape. The question is, are we going to be able to move them? Before we pick it up, we're going to want to image it in place, but tapping it. Yeah.
Are you looking for a solid rock, or you just look for...
Yeah. I'm not going to image it first until I don't want to take thousands of images of things that don't move. So let's give it a tap,
It's part of the scanning.
and then I'll image it. You can move it a little bit. You don't have to be too careful. Still just a rock.
It must have been just after I left because I didn't see it. I was looking at the messages.
Might be able to do the normal light. Come back here and sit.
You're looking at pretty pictures.
Well, it's okay. That may be a slight stretch.
Oh, perfect. That is great. Yeah, let's image that. That's a great break. Let me take a picture of it there first, so I can get where it broke off. All right, so we're gonna do some sample in situ shots. Give it a second for the sediment to clear. Yeah, I don't know. Is that
twice now?
Yeah, that's a strong action.
900 foot-pounds.
Yeah.
Crushing force. Yeah, it's just gonna take a while. Your manual focus?
Yep. All right. While the sediment clears, I'm gonna go do a quick detour here.
Is that a coral?
I think so. That's what I was trying to check out while the sediment cleared.
You want me to get you there real quick?
Yeah, yeah, that'd be great. Yeah.
Now that you're back.
There you go. It's a cup coral there.
It's a small little coral.
Very, very tiny. Yeah, that's really neat. Very tough to tell these to even species or sometimes genus cup corals because of the tissue. You really need to get at the skeleton. All right, let's do a in situ. So this is going to be sample number.
You want me to eat it now?
Yep, hit it. All right, and then we can go in with the claw and we'll image it in the claw.
Yeah, so there's two pieces in there. I'm pretty positive there are two.
Yep.
There's a smaller piece that's closer to us, and a bigger piece that's away from us.
Yeah.
If you look in this camera right here.
Yeah. Yes. Yeah, I got it.
Do you have a preference?
The back one.
Back one.
Yeah. The bigger. Yeah.
One of the lasers on?
Yes.
You ready?
Ready.
FK21
Dash 362.
360 rocks. Rock box.
It's going to be in the rock box. Yeah.
Any additional tips?
Let's wait to see what it looks like, and then we'll take a peek. Can you hit in situ? I'll do it. I'll do it. You can.
Yeah, I
Yeah,
can
just
do it.
hold that screen because you already typed in all the information. Okay, go ahead.
FK21.
That is a fine rock. I like that one a lot. You need something smaller?
No, we just need to use that arm.
Cool.
We'll just go back up just a little bit more.
Don't want to make it too easy for y'all.
On top of that one, too. I'm trying to keep doing that almost a little bit here, just not trying to close all the legs. Get a little bit of a. Yeah. You're all to your left. Yeah, it's just pushing. Try to push it. There you go.
Real firmly.
Huh. I wouldn't have suspected.
What do you want to do about that?
Can you take the chunk on the right, maybe? I mean, it's still a good rock. It just, you saved us the trouble of having to cut it with the saw. That is one rock. If it breaks apart again, we'll abandon.
And then we can get a smaller piece that's in there.
Yeah.
I think we just.
You can really see the thickness of the crust on some of these rocks. I still like it. Alright, I don't like it. In fact, I really dislike this rock. I don't want it anymore. No, I really want it.
Do you want me to add a comment on the?
Yeah, once it's in the basket, we'll do something like that. It's gonna make a cool photo though to look at the.
Alright, once he gets it over the basket, I'll get some closer
Yeah.
images of it.
Sure. Yeah.
Because it's-
If it's that precarious, just throw it in the basket.
I would not almost maybe more because it's fairly well. Don't go all the way to full shadow.
So you can write in the description, rock, broken rock, about 15 centimeters, roundish.
Here.
Yeah, that's enough of a description to be able to identify it later on.
362, yeah.
362. Yeah. Let's just go in the box. We'll limit it on deck. I don't want to break any more off of it.
Once he's over the, once he's over the basket, then I'll be able to.
All right.
Come up a little bit there. You want to come up more? I'll stop.
Yeah, maybe just on the yeah, over the basket and on the face like that. Yeah.
You got the camera?
Yep, I got it. And just turn it around to the other side. All right, good to go. Cool. All right. Yeah, we can submit that.
Oh, that was really easy.
362 cracked rock. Good to go.
A bit of a downhill, and it looks like it's 2 miles. Well, looks like the ridge is going to come up.
Can kind of zoom over the little depression here. I don't want to get bad video going downhill. Keep it at a higher altitude and just cruise over.
We've barely seen anything in this area. Oh, there's a star right there.
Yep. There's a couple of sea stars, ophiuroids, black coral whips, synaphobranchid eels, or cutthroat eels they're called. There have been some halosars as well. But this is, yeah, definitely looking very ridgy terrain. On either side of us, the seafloor drops off to much deeper depths. But up here, you know, there's actually a pretty appreciable flow. We were indicating flow directionality and strength using the dandelion siphonophore a few minutes ago, where there was very clearly a topographically induced flow where water meets the bottom. It's deflected up and accelerated up and over the top of the
fish.
Yeah, it's like a synaphobranchid.
Basically, most of the black coral right there.
Yeah,
Shows.
yeah, it's black coral habitat for sure. I think they probably do very well in the flow.
Same thing on this downhill?
Yeah, yeah. Let's move over it. little bit more diversity here. They've got some more plexauroids, black coral, iridogorgia, a couple different black corals, and well, small Walteria sponges. Is that a big fan over on the upper right? Or is that a shadow or something? Rock? Yeah. Can we take a closer look at that? This is interesting. This is a black coral, but it's one that I don't think we've seen yet.
Is it a different kind of black coral, the one that we usually see?
Yeah. So these whip stringy things, those are black corals as well, and then there's a bunch of heteropathes.
Yeah.
And this one I'm not familiar with, actually. I'm gonna do some imaging first. Thanks.
Got your menu?
Yeah, got it.
Want me to eat the button?
Let me take a closer look first. So it looks like that looks like a squat lobster in there, I think. Well, what is that? I don't know. It looks gelatinous, actually. Maybe it's anemone. Huh. It kind of looks like a part of a dandelion siphonophore just blew into the colony.
Oh, there's another one right
Let's take
there.
a look at this one.
It's a crab?
I don't know. So this is a really good close-up shot. You can see the. So I think these are dandelion siphonophores, or maybe some. I have no idea, honestly. I can't make it out. But so these are all tentacles, right, of the black coral.
Yeah.
The mouths are actually these small, darker circles. You see,
Mm-hmm.
1, 2, 3, and usually for every 3 pairs of tentacles or 6 tentacles, you have a mouth, and that would make them hexacorals, which means that they're more closely related to stony corals than to other sea fans. I'm going to take one more look, and I'm probably going to set up to do a sample here. Probably in the upper left corner, since there's something odd there. If we see something else on this side, I'll try and go for this side. Alright, can you take in situ? Alright, and then I'm going to go back to the other side. I don't see any crabby things in here, do you?
What's that, right?
Yeah.
To your left, just a little bit. Yeah.
The translucent thing?
Is that an egg?
It's like a
You think so?
Because I guess like I saw some on the right side.
Yeah. I don't know enough about deep sea egg cases, but it could be. I haven't seen anything like that, but it doesn't mean it's not.
What about that white thing right there?
Where are you looking? Yeah, that's what I was looking at first, right? Because it is so deep in the colony, and I thought this looks sufficiently similar to this, so maybe we can just do a cut here or something, or closer. So let me do a quick image on the upper left branch, and I would like to probably get a segment that includes this, whatever this is, nodule or something, ulcer, gull, I don't know, possibly associate. Alright, do another in situ. Alright, and we'll do a specimen shot here. With lasers on the colony. Alright, let me put it at the base. Alright, do a specimen shot.
Which one?
One more in situ. Alright, we're good to go. I think maybe somewhere like here. Is that okay?
Yep.
Yeah. This is going to be
6'3".
6'3". I don't know. I don't know what this is, honestly. Identification-wise, it shares a lot of characteristics with Stauropathes, but I don't think it's that. Is that thing on there still? Yeah.
There's two of them.
Cool. I'll just take a look at the backside. Yeah, so the backside of this looks like... So the thing on the right is a polychaete worm. So this is a worm. You can see the legs there.
Yeah.
But I still don't know what that is, so we'll have to take a look at it up on deck.
It's a really long thing, long skinny hair.
Yeah.
Oh, the worms is moving.
Yeah, the worms move. Let's stow it in the meanwhile. Let's see. BioBox, we have C open. I think...
Are we going BioBox?
Yeah, let's go BioBox since it's a bigger piece and I don't want to lose the associates on it. Let's go BioBox.
You said C, right?
Yeah.
Biodiversity. Any additional tags?
Red black coral with worm and mysterious orange blob.
Mysterious
Possibly, yeah.
orange
Blob. B-L-O-B.
blob.
3, 6, 3. Good, still it.
Submit.
Yep. Great, thanks for the collection. Yeah, Jeremy, that's what I was thinking, Stauropathes. It's one of the larger colonies I've seen for Stauropathes, but it's got some interesting associates with it, so I will get back to you on what some of those things are. There's definitely a polynoid or some sort of polychaete worm. We'll take a look. Our target depth for the end of the dive today was 1,200 meters, and we have about maybe an hour, a little bit more than an hour left before we're starting to end the dive, or talk about ending the dive. But we've started to see some pretty, just in the last few seconds, some pretty amazing coral diversity that we haven't seen a little bit deeper. We got Analepsemia, Victorgorgia, Iridogorgia, stony corals, Analepsemia mostly, but some really nice. Oh yeah, can we image those crabs? crabs and shrimp. There's 3 things in there. 4 things. If you want to fly steady, I'll try and be quick.
Yeah, just zoom in.
Yep.
And then I'll pan you around.
Sure.
Tell me where you want me to go.
2 crabs in front first, and then we'll kind of duck down under a little bit and get the shrimp on the bottom. It's good. I'm not gonna push too much further in. It looks good. I don't want to disturb them. Okay, let's duck down. There's a shrimp, kind of at 6 o'clock on the colony. Yeah, there it is.
I got the pan too.
Okay.
This is a beautiful column.
It's definitely one of the charismatic ones. This species and genus holds the record for the longest coral. Clearly, it could get quite large, and I think the world record was something like 3 meters, maybe larger, off of Hawaii. Although I think some of the ones we've seen in recent years, especially out in the Pacific remote islands, Monument, have rivaled it. Can I push in?
Just a touch.
Okay. If that's all we're gonna get, that's fine. I just wanted to try and get the best image possible.
Come around that branch.
Yeah. Yeah, that's okay. I think that's good enough. I didn't see all the parts of the shrimp I wanted, but it's a tough spot. Can we get kind of lasers on this? Lasers on it, kind of looking off into the distance on a perpendicular to it. I just want to get a perspective of how big this is. I'm going to try and do some fine tuning and try and get the lasers on the stem.
I'll get them for you.
Okay. And then can you hit the beauty shot button when the lasers are on the coral?
Sure.
Like you want them on the stem?
Yeah, I would like right on the stem as close as.
Or in the middle, like right
in the middle. That's in the middle.
I'm gonna get it as full frame as I can.
Yeah, that's great.
No, you want me to?
Yeah,
do it now?
just when the lasers are on the middle of the coral.
Okay.
Because that helps us do a quick size. It has some of the largest branches that I've ever seen on Iridogorgia. Trying to get some beauty shots now. There, I think I got one.
Stupid curly.
That's alright. And then kind of scan over to the lasers on the base. This one doesn't have to be so precise, just kind of swing by, hold it for a second.
It's a rocky, stony coral.
Yeah, over on the left-hand side, that's right. Alright. Good, that's good enough. Yeah, so 15 centimeters. Tissue probably covering about 10 centimeters. Nice little pirouette, and we'll escape. All right. It's a nice little topographic high point there. Bunch of really nice crusty crust rocks. Very loamy. Typical fauna for this depth. Definitely these stony corals are very prominent. Victorgorgia usually co-occurs. Chrysogorgia, we've got some nice specimens. Bamboo corals. These branching.
It's a pretty nice little feature if y'all wanted to let them look at.
Oh yeah, that's a good point. Well, I'm gonna see if I can page Brian to see if he wants to do a photo mosaic.
I'm just spin around it one more time just
Yep.
to verify.
Yep. Looks like there's a Bathypathes black coral too.
The red one?
Yeah, your name.
It depends on what the interest is. Like the tall Vittigorgia and the whips aren't going to show up. The hemicorallians probably will. So I'm on the fence. I think if there's a reason you want it, I'm game. But I don't think it's not a perfect target either.
Yeah.
And given how we've still got a couple hundred meters we wanted to get before the end, so I think I would say we should cover tracks rather than spend the time doing the mosaic.
Yep. I agree.
Okay.
Thanks. So, James, can you, waypoint 6 is our last one, or is there another one after that? Or, how?
Got
Two more?
1.6 and then 1.6.
Can you throw up that map?
Throw it up the
Yeah.
outside, yeah. About 580 meters from 1.6.
Yeah. Yeah, let's cruise a little bit, because we're not seeing a lot of new stuff other than that black coral. Try to cover 100 or so meters in the next 15-20 minutes, maybe half an hour.
You do that.
Sure.
Updated waypoint bridge.
Yeah, so we got a bit more slope. It seems to be picking up too in steepness from what I can see on the bathy. So this will be a nice comparison. So we're looking at similar depths now, but slightly steeper. I've seen a couple of species of Bathypathes now. This is a black coral Bathypathes. There seems to be two color morphs that I really can't tell apart, although we have specimens of both of them from across multiple cruises. There's a orange, pale orange colony, as well as the red. There's a red, more highly branched Bathypathes colony, which we just saw on the rock past. It's been a pretty productive dive for collections. Pretty satisfied. A lot of new stuff. Waiting to see if this red sea pen is in the quiver. But we were. Yeah, there's a red sea pen. We tried to push core it and then dump it in the quiver because it's a sea pen that likes to escape by burrowing into the mud. And second time it's done that to me. I consider it personal now. The next one I get, I won't be fooled. A couple of ophiuroids on a barren stalk.
I gotta slow down
Oh,
just a
sea
little bit for
cucumber.
that.
Sure. Well, if we have to slow down, can we look at the sponge with a bunch of stuff on it?
The first one?
The first one, yeah.
Is that a crinoid on it?
That's what it looks like.
Ooh, crinoid and orchid sea star.
So we're just doing a quick pit stop to kill some time here. Take a look at the associates on this sponge. A lot of ophiocanthids. They do this thing where they sense this pressure wave coming from the vehicle of the water, and they abandon ship. Not sure why. I don't know. Some sort of evolutionary adaptation, maybe, to make sure they don't fall too far downslope. Can do a beauty shot here, I guess. Can we do a lasers off? Great. Can you hit the beauty shot button? Just gonna frame it up.
Beauty shot. There.
Nice. All right.
A little red, huh?
Yeah. Sponge.
You want to do a white balance real quick?
Yeah, let's do a white balance while we have time.
That sponge should be
It
white.
it looks okay. It's not. Yeah. There we go. Let me do the same shot one more time for comparison. Can you hit the beauty shot button one more time?
Yep.
Thank you. Okay, coming out. Ready.
Good to go?
Good to go.
That's not a big.
No, nah. I've seen a few of those. You mean the coral? Or
Yeah,
the-
that's
this one?
ballistic going out there.
Yeah, we've seen a few of those. I'm not seeing any associated animals. Well, let me zoom from afar, quick, just make sure I don't regret this decision. So we've got crinoid with a stereoschema, maybe two ophiuroids on that. But this is a morphospecies we have good samples for, so I will pass on further exam.
What is this black thing? Oh,
That's it?
No. It's a
Oh.
maybe just a
It's there's some darker colored feather stars around here. They're usually dark purple or sometimes black. It's another one of those sea pens we collected earlier too. Not the challenging one, the easier one. There's another one of those black corals we just sampled. possibly stauropathes. So I heard we have an iris control button down here, or an ISO button. Is it this top one?
I don't think it works over there.
Not working.
They might, you could try it.
Looks a little hot, and it's only because it's just that we're going between sediment and rock.
Is that your C-pin right there?
Not that one. That's a different one. We have some of that from a different cruise. The C-pin I'm looking for is about 10 centimeters tall, thin like a pencil, and red. Yeah, this is.
Better for you like
yeah,
that?
yeah, a little bit better. Yeah. Some nice diversity on this rock, though.
Really nice.
Yeah, you got diversity of echinoderms mixing with corals.
Pretty neat.
Some pretty big colonies too.
Was that a Parentopathes before? I was just next door. It's hard to see.
No, that was
over here somewhere.
yeah, in the sand, in the sediment. No,
I didn't get a good look.
I didn't.
Sea stars too up there.
Oh yeah, we're gonna want to look at those sea stars on that sponge.
So cute.
I didn't see Parentopathes, but I'd be curious if it was.
Yeah.
So this is so we should hit the spongivory button. So the ivory button, which is the first button on the left, and spongivory there. Have we seen any spongivory recently?
No.
I think this is one of the rare occurrences.
It's a jumper.
Oh,
Yeah.
there's 3 of them.
Yeah, this sponge is getting
Hammered.
yeah, for lack of a better word.
Yeah.
Maybe this is what's causing some of the tissue loss. Oh, I think I'm about
What's
to
up
get
with this?
I'm about to get booted out of the seat, I think.
No. No booting out. I was about to call my kids, but I can't. Hi, Maddie. I couldn't resist this one.
So how would you want this?
I don't know.
Yeah.
I don't even know what I'm thinking.
You want to do some imaging first?
I do.
Okay. Because these look like the sea star you collected,
They are exactly the same sea star
Yeah.
we collected.
and now you have evidence of one on a coral and one off a coral, or a sponge.
And one on and one off a coral and one on a sponge.
Actually, that looks like a different sea star in the background. You see,
It
much
does.
more finer.
Good eyes, Steve.
It's,
What?
I'm not sure how much you enjoy eating a sponge.
Do they move and are they collectively getting together and moving en masse to kill a sponge all at the same time?
Wow.
Hi, J-Red.
I haven't seen that.
I mean, it definitely looks like they're eating these spicules that
Yeah.
you know.
That are growing all that.
Yeah, maybe.
Yeah.
Please tell me you hit the spongivory button.
Yeah.
Yeah.
Okay.
So maybe not so much eating the sponge, but what's on the sponge.
No, I think it's eating this. Look, see if you go down the bottom, yeah, or around it, the little side spicules. I'm calling them spicules, but you know what I mean. The tendrils
Yeah.
are being eaten off, and then I think the sponge. You know, actually, hold on a second. In the shallows, when you see predation and corallivory, you often see algae come in and colonize right afterwards, right? And that's partially how you tell how recently something was raised, because you see something come in, right? You know something about algal growth. These obviously are hydroids, or whatever it is coming in afterwards, but maybe it's just dead sponge tissue. That's my question. Steve, is
Yeah.
something coming in and
Yeah.
recolonizing this dead other hydroids, or is that just
Something
dead
will,
tissue?
yeah, something will.
But is it on the top up there on the brown section, is it are those hydroids, or is that just dead sponge?
I mean, there could be hydroids there. Let's see if we can
I can't tell.
see anything.
You see the difference between this side over
Yeah,
here.
yeah.
and this over there. That's all I was remarking about. Is that I don't know if they're eating the structure of the sponge.
Yeah, I mean, I would
Just
call
spicules or hydroids.
maybe some of these hydroids, but they're very, very small.
Right, but then they're coming in. If we knew anything about the rate of hydroid growth, which we don't, you might be able to get a sense of how long it might have been since this part of the sponge was killed. That's kind of amazing. See, look at all the. Yeah. Do they fly? Do they just climb up and not start eating until they're on the top? What are they doing?
Yeah, that is interesting. They're like selectively feeding.
Yeah, you don't see a lawnmower path up the side.
No.
Or I mean, probably sounds improbable. Like they could have just been there that long. The sponge had regrown at the bottom.
Could.
I mean,
That's
it
a
is a
lot,
long
yeah.
process. We know that.
Yeah.
Yeah, that would be pretty extraordinary longevity and patience on the part of the sea stars.
Where else you gotta go?
That keeps it alive.
Yeah.
Well, I suggest we take a star and some sponge. But
Let's see if we can get at this sea star and see if it's got its stomach averted in the background here.
yeah, that's a good thing to find.
It's the only one we can see right now. And it's maybe is the answer. I don't know what that is in the background. It looks, I don't know.
Crinoid? I
No,
think
no,
it-
the
Oh,
the star. It looks
yeah.
it's like something I haven't seen before.
Maybe we can do a look around the other side of the sponge, and then so we can see the stomachs of the ones that we see, and we can see what the other one is.
Let's do that. Rotate around.
You want to take a sample from this side first?
I think we're gonna want to image it first, and then yeah, we might be here a few minutes before we disturb it and the things start falling off.
Whoa. Is that kind of like collapsed or splayed open, or?
Yeah, it might be open. Yeah.
Every time I think, yeah, I know we're gonna get to see by the time we reach the top, more of the same. I get, like I'm totally humbled. We find something new. How are you doing?
I'm good.
Oh, good. It might just be another one of those. I don't know. I don't see that other star. There it is.
Yeah. This looks most similar to the star we saw on the sponge
I
in
agree.
our first collection, but I can't be absolute about that.
I think we're gonna have to take one of each star and one of each star and a piece of sponge. We don't have to take all three of the pieces of sponge, but one, two, three.
Okay. Quiver for everything in one?
Yes.
Okay.
Could be a biobox, quiver, whatever. Oh, we don't have a biobox.
Yeah, we had to burn it on the last collection.
Quiver's good.
Quiver. Okay.
All right, I'm gonna go
So-
talk to Eric. But do you need me?
No. 1:30. Yeah. Yes or no?
Find me before you. Where are we on our
Well,
track?
that'll probably take us to pretty close to the edge of this collection. We are
Yay.
at waypoint 5 roughly.
No, I think we should. Let's skip the collection and keep moving. We've got
Okay.
the imagery.
Okay.
Let's keep going.
All right. We are gonna maybe do pick the spongivory button one more time on this side, and we'll hit it twice. Once.
Sample Institute?
No, you want
Ivory?
the ivory. Yep.
Yeah.
For sponge.
So we'll do a close in on the spongivory with this. So hit spongivory and then gonna
It's submit. That's it.
image.
Yep.
It's coming around. It's coming around. All right. Hit spongivory now and submit.
Yep.
And then hit it again, and I'm gonna do a specimen shot from this side.
Oh, perfect.
I'm gonna eat it now.
No, yeah. And then lasers. Can we put lasers back on?
And I'm looking at Kai's question, and Kai, you and I are thinking the same way. I don't know, but it's a question. It's a good question. Okay. I will
There we
lead you
go.
through it. Come find me before you. I'll come back in at 1:30 before we do anything. Okay. See.
Yep, I will do so. All right, we got what we wanted here. We'll keep going. Not collecting, yeah. Sorry. There's another instance. Maybe we could just do a quick zoom as we go by on that star. Yeah, so this looks similar to the last one, but this one is just starting it. Perhaps its trek.
The hairs are much longer on this one.
Yeah, I wonder if that's a deterrent or if it's a squat lobster in this one too. Ophiurites. I'm wondering if they, maybe they start by grazing a foothold or armhold, I guess. They start grazing that way. It was really bizarre to see them on the top 2/3 of the colony, or sponge. Yeah, that's right. I don't think we need to set down again like the last one, but
I was just trying to see if you could see the underside of the structure.
yeah. I wonder if it's I don't know, maybe they just don't need to avert their stomachs as much when they're feeding on sponges, but it seems like on corals, they really put their stomach around the entire branch of the coral or whatever they're feeding on. I feel like there were more dead sponge bases in the more gentle slopes, where here there tends to be a little bit
Easier to get to.
Yeah, easier to get to, or maybe just like juicier feeding because the sponges grow bigger on these hills. One of the few fish we've been seeing on this dive are these cutthroat eels. Seem to be all over the place now.
So, one thing I was wondering about, like, in my tank at home, like, if the parameters get bad, right, and, like, corals aren't doing good, or the anemones aren't doing good, then all of a sudden my cleaner shrimp will start attacking them.
Yeah.
You think they, like, if the water changes around here, or heat or whatever,
Yeah.
that's what makes them get attracted to it, because they can sense that decay of them
It's an interesting idea.
Because y'all were saying that some areas you see a lot of them
Yeah.
eating the coral or whatnot, and there are other areas not as much.
Yeah.
Could it just be a sign of stress in that whole area and just attracts everybody to come there?
It could be. It could. I always thought of it in terms of like cost-benefit analysis. Like, do you really want to track out, to the one maybe big?
What is
What's
it
the
called?
gelatinous thing in there?
Yeah.
And there's a big like gelatinous pyrosome. Or, just want to double check if that's a pyrosome or something. Maybe it's some sort of salp. It screams pyrosome though. Just got hung up on this colony. Pyrosome?
That's my first assumption, but
yeah. Pyrosomes are pelagic tunicates, and this one looks like it, but I've never seen one this long and skinny before.
It's also holding together really well.
Yeah. It's just, maybe it's not. It's being.
Oh yeah, it's got to be a pyrosome.
Just a little bit more.
Sure. Oh yeah. You want me to zoom out? I mean, that could be something like a Thanksgiving dinner for a coral, because it could be eating that pyrosome if it has the capacity to eat the pyrosome for many months to years.
What looks like one of those jellies on there?
Is there a corallivorous jelly as well?
The pyrosome thing? The tube?
No, that on one of the
Yeah.
olives, there's a little red dot.
Yeah, I took it for an anemone. It was on kind of this side in this area.
I saw what I thought was an anemone as well.
Right here, in fact.
That's fine.
Sorry, I'm just running in for no reason.
No, we need to find one of those. So.
Oh,
We need
there
more
goes the
for species
pyrosome.
description.
No, I mean the fact that it landed on it is happen chance, really.
Is that the little red thing you want to look at?
Yeah, I think so.
It left now.
Sorry, I didn't see it.
The pyrosome.
We blew it out though.
I thought it was a jelly, but I think
That's anemone.
Don't see that every day.
All right. Yeah, keep going. Yeah, we see this a lot. So a few years ago, I was up on the Pacific Northwest during a particularly bad pyrosome year, and they're also called sea pickles, I think, in some places. But yeah, they have some resistance to them if you try to squeeze it, and they were getting hung up on everything. Like they were being torn to shreds by crabs and like massive food pulse to the deep sea. Very nutritious too. But they were so bad that you launch the vehicle and then they'd end up in like weird places all over the vehicle, lodged in boxes where you never even thought they would get in, and had it, they stink when they start to rot, so you have to just toss them. Some more, yeah, huge. That one was bigger than the last one, yeah.
I would definitely say so.
As interesting as it is that how long they are, I'm really impressed at the thickness of the axis, those curly part of the skeleton. Usually it's very fine, and I wonder if the rigidity, from being more calcified, it has some sort of benefit here where they have fast currents. Maybe it's more beneficial to be more stable than more up in the flow.
Doesn't the curl also help from the vibrations as well?
I bet it does. I bet it does.
Did they do that with
Yeah.
our antennas? Did they start putting that curly stuff on it? Does it keep it
from vibrating as much?
Could be, yeah. Yeah, it's, stability. Stability is important. Trying to, these are small polyps and the current's moving at, fractions of a meter per second, but still, these tiny polyps, which are about the size of a grain of rice, need to slow down the water. So they have lots and lots of branches to help them slow down the water. Can we look at the spot lobster on that one over there? That in the sediment, or not on the sediment, but in the rock. Purple spot lobster there.
In
the
band?
coral, yeah. Just a zoom in and out on it for ID.
Just see Pam.
This is one of the spiny ones. Oh, sure.
Is that what you're looking for?
No, I'm just getting a zoom on this for Tim, since I think this is the first one of these that we've seen on the dive, so he could get an ID. But we did sample one of these chirostylids, spiky ones. I think that's good.
Happy?
Yeah, happy, let's go. So far, we haven't seen many more transitions in the faunal community in this area after we've come up part of a steep slope. It's been pretty consistent for the past few hours, with a couple of primnoids, Norella, as well as these chrysogorgids, black corals, and Elopsamia mostly. A squat lobster or ophiurid on a rock. But everything seems to be pretty much on the available hard substrate. There's a few sea pens here and there that we've been seeing. Stichopathes probably is one of the most abundant, these wiry black corals.
Hey Ollie, do you think you can go about 0.5? Awesome, thank you.
I think we can do a white balance one more time when you have a chance.
I'm really high too.
Oh, okay.
If I get it lower,
Yeah, it's a little bit better.
little red, huh? Or is it? Might take you in white out?
No, it looks okay to me. Yeah, it's yeah the height.
I was up high, wait for the possible. Get ahead of us a little bit more.
So I'm curious, what's the like? Is there a maximum number of lights you would put? Is it a cost thing that kind of limits how much light you put on the vehicle? Is it a space thing?
Ultimately, I mean, you would get to a current issue.
Yeah.
Not able to supply that current.
Yeah.
Thing to get.
Yeah. Yeah, I've always wondered that. I guess it's more about the strategic placement of the lights than just the power, brute force.
And also being able to dim them helps out a lot. It's over here while we move.
But it's pretty clear. Yeah, we're having kind of a north to south current movement, and that it's accelerating over the terrain here fairly consistently. All the colonies are kind of streamlined, moving off in one direction. The sponges are pointing in one direction. All the iridogores here are streaming out in one direction. So when the corals and sponges are usually unidirectional, it gives some idea of the consistency of the current. So if you have variable currents, you might have colonies that are oriented many different ways.
Yeah.
You know, they don't know which way to grow to best take advantage of the food. But when you have consistent current directions, you know, over years or decades, you get growth like this.
Because these are the different ones. Those are it's just like the one that you picked up yesterday.
The sponges?
Yeah, the sponge with the spines.
It's very similar. I think it's a little bit different. They're both in the same family, but this is a different genus, I think. But it'd be very curious to see.
It's branches, like there's a branch on the spines.
Yeah. Yeah,
And
there
a
was
shrimp inside.
Mm-hmm. There are fewer and fewer sponge experts these days. There was the community lost a great sponge taxonomist in the past year, and it's being felt by everyone who has questions about what kinds of sponges they're seeing. In fact, actually, a couple of great sponge taxonomists have passed in the past couple of years. So, yeah, it's one of these difficulties where we have to try and get and pass down information from generation to generation, before things like this happen. And sometimes these types of scientific endeavors are not always the most well prioritized, but everyone really needs to know what these species are, and it's one of the problems now is we're trying to build taxonomic expertise based on new techniques, as well as trying to resolve some of the relationships between new techniques and the old morphological taxonomic techniques. But genetics and genomics are the way forward. It's getting cheaper and cheaper to learn about what these animals look like on the level of their DNA, which helps us understand how they might grow or how they might function, what environments they might live in. Can I tilt up a little bit? I mean, there's interesting stuff here. I'm keeping my eye out. There's a lot of sea pens. I'm just curious about getting it into view and seeing it ahead, so I can give you plenty of notice if there's anything we might want to look at. Bathies with a couple of brittle stars.
Yeah.
So it looks like we kind of jumped down, or the ridge is drifting under us.
This while I was waiting for the birds to go, because all that following that track line was
Yeah.
gonna be all sand.
Yeah,
I found a little
that's a
rock
good-
outcrop over here to lay it while we wait.
Good plan. Yeah. Surface current's pretty bad, or is it just
They seem pretty strong.
consistent? Yeah.
They're consistent. Hideaways down too.
Are
Very
these the
nice.
sea pans? Or black corals?
Are those sea pens or black corals?
Those are sea pens. I think those are Polypterus, or one of the close relatives of Polypterus. That looks very similar. Very spiny. We sampled a couple of those in the Line Islands a couple of years ago, and they have very sharp spines in the back of the polyp. And yeah, they'll puncture a glove. Yeah. They're very hard too. They're not squishy like some of the other sea pens. They have a lot of resistance. Can you give me an estimate of how long of a distance between where we are now and the end of our dive track? 450?
415.
415.
Yes, sir. Ship's cruising at a good speed now, so we can pick up some track quickly.
Yeah, it's mostly similar stuff here, so I won't stop to zoom in anything, but it'd be good to see something closer to the. I won't stop to zoom in anything, so we can cruise a little bit.
Sorry, 131. May I impose upon you to show me how far away we are from the end point?
415 meters.
Can you put off for a minute?
So we got a
You're holding good. And let's bump it up to a
15, 20 minute, maybe half an hour extension on today's dive, but we are approaching our end point.
If we start getting pulled or the boat goes a little wacky, we'll let you know and so okay. Thank you all.
So what we're going to do is we evaluate like every 15, 20 minutes. I'll just keep popping in and see where we are. And then you can call when we have to. But I think we'd love to see the rest of the map.
Well, the ship's about to, he's gonna stop moving starboard. We'll move forward at 0.7, which is gonna speed him up, which will speed us up.
Beautiful.
Which means we, right
Shot
now,
engaged.
just from talking, we're at 350.
Yeah, that's true. Like, we keep on seeing the same things.
Him going 0.7, us trailing.
All right. That's it. Good job. Go team. I'll see you in 15, 20 minutes.
You get closer from you.
I am just waiting for him to get ahead of me.
Yeah,
It's
sure.
a crinoid.
Yeah, we got a crinoid. This one is Phrynocrinus or Phrynocrinidae in that family. I think it's a relatively new genus too. Recently described in the past few years. Or maybe it hasn't been described yet. I haven't seen any news on it, but that is what I know about this crinoid. Possibly some Aplacophorine grazers or something on this. These crinoids often have polychaetes that are associated. They graze down some of these pinnules on the branch tips. You can see there's a little bit of regrowth, in fact, on some of these branches.
For you to end up at. So just cruise straight for that waypoint in that vicinity. Sounds good. Thank you. We do that? No. They don't know yet?
I'm gonna let my mom out there watering the plant, so.
They know that there's a new thing. Always makes me all the time, go in the doorbell and sit there. Once a week.
Sea star.
Yep. Looks like one of those sea stars that was grazing that sponge earlier. Of course, not on the sponge, but. Tilt up a tick. Okay, there. There's a few more here. This is the land of the echinoderms. That and Iridogorgia. I think Iridogorgia might represent like one of the most abundant corals in the Pacific. I wouldn't be surprised, particularly on these seamounts. In some places they can be ultra dense. But it's pretty sparse here. Interestingly, in all the places I've done research on deep water corals in the Pacific, the only place in the Pacific that I have not seen them, they're completely absent, is off of the eastern tropical Pacific. Haven't seen any in the waters around Costa Rica from the Falkor cruise video that I was looking at in 2019. That may have something to do with the environment there, the oceanography. It's a little bit less well oxygenated. It's a prominent oxygen minimum zone in the deep sea, and that may be something that reduces their ability to settle there, or perhaps their substrate requirements that are not satisfactory to the coral. A couple of anemones, shrimp. So when we get up on deck, we will be looking through our collections. First, workflow kind of goes like this. We'll get the vehicle up on deck. We'll start to set up the wet lab to keep our specimens cold, usually with an ice bath that we'll put all the quivers in. Then we'll, usually folks who need high priority collections, for example, if something's time sensitive, we'll grab a small piece of material from each of the samples and then we'll image all of this collection. We take pretty detailed imagery, mostly to document color on deck, because a lot of times when we preserve collections or tissues start to turn necrotic, we end up with discoloration, and we want to know what color it was on the seafloor as well as in the lab. And then each coral or sponge or specimen will get a variety of applications, from being used in onboard respiration experiments. We're also taking samples for microbial analysis of the corals and of their environment. We're also looking at some of the the genetic relationships between corals and associates, as well as the evolutionary relationships of the corals themselves, and trying to ID them using all of the tools we have available to us, both on the ship. We have a small microscope, but also back in the lab where we can sequence tissue. Fish. Halosaur, probably Elytravandia, a genus. We have seen mostly halosaurs, a few cusk eel, many synaphobranchid eels at this depth. A little bit different from if you had joined us a few days ago when we were in the much shallower waters where there was a lot of fish diversity.
Adam? Is that on here?
I don't know where that. It might be in the back, in the garage. It might be in the garage. No. I haven't seen him in a while. Alright, you want me to send him down to you? Yeah. Here's something. Okay, I'll send him down to you.
Found something
There you
new.
go.
Thanks, bro.
Bamboo? Bamboo coral. We saw a few of these down deeper, and just haven't seen any since we got on shift, but I don't think we need to stop. I think we'll just keep cruising.
Get a quick image of
Yeah.
the whole source.
Yeah, that's fine. It is a big colony. Probably been there for a while.
Also, after the dive today, we're gonna lose some internet
Are we?
for about an hour for system maintenance.
Oh, okay. I haven't seen anything about that. Oh, there it is.
about 150 meters to the end.
Yeah, great. Let's keep cruising.
You want to update your people on this map?
Yeah, I am communicating with them.
Make good time too.
Yeah, this is good. I mean, sometimes getting this type of information is useful. You know, quantitative. We got good zooms on almost everything so far. So this is just getting counts of things. One of the things that I've been trying to do, for the most part unsuccessfully, is try to get a better idea on how we can turn this count data, you know, counting corals on a transect, into some more spatially informative data, like density data, in terms of corals per square meter, or something a little bit more informative than corals per second or corals per minute. Because corals per square meter would give us an idea of some of the spatial heterogeneity, or how different densities of corals are across the seamount landscape. I think it peels off.
Yeah, well, it peels off to my right. I'm just looking at where you have the waypoint.
Nah, follow it up to the right. Yeah, follow your eyes. There's a little bit of, yeah. There was a little bit of issue with the bathy up at the top, so I. Yeah. There's it's not as precise. I don't know what the grid is, 50 or 25 meters.
Right now it's 10.
10 meter grid.
Yeah.
Okay. So it's probably, yeah.
Oh no, he's talking about.
Oh, the mapping grid, the bathy grid.
That's your top ours. Yeah, so make a decision. Don't mess at the time. We're gonna have to stop the ship here and then follow us again.
Or
Yeah.
I go over to that ridge over there. I might just have to go over some blue water real quick. Or I can see if it turns to my left, which it might do.
That's alright. You don't. Oh, yeah.
Yeah, I might be able to follow something that way. Yes. Yeah, it flattens out up here anyway, so I'll just cross over.
So we're basically within 100 meters of the endpoint, right? Yeah.
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 It's going that way.
We're not beholden to doing the entire length, so I just want to get confirmation from Randy before pulling off the bottom here. But I think we've pretty much satisfied our dive plan for the day. But I'm very intrigued about these Holypterus beds,
Yeah, like
these
70
sea pens.
meters before we're
It's about to get some rock.
Of course it is.
It's about to get good.
Of course it is. Xenophyophore bed there. Pretty dense too. These Holypterus are neat. Very tall, very skinny. That one looks pretty mangled actually. I've seen some Holypterus where they have been grazed before, and as something comes along and eats the entire polyp, but I've never seen, they're not able to graze down the entire sea pen because they have a fairly, there you go, big old fish. Some sort of ophidiid. That is a great fish shot. There he goes.
maybe the fishes like in the bottom, they're not colorful like in the ones like at the top.
Right.
Because it's so rocky, like they just being like those.
Yep, it helps. It certainly helps to make yourself invisible. You know, visually, you know, you wouldn't be able to see that very well on the seafloor. But also, you know, you have to consider that maybe animals down here are looking at light in different wavelengths than we perceive visible light. So they may be looking at, you know, ultraviolet or not ultraviolet, some sort of bioluminescent wavelength. But also, you know, from an energetic perspective, you know, food is pretty low quality compared to the shallow, you know, surface ocean, and pigments, especially in the tissue and the skin, it's very energetically demanding to produce. You know, when you're eating, you know, very small amounts of carbon. They're very long lived, of course, but you know, producing reds and yellows, while it may make you better at camouflage, it may actually be more energetically demanding.
What was the depth y'all trying to get to?
12, roughly 12 even.
Oh, this way, I like
So we're just about finished with our dive track, but it keeps going up, so I guess we'll just keep cruising.
Hey.
Yep. So we just finished our dive track, kind of parallel to it, following the ridge. $50 more.
It won't take long.
50. You want to go to 1,200 even? They
Sure.
said they
Sounds
have
good.
50 more meters up in the distance.
These crazy fish.
There
So.
are more fish up here. That's for
There's
sure.
a lot of fish up here.
Yeah.
Wow.
I wonder if it's a summit thing.
What kind of fish is that?
I've seen those before. I need to double check. Some sort of Bophidiid cuskeel or something.
We're hanging out together.
I usually rely on my fish colleagues,
Fish friends?
fish friends, but they're actually human.
So if you have been enjoying this dive with us, we are so glad you were able to join us. We are not quite done yet, but we're just about maybe a few minutes away from pulling off the seafloor. We have a weather window where it's really good, and we are going to head back east within the US EEZ and head to Taitov Seamount inside the United States Pacific Remote Islands Marine National Monument Howland of Baker Unit. The Seamount is a really large Giat, and it's super cool, and it's only had one dive on it, or maybe two so far, but it's got a lot of features that we haven't yet had the opportunity to explore, and we are really excited to be able to go back there, and it puts us back on the Tokelau Ridge, whereas right here we're off ridge and kind of getting in a comparison. And I would say that the exploration we've done on these seamounts in the past 2 days has been really exciting. I don't know, Steve, Anna Mary, what do you think?
Yeah,
Cool?
they're similar, but they're different. So it'll give us a lot to think about, comparing the two sites to each other, and then these sites to other sites within the monument.
Exactly. So we are going to be back on the seafloor probably, I would probably use 24, 25, 26 hours from now, in that range. It's about a 22-hour transit, but we've got to pull up now, and then we've got to get back down then. So I would say you're looking at a 24 to 26 hour break. Get some rest, get some sleep, see some sunshine, and we will see you on the seafloor again in about a day. I know, sunshine. We're all in this control room the whole time. You know, this has been a spectacular dive. And you never know what you're going to find. What is that? I don't even know. And we're just excited. Thank you so much for exploring with us today. I guess this wraps up our dive on this unnamed, unknown, unmapped, unseen before today seamount on the western portion of the United States exclusive economic zone. See you in a day. And now there's the awkward pause. What do we do now? All right. Bye, everyone.